Hi guys, hi everyone. Am I audible? Yes, please. Am I audible to everybody? Hi people. Hi everyone. Am I audible? So good evening everyone. Welcome to Unacademy NEET English. I hope you all are doing great and I hope I'm perfectly audible and visible to everybody. Good evening everyone. Good evening, good evening, good evening. So welcome to Game of NEET 3.0, right? Our one of the most successful series and obviously the most productive one, because here we complete each and every chapter in detail. We can include the NCERT as well. So here in this one session, you will get everything, the quality content, the questions, the PYQs, everything. So I hope you all are excited. So if you guys are, then quickly hit the like button, and I want to see some fire in the chat section, guys. Quick everybody. Yes, I really want to see some fire in the chat section, right? If you guys are actually excited for this particular session, then I really want to see that. Yes, please. Okay, no voice. Is that so? I guess I'm perfectly audible and visible to everybody. That's good, that's good. That's like my kids. That's like my kids. So who's ready to? Yeah, after a long time, actually after a long time again. So who's ready to rock it again? Yes? Acha, who's not going to take the class? Guys, even I am not well, but I'm not going to cancel my class because after so long, I am going to take the class and I'm really excited. And I want the same excitement in you people as well, because today in depth, we are going to complete the chapter number 1. We will have the MCQ practice, and today I'll tell you how to completely read the NCERT, like how to focus on the NCERT, and we are going to solve the exemplar questions as well. Continue. Pakka? Excited? Sure? Okay, so tell me to which region you guys belong. To which region you all belong? How long it's gonna take? How can you ask me this thing in the starting itself? Of course, this is a good chapter, lengthy chapter, important chapter. It will take time. Telangana, Tamil Nadu. Bangalore, Kashmir, Andhra Pradesh, Manipur. Okay. Okay, the people are there from the UP as well. That's good. That's good. Because I used to think that people from northeast and the southern part of the country, they watch our channel, but I'm happy. But I'm happy to see that. Karnataka, okay, Kashmir, Gujarat. Okay, so let's start the chapter. The name of the chapter is Human Reproduction, one of the easy chapters as well, right? And of course, we know that it is scoring as well. So let's get started. So today we have a lot of diagrams that we are going to use here in the lecture. And obviously you have to make the notes as well. And trust me, focus on the understanding part, because I'll share the complete PDF in our official Telegram group as well. Okay. So ready guys? Okay, so the name of the chapter, as you all know, the name of the chapter is Human Reproduction. So after long time, once again, Game of NEET 3.0, right? Done? Okay, so when we talk about the reproduction, so we all know it very well, a little bit of the intro, then I'll jump to the main topic directly. So we know it very well, re means again, and when it comes to the production, it means to produce. So basically what is happening here, students, the already existing individuals, what is happening here? Already existing individuals, they give birth to, they give birth to new organism. That is what the human reproduction is. Are you getting my point? This is what the human reproduction is, that already existing individuals, they are giving birth to the new individual, right, students? So now, because we are talking about the human reproduction, so we know it very well that in humans, what do we see? In humans, we see sexual dimorphism. What do we see, people? We see sexual dimorphism. Di means two, morph means form. So we know that there is a male and there is a female, and that we can differentiate because sexual dimorphism is there. You can look at them, right, and you can just simply tell, isn't it? Isn't it? So when it comes to the human, you know that we have the different systems, including the digestive system, excretory system, right, nervous system. So same way we have the reproductive system as well, in which all the events related to the reproduction will occur, isn't it? So we have the reproductive system. So before starting anything here, right, before starting anything here, what exactly we need to know? We need to know about the events of reproduction. So can you tell me about this? Can you tell me about this? What are these events of reproduction? I believe you people know this, right? I believe you know this. There is a simple trick for that. When it comes to the events of the reproduction, guys, there is one request, right? Please don't ask me the duration. You know that it's game of NEET 2.0, right? At least I'm going to take 8 hours, right? 8 hours. So in the starting itself, why are you asking about the duration? Let's focus on the topic. Let's focus on the understanding. Because if you guys will be like, how much time will it take? How much time am I going to attend? You're just going to break the flow. Let's have the focus on one thing, that is understanding the topic. That is it, right? You know that for that thing, for this content part, this game of NEET is famous. So let's not disrupt that, isn't it? Okay, so events of reproduction will include this trick. It's give, it's sip, and it's go. Repeat it in the chat section, everybody. It's give, it's sip, it's go. Repeat it quick. It's give, it's sip, it's give, it's sip, it's go. So what is this give, sip, and go? So what exactly is going to happen when we talk about the reproduction? Firstly, we will discuss gametogenesis, right? Then insemination is there. Fertilization, cleavage, implantation. Just listen to these words, these terms. Placentation, gastrulation, oogenesis. Then finally, parturition. Are you getting my point? That's what we have here. Okay, so these are the events here: gametogenesis, insemination, fertilization, cleavage, implantation, placentation, gastrulation, organogenesis, and parturition. So this is what we have to talk about. So the very first thing is gametogenesis. So you know that what is the meaning of the genesis. So genesis simply means formation, and here you're talking about the gamete formation. So in the starting itself, I told you very clearly, we have the male and the female here. So obviously in the case of males, there will be spermatogenesis, and in the case of females, there will be oogenesis. Okay, now the male gametes, they'll be transferred to the female genital tract. We call it as insemination. How will it be done? With the help of male copulatory organ, that is penis. Then comes the fertilization, male and the female gamete. When the female gamete is also there, is also released, and the male gamete is also there, then obviously the fertilization is going to take place. After the fertilization, zygote formation. So zygote will undergo cleavage, then implantation will occur. After the implantation, there will be, right, that what is the meaning of the implantation there, guys? So implantation means the zygote, when it will grow, it is going to form the blastocyst. Like, it will not grow in the size initially, right? The cell number will increase, of course. The blastocyst formation will be there. That blastocyst will get implanted in the uterus, right? And after that, the placenta formation will be there. Then further growth, the gastrula formation will be there. The germ layers will form. Finally, the organs will form. And when it comes to the parturition, so what is the meaning of parturition, everybody? Yes, what is the meaning of parturition? Quick. What is the meaning of parturition? What do you understand with the word parturition, guys? Quick. Parturition stands for? Yes, please. Parturition stands for? Yep. What is the definition for this? And I'm pretty sure you are aware of it. Exactly. It is the childbirth. Okay. So it is the childbirth. So this is what we discuss in the events of the reproduction. Okay. So before starting the gametogenesis, we need to know about the systems. And the very first system that we are going to talk is, it is the male reproductive system. Are you getting my point? The very first thing we are going to discuss is male reproductive system. Ready? This is what we are going to discuss first, right? Right? Male reproductive system. So let's get started. Right, beta? So when it comes to the male reproductive system, so just have a look of this diagram. Of course, this is from the NCERT. Right, let's understand few words. And I have many diagrams here. So first of all, just have a look. When you talk about the male reproductive system, we will discuss it. Firstly, I'll tell you a few words. Here you can see this is the male copulatory organ and this is the penis. What is it? It is the penis. Now, when you look at this pouch-like structure, can you see that? This pouch-like structure in which testes are placed, this is the scrotum. This deeply pigmented, having sebaceous glands. So this pouch-like structure is what? It is the scrotum. Now, in the scrotum, can you see this part? Can you see this part, everybody? Do let me know in the chat section. So this part that we are talking here, right, this is what? It is the testis. Can you see that? Right? The part that we can see here, this part I'm talking about, this is the testis. Okay? So within the testis, yes, please, within the testis, can you see this? These are the seminiferous tubules. They are forming a network here. Retitestis, right, student? Here you guys are having, we'll talk about the vasa efferentia as well, one by one. So here, the things that are visible to you, firstly, I'm talking about that. This is the epididymis. What's that? This is the epididymis. And then here you people can see this is the vas deferens. So that is how it is. So this diagram will make it more clear. So here you guys can see, right, this image is from the NCERT. This is basically the male copulatory organ, penis. So I told you about a pouch-like structure, scrotum. In that scrotum, you'll see the pair of testes here. The part which is located here, this particular part I'm talking, students, this is the epididymis. And here you people can see what? It is the vas deferens. Can you see that? It is the vas deferens, pair of vas deferens. So it is coiling over the urinary bladder, right? It is receiving the secretions from these glands. It is forming the ejaculatory duct. It is further receiving the secretion from this prostate gland. This is the seminal vesicle. And finally, it will open here in the penis. So when we talk about the glands here, these are the three names that we have to remember: seminal vesicle, prostate, and bulbourethral glands, also known as Cowper's gland. Are you getting my point? So that is what we have to and we are going to discuss. Okay? Okay? Right, so one by one we are going to talk about these parts, right? So here you guys can see the testes, because they are in the scrotum, they are not in the abdomen, so they are extra abdominal. What do we call them, students? We call them as extra abdominal. Can you see the structure? Can you see the structure? Firstly, understand the location, understand everything related to that, then we will discuss it. So you guys can see again, this is the male copulatory organ, penis, meant for the insemination, and obviously urine also passes through it, right? Then here you guys can see this pouch-like scrotum. The testis, epididymis, vas deferens, that is going to coil over the urinary bladder. This is the seminal vesicle here. Yeah, so seminal vesicle here, then this is the prostate gland here, then here you have the bulbourethral or Cowper's gland. That is how it is. Location, position, clear? Yes? Done, beta? Sure? So whenever you talk about the reproductive system, be it the male reproductive system or the female reproductive system, the very first thing that we have to talk about is primary sex organs, right? I'll give you the overview, then we'll move to the exact topic, right? So what we need to know that yeah, so initially, what are we going to discuss? We'll be talking about the primary sex organs first, right? And then we are going to talk about the secondary sex organs. So you tell me, what do you understand with the word primary sex organs? Yes, please. Yes, please. Anybody? Yes, please. Quick. Primary sex organs. Is there internet glitch or something? Because I cannot see the answers here. Not happy with that. So when it comes to the primary sex organs, what is going to happen? What are the primary sex organs? So primary sex organs are the one in which gamete formation occurs. What are they? In which gamete formation occurs. That is what the primary sex organs are. So here in the case of male, this sign is for the male. We know that that will be testis, right? Pair of testis. And in the case of females, that are going to be ovaries, pair of ovaries there, right? So you know that in the testis there is the formation of sperm. Sperm is a plural, and here there is the formation of ova. This is what we know. So when it comes to the secondary sex organ, right? When it comes to what? When it comes to the secondary sex organ. So primary sex organs are the one that are basically going to form gametes. They also release the primary sex hormones, right? Like in the case of females, we talk about the estrogen, we talk about the progesterone. In the case of males, you talk about the testosterone. And when it comes to the secondary sex organ, right? So here you are talking about the organs which will help for the passage of the gametes, passage of the testes, and that helps in the union of these male and female gamete, right? So that is what the secondary sex organs are. What are that secondary sex organs? So they are just form of passage for the gametes, right? To help the union of, form the passage of gametes, to help the union of male and female gametes, right? To help the union of male and female gametes. So that is how the secondary sex organs are going to help. So in the case of males, this epididymis, vasa efferentia, vasa differentia, etc., we are going to discuss. In the females, fallopian tube, uterus, etc., we discuss. Is that clear? Is that clear? Yeah. So secondary sex organs, they are very important for the reproduction. Okay. Now, when we talk about the male reproductive system, so basically I clearly mentioned that the testis, right? This is what I have mentioned. The testis, they are the primary sex organs. And the second point that I'm mentioning here clearly is that these testes are what? They are extra-abdominal. So what is the meaning of being extra-abdominal? They are not the part of abdomen. This is what I'm trying to tell you, right? That they are not the part of abdomen. They are not present in the abdomen. Actually, what is happening? Like when a baby boy develops in the mother's womb, during the, you know that in the case of humans, the gestation period is for the 9 months. I hope you all are well aware, right? When it comes to the gestation period, it is for the 9 months. So during the development of that baby, when there is a 7th month, right? The development is during that 7th month. Till then, this actually initially, basically this is what I can say, that initially these testes, they are abdominal only. Initially the testis formation is in the abdomen only, but during the development part, during the 7th month of the pregnancy, these testes, right, these testes, so I'm writing it here as well. So during 7th month of human pregnancy, they descend into scrotum through inguinal canal. I'll tell you the reason and I'll tell you what this inguinal canal is, right? Along with the spermatic cord. So this is going to happen here. So I'll show you the diagram here, students. Yes. So let's suppose this is what the scrotum is, that pigmented pouch-like structure. Now the scrotum, like these testes, they are connected to the abdominal part as well. Actually, here you see a kind of canal. That is the inguinal, not the kind of a canal itself. So through which the testes, they descend into the scrotum. Right? They descend into what? They descend into the scrotum. You people can see this part here. Right? You guys can see this part here. Can you see the inguinal ligaments here? Yes, everybody. Can you see the inguinal canal here? This part they are marking. See? Right? They are just trying to show you this. Right? They are just trying to show you this. This part they are just trying to show you. So basically, this is what? This is the inguinal canal. You can see the better diagram here. I hope it is clearly visible to all of you. Right? These are the testes. Right? They are showing this part. Can you see that, students? See, inguinal ligament, rings. So that is how it is. Okay, that is how it is. So they form in the testes, they develop in the abdomen only, but during the 7th month of the pregnancy, they'll descend down into the scrotum. Right? They'll descend down into the scrotum. Through what? Through this inguinal canal. Is that clear? Yes, please. Is that clear? Is that clear? Right? So through this inguinal canal, they are going to descend into the scrotum. Done, beta? Done? So they will descend into the scrotum. But now my point is, why is it so? Right? Why is it so? Why scrotum? Why not in the abdomen? Anybody? Yes? Anybody? Yes, please. Quickly. Yes, please. Anybody? Why scrotum? Anybody? Beta Fatima, there is no need to think, right? What is required, what is not. At this point of time, it is important. Kindly note down. Anybody? Temperature? Yes. Elaborate a bit. So basically, what is going to happen? Why there is the requirement? So you know that what is the function of the testis? The function of the testis is to make the gamete. It is the primary sex organ. It has to make the gamete. Now when it comes to the gametes, right, there are gamete forming cells, spermatogonia. They are temperature sensitive. They need a specific temperature. So what's going to happen? These testes, they will descend into a pouch-like structure. We will discuss the details of that as well, right? So through inguinal canal, they'll descend into the scrotum. They'll descend into what? They'll descend into the scrotum. Because the scrotum, when we talk about the scrotum, it is darkly pigmented, having sebaceous gland, having cremaster muscles, having dartus muscles, right, to regulate the temperature, to maintain the temperature. So scrotum ultimately, it maintains the temperature for sperm production, because the cell that forms the sperm, right, the spermatogonia, is temperature sensitive, right? So here in scrotum, the temperature is, you'll get the written points, don't worry, 2 to 2.5 degrees Celsius lesser than the body temperature, right? Lesser than the body temperature. So this is what the role of scrotum is. So let's suppose if your body temperature is 37 degrees Celsius, so in the scrotum it will be around 35 degrees Celsius. It is like that. Are you getting my point? And why it is required? It is required for the sperm production, because the cells that form the sperm, they are the temperature sensitive cells. They need a particular temperature. That is why they descend into the scrotum. So scrotum, this is the structure. This is that pouch-like structure that we are talking. This is the scrotum part here. Is that clear? This is the scrotum part here. Is that clear? So you people can see the external view of the scrotum. Here you guys can see, right, there is a raphe which is dividing it like this. Now, these are the muscles that we are going to discuss, cremaster and dartus muscle. Because now I will tell you that how the scrotum is going to maintain the temperature. The first thing that we have learned is that testes will be there in the scrotum, because scrotum maintains the temperature required for the sperm production. Now we will discuss that how is it going to maintain it. Is that clear? And see in this diagram itself, can you see this canal? They are showing that inguinal canal having spermatic cord, that is the connective tissue part, of course, having, you know, arteries, veins, even the vas deferens is the part here. And here we'll be talking about certain muscles. So what are the muscles that we are going to discuss, students? We will be talking about the cremaster muscle and the dartus muscle. So let's get started. Let's discuss it to the point so that we can save our time. So scrotum, as I said, it's a pouch-like structure, it's a sac-like structure. Everybody, you have to repeat it with me, okay? You have to participate in the discussion. You will get the PDF. Don't worry about that. You have to participate here in the discussion. And last time, right, some internet issue occurred in the human health and disease part. If you will check our channel, you won't get the detailed one shot for the human health and disease. It is in the parts. So after this human reproduction chapter, I have kept the chapter human health and disease. Okay. Okay, so get ready. Okay, so I'm going to target all your important chapters at the first place. Beta, my Telegram group is Love Biology by Ambika. Okay. So, Hassan, I don't understand your question. Let me tell you that baby is developing in the mother's womb. And that baby boy, that will have the scrotum. So, in that baby's right abdomen, initially the testes they develop. Then they descend into the scrotum. Are you getting my point? Mr. Hasini Yadav, I hope it is clear to you. Right? So this is what you have to keep in your mind. So scrotum is a sac-like or the pouch-like structure, right? So as I said, it is divided into two separate compartments by a scrotal septum. So each compartment will contain one testis, told you already. Up to this part, all clear? Then internally, this scrotal sac is lined by two types of muscles, the dartus muscle and the cremaster muscle, right? The dartus muscle and the cremaster muscle. Is that clear? Is that clear? So there will be the dartus muscle, and after the dartus muscle, there will be the cremaster muscle. So let's talk about it. So when you talk about the dartus muscle, it is a smooth muscle. Now, if it is a smooth muscle, and where is it located? Like, let's suppose this is your scrotum, right? Here, just below the skin here, you are going to find the dartus muscle. Just below the skin. Which muscles are you going to find? You are going to find the dartus muscles. So these muscles are what? They are smooth muscles. Now you tell me, if it is a smooth muscle, is it going to be voluntary or involuntary? Yes, please. Is it going to be voluntary or involuntary? Is it going to be voluntary or involuntary? Yes, please. Yes, please. Voluntary or involuntary, guys? Session should be interactive, so be fast. Of course, these muscles are going to be involuntary. So obviously they are under the control of autonomic nervous system. I'll tell you their role. Firstly, you understand their location, right? Understand their location. So this is what we have initially. So we will see that we have the dartus muscle, and the second thing that you have here is the cremaster muscle. What do you have, students? What do you have, students? Cremaster muscles, right? Right? We'll show you the location as well. So dartus muscle, because it is a smooth muscle, it is involuntary. You cannot control it with your own will. So it is in the wall of the scrotum, just beneath the skin. Okay? Okay, so how is it going to work? See, it is just beneath that scrotal skin. So basically, it is involuntary. You cannot control it with your own will. So when it contracts, right? When it contracts, it will cause the wrinkling of the scrotal skin, and it will contract during cold, right? Right. Let me write it here. So dartus muscles. So they are present in scrotum, just beneath the skin. Number one. Number two, they are involuntary. So basically, smooth muscles they are. Contracts during cold, cause wrinkling of scrotal skin, and then reduces surface area and retain heat. That's how they maintain the temperature. Okay, then it will do what? It will retain the heat. I hope you are making the notes as well. Okay, so contracts during cold, reduces the surface area so that it can retain the heat, it can minimize the heat loss. Okay, right? And then obviously during warm, it relaxes, makes scrotal skin smooth, and due to which what's going to happen, students? When the scrotum will be smooth, the heat loss increases. So, and in the warm season, obviously this is what is required. So that is how the dartus muscle help. The first point clear? The first point clear? Now the next point is cremaster muscles. Now cremaster muscles, where will you find it? You will find it in the wall of the, right? Where will you find it? In the wall of scrotum and in inguinal canal, right? So it will basically help in descending the testes as well as per the temperature. Done, beta? Is that clear? Is that clear? Is that clear? Yes or no? Yes or no? So these muscles, right? These muscles, they are basically, yes please, they are basically, they lift or lower the testes. What will they do? They will lift or lower the testes. Okay, so cremaster muscles are what? They are not involuntary, they are skeletal muscle. Like abdomen is having some internal oblique muscles, na? They are formed from that. So they are the skeletal muscle. So if it is skeletal, you know that it is going to be what? It is going to be voluntary. It is going to be what? It is going to be voluntary. So this is what you have to keep in your mind. Is that clear? Is that clear? So wall of the scrotum and in the inguinal canal they are present around the spermatic cord. So you imagine this is a canal through which the testes descend. So here you have the muscles, here you have the spermatic cord as well. So I hope you know what the spermatic cord is. This is nothing but it is the connective tissue. Okay, it is the connective tissue. Okay, in the chapter structure, right? In the chapter structural organization in animals, we discuss about the different type of connective tissue. I hope you remember. We talk about the dense connective tissue, loose connective tissue, dense regular connective tissue, dense irregular connective tissue, and we also talk about the cord, the sheath. So this is what we are talking here. The spermatic cord is there, having the arteries, veins, everything. So here you will see the cremaster muscles. So they will help in, you know, lifting or lowering the testes. Like let's suppose when the temperature is down, what will happen? These cremaster muscles, they will bring that testes closer to the pelvic region so that they can absorb the heat. Are you getting my point? So that they can absorb the heat. Are you getting my point? And when the temperature, right, when the temperature is not that low, when the temperature is warm, again the testes will, right, they'll descend, then they will not be that closer to the pelvic cavity, right, they'll be down. Are you getting my point? So that is how when we say the lifting or the lowering the testes. Are you getting my point? So basically you'll see this, in the case of, in the pouch-like structure, in the scrotum only, right? In the scrotum only, just below the skin, this dartus muscle is there, and I told you where you have the cremaster muscle. So any doubt from this part? Yes, please. Any doubt from this part? So they surround the spermatic cord as well as the testes. Okay? Okay? Yes, please. Any doubt? Sure? So where are they present? Around testes, this is how you can write. Right? This is how you can write that they are present around spermatic cord and the testis. Right? And testis. Contracts, pull testis closer to body during cold, and again relax during warm conditions. away from body. So that is how it helps. That is how they regulate the temperature. Moreover, when you talk about the scrotal skin, as you know, darkly pigmented, plus it is having the sebaceous glands as well, sweat glands as well. You know that sweating will also be there. That will also sweating after sweating also, you know that the cooling will be there. That is also that is how it maintains the temperature. So it is very important to focus on this part, because if the temperature will be inappropriate, then the sperm production is affected. That is why males, they are advised not to take bath with too much of cold water, number one, right? They should not wear very tight jeans, because obviously if they will be very tight, then the sweating in that region will be there. That can also affect the muscle production, not the muscle, sperm production, right? Plus if you know, males, when they work in the industries where there are the temperature is high, like somewhere near to the heat or something, right, near to the furnaces or something like that, in that case also their sperm production, it gets affected. Are you getting my point? Are you getting my point? So this is what you have to understand. That is how they regulate the temperature. Okay, so again and again I'm using the word spermatic cord as well. As I said, this is the connective tissue part, and moreover spermatic fascia is also there in the inguinal canal, students. That is the connective tissue surrounding testis and that spermatic cord. Is that clear? And moreover, in the scrotum skin, many pigment cells, right, the melanin cells, which will give it the darker color, they are also present. That is why the scrotal skin is you know, dark in comparison to the other regions of the body, right? And as I said, sebaceous gland and sweat glands are also there, right? They also help in regulating the temperature. That is it. Okay. Okay, so here I have even mentioned, right, that how these muscles, they help, though we have though I have mentioned the entire all the things here, so you guys can note down. Okay, okay, done. So testes, they descend into the scrotum. So if by any chance, if testes, they do not descend into scrotum, then this condition is known as cryptorchidism. Write down this word, everybody. This is important. PYQ. Yes, please go for it. Beta, it is for the NEET. You want to write it, you can. If you don't want to, your choice. I don't know, after checking NEET 2025 paper, I really don't understand how can students type such kind of things. They are still not focused on the concept part, isn't it? Right, so this part is clear. So now let's talk about the testis. So here you people can see the spermatic cord, right, surrounding it is also having the spermatic fascia. Here you'll see the, as I said, the gonadal arteries, veins, even the vas deferens is also present here, right? So this is what you people can see. The look at the position of the cremaster muscle there. Okay, just look at it. Done, students. So next part that we have to discuss is testis part. So any doubts? Any doubts? So I hope you have mentioned it. It is important. Okay, so whenever there is a premature baby birth, especially boy, so in that case, doctors, right, they make sure that the testes, they descend into the scrotum or not. They check it, they press it, they even feel it, they even check it that whether testes are there in the scrotum or not. Okay, okay, done. Chaliye. So next point that we have to discuss is, let's talk about the testis part. Done, students. Let's talk about testis. Done. Up to this part, all clear? If there is any doubt, do let me know. Do let me know. So before that, mention this thing. Scrotum is connected to the abdominal cavity. This is scrotum is connected to abdominal cavity by dash. Answer this, everybody, quick, and mention it in your notes as well. Scrotum is connected to abdominal cavity by dash. Yes, please. Yes, please. Yes, please. Quick. Excellent, Ritesh. Miss Chino, sorry, not Chino, Miss Echinodermata. Very good, Mobina. Exactly. So the answer is going to be inguinal canal. So please write it down in your notes. Quick. So next part is testes suspend into scrotum by dash. Testes suspend in the scrotum by the dash. Yes? Quick. Everybody quick. Guys, here the answer is going to be spermatic cord. What is going to be the answer here? It is going to be spermatic cord. Kindly write down. Yes, please. Next point, a fibrous cord that extends from caudal end of testis to the wall of, right, from the caudal end of the testis to the scrotal wall, is called dash. So this is gubernaculum, right? So it's a very, very old Ames PYQ. Okay, it's a very old Ames PYQ. So basically, inguinal canal is like a connection, right, through which the testes, they will descend into the scrotum. Along with that, spermatic cord is also there. With the help of that, the testes will descend into the scrotum. Now, when you are talking about the testes, right, their caudal end, their tail end, from that there is a fibrous cord that will help that testes to get attached to the scrotal wall. Like imagine it in this way. They are attached here, right? So what do we call it? We are calling it as gubernaculum. Right, we are calling it as gubernaculum. Is that clear? So it is not in the NCERT, I'll tell you honestly, but this is a very old Ames PYQ, and I told you already that this time the level of the session is going to be very high because from the last time, right, this is what I have learned, that students, you should be open, you should accept it. We are getting some information, even, right, even if it is from the deleted portion, let's accept it, right? Right? Just do it. Quick. Okay. So overall testis, you know that 4 to 5 centimeter in length, and the width is going to be 2 to 3 centimeter. Right, students? So the peritoneum that supports the testis, is called mesorchium. That is the word. Okay, mesorchium. So you know now, peritoneum is a serous membrane that surrounds our viscera, that protects our viscera, the body organs. So when it comes to the testis, you are going to call it as mesorchium. Right? You are going to call it as mesorchium. So up to this part all clear? Up to this part all clear? Any doubts? Any doubt? Okay, so now let's talk about the protective covering. So I hope up to this part everything is done and dusted. Okay? Right? So this is the diagram that now we have to discuss. This one, and I'll talk about this one. Can you see that? This one. So now we'll be talking about this internal structure. Can you see that? Okay, so let's get started. So I'll start directly with the testis here. Just a minute. Okay, so let's start with the internal structure. So when it comes to the internal structure, we will start with the layers, right? We'll start with what? We will start with the outer coverings. And students, when we talk about the outer coverings, you have to remember a trick, that is web. What is it? It is web that you have to remember. What do you have to remember? Yes, everybody in the chat section. What do you have to remember? You have to remember web, right? So what is the meaning of this web? It is simple. When you are talking about the internal structure, we are starting with the outer coverings, right, of the testis here, of course, right? So here you will see, like, let's suppose, firstly, I'll teach you it like this. So web stands for, so first of all, it is tunica. Tunica means covering, right? What is the meaning of tunica? Tunica means covering, right? Tunica means what? Tunica means covering. So outermost is tunica vaginalis, right? Then comes the tunica albuginea. Then comes the tunica vasculosa. So tunica word you have to remember. So as I said, what is the meaning of tunica? Tunica means covering. What is the meaning of tunica, students? Tunica means covering. So these are the three outer protective coverings. So what we have to remember? We have to remember web. So tunica vaginalis is there, albuginea is there, vasculosa is there. So if you ever get confused in between vasculosa and vaginalis, vasculosa, vascular supply, blood supply, so it should be towards the inner side. That is how you can remember. Is that clear? That is how you can remember. So these are the three outer coverings. Okay, okay, so see this, tunica vaginalis, the outermost. Tunica vaginalis, the outermost, it is showing like this parietal layer, cavity or visceral layer. Then next to it, this part, right, this capsule like part, this is the tunica albuginea. And then comes what? Then comes your tunica vasculosa. So this tunica vasculosa, right, this is also lining. Can you see these spaces here? Right, it is also lining that. So now this is what we have to discuss. So students, when you talk about, I'll mention the points as well. So when you talk about the structure, what you have to remember, when you take the section of the testis, we know that in the testis, sperm production is there. I told you about the outer coverings. When it comes to the outer covering, tunica vaginalis, it is not covering the testis entirely. It is not covering it at the posterior end, through which the arteries and veins are coming and going. Then comes the tunica albuginea, which is covering it completely. Then comes the tunica vasculosa. So before understanding or before mentioning that things in points, right, so what are we going to do? See, imagine, I'll teach you it in this way. Let's suppose there is a building, right? There is a building. In that building, just imagine, there is what? There is a building. In that building, you see around 250 floors. How many floors are there? Around 250. And each floor, right, each floor is having 1, 2, 3 apartments or the flats. That's how you can remember it. So when you look at the testis, coverings are there. So in the testis, you are going to see the testicular lobules, right? Some compartments. You are calling them as testicular lobules. And each testicular lobule will have 1, 2, 3 highly coiled seminiferous tubules. That's how I always make it, right? You understand this part, then we'll write the notes. Don't worry. Imagine it like this, right? Like this is the testis, right? When we see it inside, so what do we see? We see that we have the 1, 2, 3 highly coiled seminiferous tubules, right? Seminiferous tubules, right? So these seminiferous tubules, where are they present? They are present in some compartments. You're calling them as what? Testicular lobules, right? Testicular lobules. So this is what we have in the testis. And this seminiferous tubule, okay, it is not any accessory duct. Okay, these seminiferous tubules, my dear students, this is actually the site of what? The site of spermatogenesis. Okay, so in the sperm, in the seminiferous tubule, you'll see the spermatogenesis. Am I clear? You'll see the spermatogenesis. Am I clear? Am I clear? So that's how the structure is. So you understand the structure first. Okay, you understand the structure first. So we are talking about the testis. We have discussed the 3 outer coverings. Then we are saying that within the testis we see the testicular lobules, right? Around 200 to 250 compartments they are. Then we are saying that each compartment is having highly coiled, right? Each compartment is having highly coiled tubules where the sperm production is there, and we are calling them as seminiferous tubules. Now all these tubules, right, they are going to form a network, students. Here I'm keeping the NCERT part only. So what are they are doing? All these tubules, they are forming a kind of network. All they are connected. They are forming a network there. So what do you call it? You call it as retitestis, right? So from that retitestis you'll see some other, right, some ducts are there. This is vasa efferentia, or you can call it vas efferens, right? And then finally what will they form? They'll form what? They'll form this epididymis, and then finally that epididymis will form your vas deferens. So firstly tell me which is also known as ductus deferens. So firstly understand this part, then we'll write the notes. Okay, so we are talking about the testis. I'm saying there are 3 outer coverings: tunica vaginalis, tunica albuginea, tunica vasculosa. Tunica vaginalis is covering it, but not at its posterior end, through which the, I would say, the arteries and veins, they are coming and going. Then comes the tunica albuginea, right? Fibrous connective tissue, it is like a capsule. Then comes the tunica vasculosa, which is obviously lining these testicular lobules, right? These are the testicular lobules. So each testicular lobule is having 1 to 3 seminiferous tubule. They all are forming a network. We are calling it as the retitestis, through which we have vasa efferentia. So obviously these seminiferous tubules, they are going to form the sperm. They'll pass it to the retitestis. Retitestis will pass it to the vasa efferentia. Then finally to the epididymis. And this epididymis is what? This is the place where the temporary storage and maturation of sperm occur. Where the temporary storage and maturation of sperm occurs, and then finally it will pass to what? Vas deferens, which will carry it further. So firstly tell me this part clear. Are you able to understand it? Because then I will write it. Yes, please. Sure. Now look at this diagram. Can you see these testicular lobules, these chambers? Can you see the seminiferous tubules? This network, can you see? This is the retitestis. From the retitestis, right, these tubules, these are the vasa efferentia, also known as efferent ductules. Then finally they'll go to this epididymis, and that epididymis will further form the vas deferens. So remember, in your spermatic cord, right, not just the gonadal artery or veins, vas deferens is also present, which is also known as ductus deferens. Is that clear? This flow and everything clear? Yes, this flow and everything clear? You tell me yes or no. Right, send me some thumbs up, right, so that I'll be clear that yes, you are aware of it, and then I'll teach you further. Quick. Guys, I want to see some fire in the chat section. Quick. Are you sure? Pakka? Done? Okay. So tunica vaginalis, outermost layer having visceral and parietal layer covering testis except at posterior end, through which arteries and veins enter and leave. Then comes tunica, albugenia, so covering testis completely, having white fibrous connective tissue. Then comes tunica vasculosa. Okay, innermost also line the testicular lobules. Am I clear? So also lines the testicular lobules. Am I clear? Everybody done? Right? So underline this part. This line is important. Okay? So each testicular lobule contains 1 to 3 seminiferous tubules. So this is the site of sperm production. I hope it's done. Yes. So this is the site of sperm production. Okay. Sure. So each, I'll first write this quick. Done. So the tips of the tubules join and form tubuli recti, which further form a network called rete testis. So rete testis is important, given in the NCERT as well. So we have already discussed it. Right now, I'm just writing the notes for you. Is that clear? Everybody done? Yes, please. Okay. So before sending you the PDF now, I'll rearrange all the slides. Done, students? I'll rearrange all the slides. Done, students? So rete testis form efferent ductules or vasa efferentia or vas efferens, right? So rete testis form efferent ductules or vasa efferentia. Done. That basically that will come out from the posterior part of the testis, and then it will join and it will form the common epididymis. So I hope this route is clear to everybody, because it is again a PYQ. Okay. Okay. So rete testis form efferent ductule or vasa efferentia. They fuse and they will form. Okay. Okay. That come out from posterior part of testis and then form a common, highly coiled epididymis. So do mention it, do write it. Very, very important it is, students. So epididymis is a place where there is, the word is temporary, it is not permanent, you have to mention it, storage and maturation of sperm occur. Right? So that is again a previous year question and the important one. Okay, so this is something that you have to remember. So if the sperm, if they don't get stored in the epididymis, they will not get the mobility. Like if let's suppose you directly take the sperm from the seminiferous tubule, if they are not stored at least minimum for the 18 hours, if they are not stored in the epididymis, they will not at all be motile. Okay, they will not at all be motile, right? They will not move. Is that clear? Is that clear? So this is for the epididymis. Okay, this is for the epididymis. Done, students? So now when you look at the epididymis here, see, look at the epididymis here. So this epididymis, can you see the diagram? Now you see the diagram? So see some tubule recti, these are the straight tubes, they are further joining, they are forming a network here, retitestis. From that retitestis, right, they all will fuse, they will form this around 10 to 20 efferent ductules or the vasa efferentia. Then they are opening into this highly coiled epididymis. So as I said, temporary storage and the maturation will occur. So when you look at this, can you see this head part? So head is cap. So this is also known as the caput epididymis. This is the body part of the epididymis. So for body of epididymis, we use the word corpus epididymis. This is the tail part of epididymis. Tail mane cauda. So this is the caudal epididymis. Right? That is how you can remember. Just like we put the cap on our head. So head part is also known as capitid epididymis. Body part is the corpus epididymis. And the tail part, the ends are the caudal epididymis. And further you know that what will be there? There will be vast difference. There will be vast difference. So any doubt? Any doubt? Now I'll show you one question as well. Right? See, I hope you guys have seen my last video, right, where I was discussing about the NCERT part, that how you should read the NCERT. So if you will read that part, okay, then see, just I'll give you an example. Can you see this question? Right, it is not a MCQ type question, right? So this is a very short answer type question. The path of the sperm transport is given below. Provide the missing steps in the blank boxes. So you try to fill it. You try to fill it quick. And do you know that it's a repeated PYQ? It's a repeated PYQ. Do you know that? The pathway, you can even write it down. The pathway of sperm transport. It's a repeated PYQ. So you fill it. So this is the exemplar question. Done? Answer this. Quick. Yes, please, done. Exactly. So retitestis. Then comes the vas deferens. So that is how it works. So it is the temporary storage and maturation. So if sperms are not stored here, they are not going to be, they will not get the functional maturity. Okay, excellent. So that is how we have to complete the NCERT part. Okay, so let's try one more question. Okay, so select the odd one out from the following structure with reference to the male reproductive system. Try to solve it. Quick. Try to solve it. Quick. Exactly, very easy it is. Done? So do solve the exemplar. Fine, do solve the exemplar. So here you people can see the testes are situated outside the abdominal cavity within a pouch called scrotum. NCERT part is done. That maintain the low temperature. Done. That is again a PYQ. Then we talked about the dimension 4 to 5 centimeter, about 4 to 5 centimeter in length. This is important. 2 to 3 centimeter in width. Then width. The testis is covered by dense covering. Each testis has about 250 compartments that we call as testicular lobules. Done, students? NCERT part we will also cover. Now when it comes to these cells, we'll discuss it, but after this part. So now, the male sex accessory ducts, what are they? See, retitestis, vasa efferentia, epididymis, vasa differentia. So I hope this part is clear. Okay. So seminiferous tubule, they open into vasa efferentia through retitestis. This is again a direct PYQ. So vasa efferentia leave the testis, they open into epididymis along the posterior surface of each testis. So yes, we have to remember anterior posterior part as well. So again, that's an important PYQ. Fine, that's an important PYQ. Is that clear? Is that clear? So do underline this part in your book. Right, so it is the upper or the posterior surface of each testis. Done? Sure? Now, epididymis leads to vas deferens, right? That ascend to. Now you imagine, this is the pair of vas deferens, right? It will come to the abdominal cavity through the inguinal canal. Then it will coil over the urinary bladder. Okay, so see, vas deferens ascend to the abdomen, loops over the urinary bladder. It receives a duct from the seminal vesicle. Now this is the part that we need to discuss. Okay, see, now you are aware of the accessory ducts. Now after the accessory ducts, we need to talk about the accessory glands. So when you talk about these glands, we will discuss about the seminal vesicle. It's a pair. Then prostate gland. Single. Then bulbo-urethral or corpus gland, again pair. So now this is what we need to discuss. Okay, so sperms, they'll pass further. So sperms, they'll pass further through the pulsation of that vas deferens. That is muscular, na? So the pulsation of that vas deferens, the sperms will further pass. Okay, so we already discussed about the vas deferens, right? We highlighted it from the NCERT. Okay, done. So this is in pair, this is single, and this is again in pair. So let's discuss it, but before that, let's see their position. See this. Can you see that? Testis, epididymis, vas deferens, coiling over the urinary bladder. And now you can see that this is the seminal vesicle, it is in pair. Okay, so let's suppose this is vas deferens. Here you have seminal vesicle. This is the duct of that seminal vesicle. Sorry. When vas deferens receives the secretion from the seminal vesicle, then it will form this ejaculatory duct. Can you see that? And that will further. And here also you can see This is also vast difference. Receiving this. See this, ejaculatory duct. So finally it will open in the urethra. So this is the penis part. Inside there is the urethra. So this part is the pineal urethra, students, and in the surrounding the prostate gland is present. Okay, so it is single. Okay, and just below this part here, this is the bulbourethrocarpus gland. So you should know the other name as well. It is also a previous year question. So firstly, just look at the diagram and tell me if it is clear. Yeah, this is a very important lecture. This is a very important lecture. Noted? So here also you can see the position bulbourethral, prostate, and then here this is the seminal vesicle. Just have a look. Quick. So you write it first, then we'll talk about the next part. Done, students? Sure? So let's talk about these glands. So we will start with the seminal vesicle. Okay, so this seminal vesicle, see, males, right? The males basically they ejaculate semen. Semen contains the sperm. Along with that, there are some other things also. And who's going to form that? These glands. So when you talk about the seminal vesicle, of course, as I said, it is in a pair. So it forms 60 to 70% of the semen. Do you know that? Right. So secretions of seminal vesicles have alkaline pH, students. What sort of pH do they have? They have alkaline pH around 7.3, right? And it contains fructose that will act as a fuel for the sperm. Okay, it will also have prostaglandins, like sperms, they use the fructose for their energy, prostaglandin, that is going to help in the contractions, right? Contractions of the female uterine cavity, and in the case of male urethra as well, right? Other than that, you'll see citrate, inositols, even the clotting proteins like fibrinogen, etc. They all are present. Okay, they all are present in this secretion. Now, when you talk about the secretion, when you firstly talk about the fructose, so as I said, it acts like a fuel, provide energy to the sperm. Do you know that? Provide energy to sperm, right? So this fructose, it will not be found anywhere else. So, you know, this presence of fructose in the female genital tract, it was used as a forensic test to detect the rape or something. Because if there will be the rape, if there will be rape, then obviously the male will discharge. When male will discharge or ejaculate in the female genital tract, so semen will be there. Semen means the fructose will be there. So this the presence of fructose in the female genital tract, it indicates the sexual intercourse. Okay, it indicates the sexual intercourse. So this is the role of the fructose here. It provides the energy. Then comes the prostaglandins. As I said, they are meant for the contraction. So they are helpful for the contraction of the uterine cavity. They'll help that sperm to reach the part where the egg is present, right? Where the egg is present, and even So that is how it works, right? It stimulates that uterine contraction. So clotting proteins are also there, right? Fibrinogen, you know that it is inactive, right? So clotting proteins are also there. So how will they help, right? They will basically clot the semen or coagulate the semen after coagulate the semen, right, after ejaculation, so that obviously sperms can, you know, just conserve their energy. So that is how it works, right? And how this alkaline pH helps. So this alkaline pH, basically it reduces acidity of male urethra, because the urine will also pass through this, and of female reproductive tract, because and of female reproductive tract. So that's how it works. Any doubt? Yes, please. Any doubt? Quick. Any doubt? Done? Done? So next is prostate gland. This one is single, open in penial urethra, forms around 30% of the semen, right? It forms around what? 30% of the semen contains, sorry for this, so contains citric acid, inositols, citric acid, calcium, phosphates, even profibrinolysis, other clotting enzymes, clotting proteins, etc. So basically what is going to happen, right? Like let's say secretions of seminal vesicle and prostate gland, what will happen when they get mixed? Then what is going to happen? The semen coagulate, okay? And in coagulated semen, right, the semen will coagulate, and in coagulated semen, sperm movement is less, so energy is conserved. Done? So that is how it works. Write down quick. Done. So semen gets coagulated, so coagulated semen में sperm movement is less and the energy is conserved. ठीक है? Now obviously in female genital tract or overall, you know that due to fibrinolysis, semen gets liquefied, and after that semen gets liquefied, and after that what's going to happen? Sperm movement starts. So that is how it helps, okay? And when you talk about the next part, that is your Cowper's gland or bulbourethral gland. Again, it's in pair and its main function is lubrication of penis tubes. What will it do? Lubrication of penis. Is that clear? Is that clear? Quick. Any doubts? Done, students? Right, so here also the secretions are alkaline, around 7.2 pH. So it is clear secretions. Okay, so that's how you can remember. So obviously, no doubt this bulbourethral gland, and it is basically a pair. So it is like, see the diagram, see the diagram. See, so it opens into the, like this is the right prostate urethra, right? This penial urethra, like this part is prostate urethra also. This is a membranous urethra. So it opens into this membranous urethra. So all these secretions, they are making the seminal plasma or the semen, right? So this is important for the sperm. So sometimes, you know, the secretion of this corpus gland, it lubricates the penis so that, you know, smooth movement can be there during the insemination part, and plus the sperm transport is also like it will be smooth. So sometimes what happen that these secretions of this corpus gland, sometimes it also contains some sperms, like before ejaculation. So we will read about it in the reproductive health chapter about the coitus interruptus withdrawal method, that before ejaculation male withdraw the penis, right, or male ejaculates outside the body, outside the female body. Okay, so sometimes what happened, even because of this withdrawal thing, right, sometimes female just get pregnant, or the failure rate of this withdrawal method is there, because sometimes in these secretions the spermatozoa can be there. So that is the point, right, students? So this is all about the male reproductive system, right? So all these secretions, all of it collectively form what? Semen, right? All of it collectively, all these secretions collectively, they are going to form what? They are going to form the semen. So sperm plus accessory reproductive gland fluid, this is going to make it. So you know that, right? So it will have sperm plus secretions of accessory glands. The pH is near about 7.3. The volume is 3 to 4 ml during each ejaculation. So you can see that on an average, this is what we can see, and that too in a healthy individual, right? Normal sperm count, if I talk about the normal sperm count, so it will be 20 to 120 million per. So normal sperm count on an average is 20 to 120 million per ml. Okay, this is it. Noted? 20 to 120 million per ml. Is that clear? Yes, everybody. Is that clear? Okay? So human males ejaculates 200 to 300 million sperms during a coitus, and for the pregnancy, right, 60%, like I'm not saying all of it, but at least 60% sperms, they must have normal shape and size, and at least 40% must show vigorous motility. Okay, so 60% must have normal or proper shape and size, and 40% must show vigorous motility. So that is what you have to remember, students. So if sperm count is less than 20 million per ml, then it is oligospermia. Understood? So that is it. Then it is oligospermia. Done? So please write down. Then let's talk about the seminiferous tubule and the penis part. Firstly, let's talk about the penis. Quick. Yes, students, is it done? So can we move to the next part? Okay, so next is, students, we will talk about the penis. So when you talk about the penis part, so can you see this conical, right? This terminal shaft here. So this bulged part is what? It is the glans penis, right? It is the glans penis. Here we talk about the opening, and you know that this penis is having the urethra, right? So this terminal bulging part is what? It is the glans penis, students. Very sensitive it is. It is covered by a skin, right? Loose fold of skin. Covered by loose fold of skin, that is foreskin or prepuce, right? So here you know that opening is there. This is known as urethral meatus or urinogenital aperture. Are you getting my point? Urethral meatus or urinogenital aperture it is, because urine and the semen comes out from the same opening. But obviously when semen is coming, urine will not come. When urine will come, the semen will not come. And yes, prostate gland will open into the prostatic urethra, and this terminal part, the end part, is having that pineal urethra. This is what you have to remember, right? So loose fold of skin is there. So the removal of the skin, this removal of prepuce is basically known as circumcision. Okay, and here when you talk about this penis, so this penis is made up of what? It is made up of, right, 3 bundles of erectile tissue. How many? 3 bundles of erectile tissue. How many, students? Yes, please. Yes, please. It is having 3 bundles of erectile tissue, right? Right, and they are longitudinal. So when we take the cross section here, let's suppose, like, see this. So you will see right and left will have see, this is corpora cavernosa, again and this is corpus spongiosum. Okay, so again this is again an Ames PYQ, not of NEET but from Ames. Okay, it is from Ames. So 3 longitudinal erectile tissues are there. Why erectile? Because the blood flow will be there during the sexual arousal. So right left is corpora cavernosa, this one is corpus spongiosum. Corpus spongiosum will be till the glans penis, but this will not reach at the ends, right? So when there is the sexual arousal, right, the blood flow will be there, and that will result in the erection of the penis. This is what you have to remember. Okay, so that will result in what? That will result in the erection of the penis. Okay, so this is what you have to remember. And when we talk about the erection of the penis, so it is controlled by, right, I'm not going to discuss the detailed mechanism here, not required for you. It is controlled by ANS. So ANS, you understand now? Autonomic nervous system, that will include your parasympathetic or sympathetic nerve fibers. Am I clear? Please write down. Quick. Right? right? So this corpus spongiosum, it is traversed by urethra. Remember this, throughout its length, it is traversed by urethra, this corpus spongiosum. Okay, so erection of penis is, you know that it is vascular phenomena controlled by ANS. It occurs due to the blood flow, increase in the blood flow during the sexual arousal. So please write down, then we'll talk about the seminiferous tubule, the histology, and we will also discuss about the spermatogenesis here. So these ducts store and transport the sperms from the testis to the outside. This part is done. Urethra originates from, see this, urinary bladder, extend through penis, right, to its external opening called urethral meatus. Done. So penis is the male external genitalia. It is made up of special tissues. So as I said, special erectile tissue. So basically 3 longitudinal bundles or the masses are present, right? So you can even write it down in the NCERT. Okay, so enlarged end of the penis is called the glans penis. So your corpus spongiosum will be expanded to that part. So this is the foreskin, or it is also known as prepuce. So removal of it by surgery is known as circumcision. Done, students, any doubt? So about the glands also we have discussed. Now we will discuss about the histology of the seminiferous tubule, right? The internal part of the seminiferous tubule, that is it. So if there is any doubt, do let me know. Yes, please, do let me know. So now we'll discuss about the histology, and along with that, of course, we are going to discuss about the spermatogenesis here. Chalo, so now the time for the doubts. Yes, please. The time for the doubts. Please tell me. Done. Any doubt? If there is any doubt, please feel free to ask. Sure? Sure? Okay, so let's talk about the histology of seminiferous tubule. See, seminiferous tubule is not the accessory duct. First of all, understand this thing. Basically, testis, as I said, so as I said, that testes are actually what? Primary sex organ. So within the testis, testicular lobules are there. That lobules will contain 1 to 3 seminiferous tubules. This is what we have discussed now. Okay. So I have seen students making this mistake. They think that seminiferous tubule is also an accessory duct. No, this is the part of the testis. It is actually the site of, right? It is the site of spermatogenesis. So now this is what we have to discuss. Right? This is what we have to discuss. Chalo, let's get started. Oh, sorry. So now we have to discuss the histology of the seminiferous tubule. Like, let's see. In the testicular lobules, as I said, 1 to 3 highly coiled seminiferous tubules are there. Now, what are we going to do? We are going to take a cross section of that. Let's suppose this. Okay, let's suppose this. Now let's understand this part. So now what is going to happen here? See, when you talk about the seminiferous tubule, outside they will have the connective tissue, right? Outside, what do they have? Outside, what do they have? They have the white fibrous connective tissue it is, right? And you call it as tunica purpura, right? Let's suppose this. And now just next to it, what will we see? We'll see cuboidal epithelium, right? You make sure you write it. And at places, you'll also see some columnar cells also. So when you look at this part, okay, so this part is actually germinal epithelium, right? And it is a previous year question, my dear students, right? Germinal epithelium. So it is made up of cuboidal epithelium. That's a PYQ. Okay, that's a PYQ. In between, you will also find columnar cells. These cells are known as Sertoli cells, nurse cells, testicular cells. Are you getting my point? So these cells are Sertoli cells, nurse cells, testicular cells, right? These are the columnar, and these are the cuboidal epithelium, germinal epithelium. Do you know why do we call it as germinal epithelium? It is believed, and it is also mentioned in the NCERT, that because they contain the spermatogonia cells, the germ cells that are going to form the sperm. But actually that is not the case, because students, during the embryonic development, right, during, when I'm writing this point here, that during embryonic development, plant growth regulators, I'm so sorry about this, right? Suddenly the same thing came into my mind. So during embryonic development, right, primordial germ cells, right? So PGCs, I call it as plant growth regulators as PGR. So suddenly that came in my mind. So during embryonic development, primordial germ cells from yolk sac find their way to gonads that are going to form these germ cells that will further help in the gamete formation. Is that clear? Is that clear, everybody? Sure. So PGCs, they enter the primordial germ cells, they enter the gonads, right? Then they will form spermatogonia. After several mitotic division, right, they finally arrest at the G1 phase of the mitotic cell cycle. So they'll remain there until even after the birth. Okay. So these are the PGCs that will enter the gonad. Is that clear? Is that clear? Sure. Done. So they'll find their way to gonad. So these PGCs, then they will form, students, spermatogonia, right? That spermatogonia mitotically they'll divide, and then they will have an arrest in the G1 phase only. That interphase, right? Initially we talk about the G1 phase. They'll stay there in the G1 phase till the puberty, and after that, when they attain the age of puberty, right? In that case, what's going to happen? These spermatogonia, they form some, kind of proteins that is not required for you. When that protein is there in the critical concentration, then students, the germ cell differentiation starts, and after that, what's going to happen, right? After that, what's going to happen? The further process will start. Is that clear? Is that clear? Is that clear? Any doubt? Done? Any doubt? Sure. So students, in humans, the progression of spermatogonia, or you can call them as a germ cell, to spermatozoa, obviously when they attain the age of puberty, takes around 65 days. So actually in one of the standard books, I read 65 days. In one of the books, it was around 73 days. And it is the, obviously, spermatozoa, mature spermatozoa. It takes place around 65 days. So in some books, it was around 73 days. So you can somewhere consider it in the range of 65 to 75. So I have read this in a standard book. So I'm mentioning this here. So this is what it is going to take after the puberty. So basically, right, initially in the NCERT, it is mentioned that these germinal epithelium is having the germ cells that form the, right, obviously the gametes, right? That's why this layer is the germinal epithelium, but actually that is not the case. So this part is clear. So now when you look at the seminiferous tubule, just imagine it like this, right? Just say, let's suppose this is the lumen of the seminiferous tubule here. So this wall here, right here, we will see the spermatogenesis will occur. What will occur, students? The spermatogenesis, it occurs. Keep it in your mind. What will occur? Spermatogenesis will occur. Now this is what we are going to discuss. So when will it occur? When they'll attain the age of puberty. So up to this part, all clear? Yes, please. Up to this part, all clear? Any doubts? Any doubts? Sure? Sure? Any doubts? Take him. So one thing that is clear to us, that it is the germinal epithelium having the cuboidal cell. At places, the columnar cells known as Sertoli cells are there. Sertoli cells, or the nerve cells, or the testicular cells, they provide nourishment to, right? What is their function? They provide nourishment to, right, the developing sperms. It is their main function. There are other functions as well, but it is their main function. Now you know that there is no just, we don't have just one seminiferous tubule. Just say, let's suppose these are the seminiferous tubule. Obviously, here you will have the stroma, the connective tissue. So you also see there are many other cells. We'll talk about the main cells here. So here, though, I told you about these, columnar Sertoli cells, right? Because this is what we need to know to understand the process of spermatogenesis. Now here in between, you'll also see the another cell, that is the Leydig cells. We can also call it as interstitial cell, right? This is the Leydig cell, and it is also known as interstitial cell. So Sertoli cell, I told you, they'll provide the nourishment, and these Leydig cell or the interstitial cell, under the influence of the pituitary hormones, it is going to release the testosterone as well, right? So they are present here in the stroma. Is that clear? They are present here in the stroma. Is that clear? Is that clear? Sure. So I'll tell you the function of these. So I have already mentioned the main function of these Sertoli cells and the interstitial cells. But now we have to discuss their function in detail. But before that, let's talk about the spermatogenesis. So guys, if you want to understand, because when we will start this spermatogenesis topic now, then of course, with that particular topic, you'll get to know the function of these cells. But even before that, right? Even before that spermatogenesis part, what exactly you need to understand? See, we will discuss about the hormonal control of pituitary, right? Hormones of pituitary rather. This is what I'm going to mention. Okay, so let's discuss about hormones of pituitary first. Is that clear? Is that clear? And this part is for the male and the female system as well. So listen to it very, very, very carefully, and then, okay, listen to it very carefully, and then, because I will discuss the same in the female part as well. So basically, you know it very well. We have a supermaster gland and we call it as hypothalamus. What is it? It is the supermaster gland, right? And this hormone, it releases the releasing hormone or the inhibitory hormone, right? Then we have the pituitary, which is the master gland. That is the master gland. Jaise, that's the sort of diagram, if you remember. Remember this part, right? Hypothalamus, the stalk that is connecting it with the pituitary. Now the point is, this pituitary, which is also known as hypophysis, right? So this hypophysis, we discuss it like this: adenohypophysis, right, which will include your anterior part mainly, and you have neurohypophysis, right? This is a class 11 topic, students. I believe you know this. Okay, so we have, what do we have? Hypothalamus, supermaster gland. Then you have the pituitary, which is also known as hypophysis. See, we talked about the male reproductive system. We discussed about the histology of the seminiferous tubule, right? We discussed about the main points there. Now we are talking about the hormones of pituitary, because we have to start the spermatogenesis, and before that you need to understand this part. So adenohypophysis is having two parts. One is pars distalis, which is actually the anterior lobe of the pituitary, and then you have pars intermediate lobe or the middle lobe of the pituitary. They are, kind of fused, so it is making adenohypophysis. So neurohypophysis, which is not even the true endocrine gland, this is the pars nervosa. Okay, pars nervosa, it is the posterior lobe of pituitary. This is what you have to understand. So students, the point is, this neurohypophysis, it is under the direct neural regulation of the pituitary gland. Okay, direct neural regulation, right? Hypothalamic hormones, neurons, they will make the hormone, they will provide it directly to the neurohypophysis. It doesn't make its own hormone, right? It just stores the hormone. So it is going to release oxytocin, which is also known as bonding hormone, love hormone, right, students? Milk let down hormone, okay, labor hormone, and then it also release antidiuretic hormone. Matlab it stores this hypothalamus. Sorry, I made one mistake. Yes, so neurohypophysis is not under the direct regulation of hypophysis. No. It is under the direct regulation of so it is under the direct neural regulation of hypothalamus. Right, it is under the direct neural regulation of hypothalamus. Okay, direct neural regulation of hypothalamus. Hypothalamus will make the hormone, will give it to it, it will store it, and when required, it will release it. So oxytocin and ADH, because oxytocin also we need to discuss. Okay, then pars distalis, pars intermedia. When hypothalamus, when it releases GnRH, what is this? Gonadotropin releasing hormone. What is the meaning of gonadotropin? The hormones that act on the gonads. Then pituitary, the adenohypophysis, particularly this anterior lobe, it release gonadotropins. It release gonadotropins. What will it release, students? It will release gonadotropins. So that gonadotropins are FSH or LH. What's that? FSH or LH. FSH stands for follicle stimulating hormone, and LH stands for luteinizing hormone. These two hormones are gonadotropins. Is that clear? FSH and LH. FSH and LH. Follicle stimulating hormone and the luteinizing hormone, LUT, EINI. So follicle stimulating hormone and the luteinizing hormone. So these two, they will act on the gonads. Now what's going to happen when the boys, they'll attain the age of the puberty, right? Obviously these hormones will release. Plus I told you there is a factor, right? There is a factor which is not required for you, right? There is a, that is a BMP8B, right? Like when it is at the critical concentration, the germ cell differentiation occurs. Then further the process occurs. But let's talk about the hormones here, right? Now what's going to happen? See, this FSH acts on, from this S, remember, it acts on Sertoli cells, and this LH, luteinizing hormone, it acts on Leydig cells. So, right, Leydig cells, you know that we also call them as interstitial cells because they're present in between two seminiferous tubules. They are in the connective tissue. So, in the case of males, LH is also called as ICSH, that is interstitial cell stimulating hormone. Am I clear? Firstly, tell me, am I clear? This hormonal control part is clear? Quick. This hormonal part is clear? Yes, please. Yes, please. Quick, students. So once it's done, do let me know. Yes, please. Done? Sure? Okay. So now, because you know about the hormones, so now understand it. These Sertoli cells, firstly, provide nourishment to developing spermatozoa, form blood testis barrier. I'll explain it. To protect sperm from immune system, right? To protect sperm from immune system. Then Sertoli cell releases anti-Mullerian hormone, androgen binding hormone, that is known as ABP, and inhibin hormone. That's again, that is proteinaceous in nature, right? That is again a PYQ. I'll explain the function here. You make sure you write the notes as well. Okay? Okay? Done? So these are the hormones released by it. Now students, when you talk about AMH, anti-Mullerian hormone, inhibits Mullerian duct formation in males. So Mullerian duct during embryonic development, you'll see Wolffian duct and the Mullerian duct. Mullerian duct forms the female part, Wolffian duct forms the male part. So this Sertoli cell, they will not allow these Mullerian duct to form in the male. Then when you talk about this ABP, it concentrates the testosterone right? It concentrates the testosterone that diffuses into seminiferous tubules, and you know that testosterone, the androgen, right? It is the androgen, it is the steroidal in nature, right? Steroidal hormone it is. Okay, so it is the steroidal hormone. So it doesn't have its receptor on the plasma membrane. Its receptors are within the nucleus. So it concentrates the testosterone that diffuses into the seminiferous tubules and helps in spermatogenesis. And this protein hormone inhibin, right? What is the role of this inhibin? So this inhibin means to inhibit, right? It sends a negative feedback to pituitary and hypothalamus. So you know that if it is giving the negative feedback, so it simply means it wants to stop the secretion of the FSH, right? So that they can stop FSH secretion. So firstly, tell me, this part clear? The function of Sertoli cells should be very clear to you. Yes? Done? Any doubts? Done? Sure, students? And even Sertoli cell can form estrogen from testosterone. Yes, even in the male body, estrogen is required. Estrogen provides the motility to the sperm. If there is no estrogen in the male body, then there will be no motility to the sperm. And of course, its concentration is less, but it is required. And Sertoli cells phagocyte dead cells. So that's what you have to remember regarding the Sertoli cells. So I have told you already, FSH acts on it and helps in spermiogenesis. You write it, I'll explain it. It's not spermatogenesis, it's spermiogenesis. So this is the function here. So is it clear? I'll explain all the points here. So is it clear? Is it clear? Sure? Any doubts? Any doubts? Okay, so as I said, LH acts on Leydig cells. It is also known as ICSH here. Done. Releases androgen, right? And you know that here you are talking about the testosterone. So this testosterone is important for spermatogenesis. For secondary sexual characters, like when boys, when they attain the puberty, right? This larynx, you know, na, it gets prominent, Adam's apple form, right? Adam's apple form, and moreover, the beards and all, they start growing, isn't it? Isn't it? Isn't it? So that is how it helps, right? Even it is formed in the fetal life also, so that, you know, the testis can descend. So these are the main function. Now what is going to happen? Now let's discuss this spermatogenesis part. Do you know, when we discuss it in too much detail, we also talk about the spermatocytogenesis, where we discuss that how the spermatogonia keeps on dividing, it keeps on dividing, it keeps on dividing, it keeps on dividing, and it's replenish itself. It makes a stock of it. You know that in the case of males, once the spermatogenesis starts, like when they attain the puberty, the spermatogenesis starts and it will remain till the end. Like in the case of males, just like females, they don't have the menopause. Though though due to the decreased secretion of androgen formation, right, in their case also the sperm productions it get inhibit, right. I'm not saying it will be for the lifetime, no, it depends on that individual. So if there is the less androgen synthesis, if testosterone level is less, so of course in that case you will see the sperm production also inhibit in the case of males, but the age is not specified, as I said. Okay, right. So now what is going to happen? So as I said, when you are talking about the spermatogenesis, you know the site already. So let's talk about the cells. So spermatogonias are they, you know that PGCs, primordial germ cells, they'll form these spermatogonium, they'll proliferate, they'll increase their number mitotically, they form so many spermatogonia. So spermatogonia is basically diploid. So let's start this process. Just say, let's suppose you have spermatogonium here, right? So we will discuss it like this. We have the type A and we have the type B spermatogonia. That's how we are going to discuss, right? Type A or type B, it is 2N. So when you are talking about the type A spermatogonia, sorry students, sorry. Just a minute, just allow me 1 minute. Done. So now type A, like as I said, when it will divide, it will proliferate. Then again it will form a type A spermatogonium. So its function is it will just show the multiplication phase. It will keep on dividing, keep on dividing, keep on dividing, and it will keep on dividing. That's how it works. Okay, type A spermatogonia, keep on dividing, keep on dividing, keep on dividing, so that there can be the replenishment or the stocking of these spermatogoniums. Okay, this is also known as spermatocytogenesis, not given in NCERT, so not talking about it. So it also form a type B spermatogonium. Now this type B spermatogonium, it will grow. Okay, its size will increase, it will grow, its size will increase, it is going to form, when its size will increase, cytoplasmic increase is there, we will call it as primary spermatocyte. What are we going to call it? Primary spermatocyte. That is again diploid. Now this primary spermatocyte, it will undergo meiosis. We know that meiosis is the reductional division. Firstly, there will be meiosis 1, right? So this primary spermatocyte, which is 2N, right, right? So this will undergo what? It will undergo the meiotic division, meiosis 1. That will result in the formation of 2 haploid, right? That will result in the formation of 2 haploid secondary spermatocyte. Okay, secondary spermatocyte. So from this part, I'll continue here. So, see, this is your primary spermatocyte, which is diploid. Now, you'll have 2 cells, they are what? Haploid. So, how will you get it? You'll get it after meiosis 1. So, this question can be asked that in which cell the meiosis occur. So, it is going to be your primary spermatocyte. So, after meiosis 1, you will have secondary spermatocyte. Now students, this secondary spermatocyte will undergo what? Right, obviously meiosis 2. Here also meiosis 2. So you will get 4 spermatids. You will get 4 spermatids. Right? How many spermatids? 4. So yes, we can ask you the ploidy here, spermatids, and they show the continuous cytoplasmic connection. Why? Because you know that in the case of males, X and Y chromosomes are there. Some cells will receive X, like 2 will receive X, 2 will receive Y, if you are talking about one spermatogonium. So, of course, Y chromosome is holindric, size is small, less number of genes, less proteins. So, through this continuous cytoplasmic connection, they can also exchange that proteins. Is that clear? Is that clear? So this is what we have, spermatids. Now, these spermatids, right? I'll talk about the changes as well. These spermatids, then they develop into the mature. First, you understand the stages. Spermatozoa. Singular is spermatozoa, plural is sperm, right? And this process here is known as spermiogenesis, right? FSH is playing role here. It is also known as spermatogenesis. Spermiogenesis and spermatogenesis. I'm writing it here. So when spermatid, it forms sperm, then this process is known as spermio. It's not spermato, it's spermiogenesis or spermatogenesis. This is the name. Okay, this is the name. So this is going to happen here. These are the stages here. So if there is any doubt, do let me know. Do let me know. Quick. ठीक है? Done? So here during this transition, here histones are replaced by protamines. Okay? Here histones are replaced by protamines. Yes, please. So protamines are having more, like histones are having glycine or arginine. Na, here it is having more arginine. So this is going to happen here. So spermatids are going to form the sperm, right? Spermatids are going to form the sperm. So this is what this complete process is. And I told you, right, when the boys, they'll attain the age of puberty, so this spermatogonia, right, it will take around 65 days to form the spermatozoa. Understood? So I'm writing the sequence here. This is how you can start. Okay. So you have spermatogonium, one spermatogonium, 2 egg. It is growing. It is forming one primary spermatocyte. Again, 2n. Then it will undergo meiosis 1. You will have 2 secondary spermatocyte, again haploid after meiosis. Then again there will be meiosis 2, then 2 secondary spermatids will give you spermatocytes, will give you spermatids, 4 spermatids, then they will give you 4 sperm. So this process is known as spermiogenesis or spermatogenesis. Now tell me if there is any doubt here. Quick. Quick. Yes, please, quick. Guys, please, quick. You have to be fast. Guys, quick. Any doubt? Obviously, this is also haploid. This is also haploid. I believe it's done. Okay. So here you people can see, like let's say, where is it going? Where is it taking place? Here na. So this is a Sertoli cell. Okay. So Sertoli cells, and here what will you see? What will you see? Guys, here you are going to see the spermatogonia in the wall here, right? In the wall here. Let me continue it here. Like let's say you know that you have the cuboidal epithelium, right? Germinal epithelium, and then this is what you have. You have the Sertoli cells. You have what? Sertoli cells, columnar one. So remember this. Here you people are going to see the spermatogonium. right? Spermatogonium. So how are they connected to each other? Right, there will be, you know, some protein, जैसे N-cadherin, right? That is how the Sertoli cell and spermatogony are associated. So here in the wall, right? Here in the wall, the stages of development are there. Okay, as we said, see, FSH is going to act on the Sertoli cell. Then it will help in its maturation of spermatid to spermatozoa as well. Moreover, students, I told you that this is going to form the blood testis barrier. Okay, so the sperms, they are not getting the nutrition directly from the blood, because if they will get it, obviously the immune cells of the blood, they are going to attack these sperms. So it is the Sertoli cell, right, that will absorb the nutrition from the nearby, and it is going to provide it to the, right, then it is going to provide it to the spermatogonia. Okay, right, then it is going to provide it to the spermatogonia. The Sertoli cells are going to provide it to the spermatogonia. So here you will see the stages of development, right? And once the spermatid, once it forms the sperm, then it leaves the Sertoli cell and it releases into the lumen here, and this is the spermiation. So I always mention this thing that students, they just get confused in these three terms, these three words. This is spermatogenesis. So when it is spermatogenesis, so here spermatogonia will develop into sperm. Okay, this is the meaning of spermatogenesis. Now when it is spermiogenesis, right? When it is spermiogenesis, which is also known as spermatogenesis, in that case it is from spermatid to sperm, spermatid to spermatus, right? The changes will be in accordance with that. Now when it is the spermiation, when it is what? When it is the spermiation, then the sperm, mature spermatozoa or sperm, in simple words I'm writing, leave Sertoli cells and release into the lumen of the seminiferous tubule, right? And they will release into the lumen of the seminiferous tubule. So after that, students, you know that they'll pass to the tubule recti, but I'm not writing it because this is not in the NCERT. Retitestis, then vasa efferentia, then to epididymis, where their temporary storage and the maturation, the functional maturation will occur. Then vas deferens, then ejaculatory duct, finally will pass to the urethra. So that is how it is, right? That is how it is. So now tell me if there is any doubt, and please take the notes. Quick. Any doubt? So tell me, are you able to understand it properly, right? Are you able to understand it properly? Yes, please. I really want to see some comments in the chat section, guys. Are you able to? And why you people are not liking it? Quick, hit the like button as well. Isn't it done now? So basically in 2 hours, male reproductive system spermatogenesis done. This part takes time, right? The fertilization, because in the NCERT portion, they didn't, in this particular chapter, they didn't emphasize the fertilization, the stages part in that much detail, okay? Right, they didn't. So that part we will quickly shuffle, okay? So how's it? Able to understand it? Sure? Sure? Then great. So this is all about the system here. This is all about the spermatogenesis here. So there is one more term that I'll tell you, that is the retrograde ejaculation. So it can, so sometimes what happen now, you know that Semen passes through the urethra, but sometimes what happens ki semen, it passes through urinary bladder. So with the urine, it starts coming out. So that is the retrograde ejaculation. Obviously, it is the problem related to the sphincter. It can even cause the, right, it can even cause the infertility in the male. So this is your homework. You'll read a little bit more about it. Okay, so what's that? Retrograde ejaculation. Right, retrograde ejaculation. So this part from the NCERT done. These are the diagrams here: spermatogonia, spermatozoa, interstitial cells. Right, so this part is also done. Right, the hormonal control part is also done. So hormonal control mein LH, FSH, right, the way they trigger the production. So see, so look at this, right, pituitary will release LH and FSH. It will act on the testes. So LH and FSH trigger testosterone production in the testes and estrogen production in the ovaries. So male may, the secondary sexual characters are penis and scrotum will grow, facial hair growth, larynx elongate, lowering voice, shoulders they will broaden, right? The body, armpit and the pubic hair will grow, musculature increases, right? Body becomes wide. Okay, so negative feedback. This is the negative. When too much of testosterone is there, that is also acting as a negative feedback. Now the gonadotropin releasing hormone should stop, right? So this is the diagram here, right? This diagram is not that appropriate. So see, this part is completely, this is actually the nucleus of the Sertoli cell. So this is the Sertoli cell. So these different stages of spermatogenesis are present here. So spermatogonia and the Sertoli cells are associated with the help of the proteins. So you call them as N-cadherin. Done, they are going to join it. So here you people can see, right? So now I'll ask you one question and then we'll proceed. 100 secondary spermatocytes will give you how many sperms? Quick. 100 secondary spermatocytes will give how many sperm? Answer this question, guys, quick. Answer this question quick. Please be quick. 100 secondary spermatocyte. Very good. So after looking at the answers, I'm sure, right, that you didn't get even a single word. Are you kidding me, guys? One secondary spermatocyte will give you 2 spermatid, means 2 sperms. So 100 will give you 200 sperm only. Right? If I will write 100 primary spermatocyte, because it will undergo the meiosis, right, will give how many, you will form how many sperm. Then you can write the answer as 400, but not here. Use some brain, right? Use some brain. So this is the NCERT part. You guys can read it, right? You'll be able to understand each and everything here. These are the stages, right, students? So now we will talk about the sperm structure, but before that, you see early stage spermatid, then elongated spermatid. The changes there, obviously I told you, the main thing that you can remember is ki histones, they'll be replaced by protamines. Sorry. Just allow me a minute. Okay. Done. So here you can see spermatid, which is rounded, having more cytoplasm. When it gets changed into the sperm, right, having less cytoplasm, the residual cytoplasm will be removed. Plus you will see in the sperm, acrosome formation will be there, right, which is having the license, right, that will help during the fertilization. Plus mitochondria, look at the way they are arranged. Flagella formation will be there. The nucleus, students, the nucleus, it will become condensed. How come it will become condensed? Because chromatin will also become condensed. So these are the changes that are going to take place. So histones will be replaced by protamines. So this is what we have to discuss. So here you people can see, right, about the hormones also, right. Spermatogenesis starts at the age of puberty due to the increase in the secretion of gonadotropin releasing hormone, right. So it's a hypothalamic hormone. It acts on the anterior pituitary gland, then secrete LH and FSH. So LH acts on the Leydig cell, stimulates synthesis and secretion of androgen. So here you are talking about the testosterone, right, that will help in the spermatogenesis. And FSH acts on the Sertoli cells and it will stimulate secretion of hump factor. So this part is already done and dusted, okay. So acrosome, who forms it? Golgi apparatus. That's again a PYQ, right. Which organelle forms it? Golgi apparatus. Okay, so here how many? The tail is there, right. This is the flagellum part. So obviously flagella or tail will have 9+2 arrangement of microtubules. Okay, 9+2 arrangement of microtubules. And moreover, right here in the neck region also, can you see the centrioles here? So in the sperm you will see proximal and distal centriole. So centriole, which is having 9+0 arrangement, right, which is also acting like a basal body. So this one will form the flagella of sperm, and this one will pass to the egg during the fertilization. Is that clear? And here you see the mitochondria, right. So it is basically spirally arranged, provide the energy during the sperm motility. So this is basically known as, right, chalo, one by one we'll discuss it, otherwise you guys will get confused. So this part clear? So here you people can see there is a head, there is a neck, a middle piece and the tail. When you are talking about what? When you're talking about the sperm structure, right? The plasma membrane, it covers the whole body of the sperm. We have to follow the NCERT here. So sperm head is elongated, having haploid nucleus, right? And it is having a cap-like structure known as, students, acrosome. So this is filled with the enzymes that help in the fertilization. Jaise acrosome mein you will see the license. So this is a PYQ. Which organelle forms acrosome? So the answer is yes, please. So the answer is Golgi apparatus. Am I clear? So middle piece, right, it is having numerous mitochondria that will provide energy for the movement, right. So human male ejaculate 200 to 300 million sperms. So this part is already discussed, right. The semen part is already discussed. So few points from the sperm section. Okay, so let's discuss that. Okay. Few extra points. Take. Not much. So neck, you know that it will have proximal or distal centriole. So this distal centriole is also acting like, as I said, basal body, right. In the cell chapter also, students, we have discussed. So through this beta, axial filament will form, right. That is going to form the flagella. That's what you have to remember. So let's suppose this is the, like this part is the neck region. Then you have the middle piece. So obviously around this axial filament, as I said, you'll see spirally arranged, you know, mitochondria part here. So that we also call it as Nebenken sheath, right. We also call it as Nebenken sheath. That's what you guys can write. Okay. Right. So part of axial filament that lies in the middle piece and is covered by spirally arranged mitochondria is called Nebenken sheath. Sorry. Done, students? Sure? Done, students? Any doubt? And do you know there is one more word, that is manchete. So manchete, it also formed, matlab it is a microtubule, kind of microtubule sheath, right? Like behind the acrosome there is a nucleus now, so posterior part of that you will see that there is a kind of temporarily formed sheath, that is manchete, which will form when there is a transition of spermatid to the sperm. So that is not much required. So this is all about this part, students. Now we have to start the female reproductive system. So I'm thinking that let's talk about the female structure first and then let's take a break. Okay, oogenesis we can complete after break, or you want the break now? You tell me. Arey yaar, it's just 2 hours. Show some enthusiasm, guys. Show some enthusiasm. Achha, we started at 5:15. Hi, yaar. Right, so let's start the female part. What say, guys? Let's discuss about the structure, then let's have a break. So whatever majority says. Acha, break. Chalo, let's break you. Hai na? So what should I break? Your nose or legs? Okay. No, not 8 to 8:30. Chalo. So let's have a break. Let's meet at 8:15. Hi everyone, I hope I'm perfectly audible and visible. I hope I'm perfectly audible and visible to everybody. How are you doing, guys? Done with dinner? Yes, please. Are you people done with your dinner? Huh? Are you people done with your dinner? Okay, so let's get started. So now we will be talking about the female reproductive system, okay? So that is what we are going to discuss. Done? That is what we are going to discuss. Let's talk about the female reproductive system, okay? So when it comes to the female reproductive system, you know it very, very well that here we are going to talk about the ovaries, we are going to talk about the ovarian ducts, the uterine duct, the fallopian tube rather, right? The uterus, the vagina, right? And the female external genitalia. So that is what we are going to discuss. Ready for it? Yes, everybody. Ready for it? Sure? Sure? Are you sure? Uh-oh. Sure, students? Okay, so in the female reproductive system, we will talk about the pair of ovaries, pair of fallopian tube, uterus, vagina, right, female external genitalia, even here we are also going to talk about, right, mammary glands, secondary sexual organ. We are also going to discuss about the other accessory glands. So that is what we need to talk about. So before starting it, let's see the diagrams first, okay? So here you people can see, right, this is of course the side view. Can you see that, students? So obviously here, this is the pelvic region and you can just clearly see that the urinary bladder is there. Just behind it, can you see the uterus? Yes, everybody. Sorry. Can you see the uterus? Yes, please. Can you see the uterus? Then you can see this pubic symphysis. Can anybody tell me what pubic symphysis is? This is a homework. No, not the homework. This is a question. Anybody? What is this pubic symphysis? Then you can see the urethra, clitoris, which is basically also considered as the reduced penis. So it is also the erectile tissue, right? It is also made up of erectile tissue, labium minora, majora, vaginal orifice. Orifices means opening. So this is the NCRT diagram, right? And then here you guys can see the cervix. Okay, the rectum is there, the part of intestine. This is the vagina. Okay, and here you have the anus. So obviously the question can come from the diagram part as well. So this is something that you people should know. So any doubt here? Yes, everybody, any doubt here? This is the adipose tissue, student. This is the adipose tissue. So see this, the better one. So here you can clearly see the bladder, right, outlining. This is the uterus part. Then can you see the ovary, uterine tube, right, the rectum part, the anus part, the vagina, urethra, clitoris, labia majora or minora. Just have a look. Just have a look. Very good, Haritha. Excellent. Very good, excellent. Okay, so this is all about the diagrams. So now see this front view. So now students, when you see, this is the pair of ovaries. So can you see that these ovaries are attached to the uterus here? Yes, please. So this is nothing. This is what the ovarian ligament. Okay, this is the ovarian ligament, right, which is helping this ovary to get attached to the uterus. When you look at this part, this is the fallopian tube. So this funnel-like structure here is infundibulum. The widest part here is ampulla. This is the isthmus, very thin part here. And this is the uterus part. Okay, this uterus, right, when you talk about the uterus, we also use the word hystera for it. We also use the word womb for it. And this uterus is pyriform. So when we use the word pyriform, it means, students, pear-shaped. What is the meaning of being pyriform? It is the pear shape. So this is the ovarian ligament. And can you see this part? Can you see this part? So this part is marked as the round ligament, mesometrium. This is not we are going to discuss. We will talk about the mesovarium. This is the broad ligament part. So broad ligament part, ovarian ligament part, and the mesovarium, that is what we are going to discuss. So see, this is the uterus part, and here, can you see this canal here? This is the cervix. Okay, so this is the uterus, this is the cervix, this is the vagina, and then the external genitalia will be there. So just look at this particular diagram. Can you see that? Right, it is in the case of a non-pregnant woman, right? Look at this uterus, my dear students. Just look at it. And in the case of pregnant women, just look at it. Just look at it. Right? Look at the difference here. Right? Just look at the difference here, students. Can you? Yes. Guys, I really want you people to revert here in the chat, right? If you can, then very good. Otherwise, tell me, I'll end the class. Right? It's too cold here. If you are not interested in studying, I'll end the class. No worries. Right? No worries. Okay, so that is how it is. Look at the position here. This is the round ligament, the way it is present. Now, see, in the case of a pregnant woman, right? That is the difference here. Okay, students, this is the difference here. So this is the complete diagram. Okay, so just have a look, the diagram, the position of all the parts. It should be clear to you, right? Then everything will be easy. Okay, then everything will be easy. So ovaries, we know that when it comes to the pair of ovaries, they are the primary sex organ. Why? Because we know that here the gamete formation occurs. Yes, please, everybody, please focus. Because gamete formation occurs and it releases estrogen, which is again steroidal in nature, and even progesterone. Okay, now we will talk about the ligaments. So obviously we know that, we have just seen that the ovaries are attached to the uterus, and of course uterus has to be fixed there. It's not like that ovaries, they'll just fly like this. You can imagine it in this way. This is ovary. Let's suppose this part is the fallopian tube, the complete part. This part is the uterus, something like that. But what you have to remember, guys, that these I'm sorry, not well. So these ovaries are also fixed there. Okay, so firstly, when you talk about the ovaries, I told you they are the primary sex organ. They are 2 to 4 centimeter in length, right? They are what? They are 2 to 4 centimeter in length and are connected to uterus and pelvic wall. Right, uterus and pelvic wall. So ligaments are going to attach it. Right, ligaments are going to attach it. So there is a question from the ovarian ligament as well. Right, there is a question from the ovarian ligament as well. So ovarian ligament, what will that do? That will support and stabilize the uterus and the ovary. So when you are talking about the ovarian ligament, you can just simply write it. Right, now let's give the title ligaments here. and then you people can simply write first thing is ovarian ligament, right? Attaches ovary to uterus. What is the function of this? Attaches ovary to uterus. Obviously, if it is attaches this ovary to the uterus, it is going to support it, right? It is going to do what? It is going to provide the support to it. Next point is what? Next point is uterosacral ligament. Next point is what, beta? Uterosacral ligament. So do you know what sacrum is? Yes? Anybody? Anybody? Yes, please. Uterosacral. Obviously sacral, sacrum. Do you remember the vertebral column? Right, CTLSC, cervical, thoracic, lumbar, sacral, coccyx. So this is what you are talking. So it will connect uterus to sacrum and prevent, right, because it has to fix it. So it is going to prevent its interior, right, it is going to prevent its anterior movement, right, it is going to prevents uterus anterior movement of uterus. And what about the round ligament? There is one more ligament which we call as round ligament. So this ligament, what will it do? Obviously it is also going to support, so it stops posterior movement. That's how you can remember. Okay, it is going to stop posterior movement of uterus. So that is how it is going to work, students. So other than that, we will also discuss about the broad ligament. But before that, let me remind you that when it comes to the ovarian ligament, in the ovarian ligament, you will also see one thing, students, right? Sorry. That it will connect ovaries to the uterus, but it doesn't contain any blood vessel. Okay? Okay? Yes? Have no blood vessels. This is what you people can write here. It doesn't have the blood vessels. Now, we have the broad ligament. Now, it's a two-fold, right? Two layers fold. Like actually, this is what broad ligament. Firstly, I'll show you the diagram here. You see this part. Can you see that? This part is the broad ligament, right? This part. This is the ovarian ligament only. This is the ovarian ligament, this one or this one. But this part that we are talking here is, it is basically beta. It is basically what? It is basically the, yes, everybody. It is basically what? Do let me know, people. Quick. Do let me know. It is basically what? It is the broad ligament. So when you people are talking about the broad ligament, so in the case of broad ligament, what you people are going to notice that it is two-layered fold, and it is the fold of peritoneum, right? It is a two-layered fold of peritoneum, and it will enclose everything. It's a two-layered fold of peritoneum that encloses, you can see that it was even attached to the uterus part as well. So that encloses uterus, even the ovarian ducts, right? So see, it is what? It is peritoneum. I already told you that peritoneum is going to do what? Peritoneum is going to cover the viscera, right? Peritoneum is going to do what, students? Peritoneum is going to do, it is basically going to cover the viscera. So it is a two-layered fold of peritoneum, having the uterus, having the ovarian duct, and of course the ovaries are also the part. So it will provide support and contain other ligaments as well, right? So it provides support and contains other ligaments as well, and contain other ligaments as well. So this is about the ligaments here. Then you have mesoovarium. So what we discussed in the case of male reproductive system, we talked about the mesorchium. Yes, students, what we discussed? Yes, please. What we discuss? We discuss about the mesochium, but here we are talking about the meso-ovarium. That is again the fold of the peritoneum only, and that fold will attach the ovary to the broad ligament. Are you getting my point? So meso-ovarium, what is it? It is the fold of peritoneum that connects ovaries to broad ligament. So I hope all these terms are clear to all of you. Yes, please. I believe all these terms are clear to you, right? So broad ligament of uterus will get attached to it. Is that clear? Is that clear? Yes, please. Is that clear? Sure? Sure? Done? So this is about the meso-ovarium. Now, this is all about the ligaments. Now, the next part that we have to discuss, because ovaries in detail we will discuss once we'll start the internal structure, we will discuss once we'll start the oogenesis. Then let's talk about the ovarian duct or the fallopian tube or uterine tube or uterine duct, right? So when you talk about it, we first start with infundibulum. What we have to discuss here first, infundibulum. Now understand one thing. It is a funnel-like structure. See, look at this particular part, students. Can you see that? Can you see that? This is the infundibulum, right? This is the infundibulum. Imagine these are the ovaries. So it is close to the ovaries. It is very close to the ovaries. That is a PYQ. We will write it as well. So it is close to the ovaries. Right, it is close to the ovaries, and when you look at the infundibulum, as I said, it is funnel-like, right? So here you will see some finger-like extensions are there, right? And they are what? They are the fimbriae, not even considered as the true parts of fallopian tube. So overall, this fallopian tube, you people can see here ovary. This is the uterus. So what exactly is going to happen here? The ovaries, they are going to release the secondary oocyte, these fimbriae. Right, we'll pick it that up. Now, the epithelium here, this epithelium inside the ovary, it is all ciliated. It is having cilia. So that movement, right, that beating of cilia will help to push this secondary oocyte, will carry it to the ampulla. So what is ampulla, students? It is the widest part here, right? And here it is the site of fertilization as well. It is a place where fertilization is going to occur, and this isthmus connect this part to the uterus. So this is what we have to discuss now. So as I said, that firstly we are going to talk about the infundibulum, which is basically the funnel shaped, okay, and that's a PYQ. It is the part which is closest to, close to ovary. So that's a PYQ. So you need to write it, right? Which part is the close to ovaries? Which part is the close to ovaries? It is the infundibulum. Is that clear? Is that clear? So overall fallopian tube is what? It is 10 to 12 centimeter long, comma, paired structure, right? 10 to 12 centimeter long, comma, paired structure. Is that clear? Is that clear? Is that clear? Right? So it is long, comma, paired, comma, tubular, comma, ciliated, extends from posterior part of ovary yes, so extend from posterior part of ovary to the uterus, right? So as I said, which part we have to discuss? It is infundibulum, and that is, as I said, funnel shaped. And what's the PYQ, students? Right, what is the PYQ? That it is close to ovary. This is actually the part close to ovary, not the fimbriae. Am I clear? It is not the fimbriae that is close to the ovary. It is this. Is that clear? Is that clear? Sure? Any doubt? Done? Okay? So whatever we have seen in the diagram, the edges of infundibulum have finger-like structures, right students? And that finger-like structures are what? Finger-like structures called fimbriae that collects, you have to mention it students, that collects eggs, or I would say the better thing is secondary oocyte when discharged by ovaries. Okay, when discharged by ovary. Is that clear? So that is the role of the fimbriae. What is the role of the fimbriae, everybody? Yes, students. What is the role of the fimbriae? That they are going to collect that egg. Is that clear? Is that clear? So after ovulation, this is going to collect the egg. Is that clear? Sure? Sure? Okay. Are you sure? So infundibulum. Next is ampulla. So for ampulla, you can simply write widest part, as I said, plus site of fertilization, right? Site of fertilization. So infundibulum continues into the ampulla. Infundibulum leads to ampulla. So infundibulum is the funnel shaped, ampulla is the widest. And then students, what else do we have? Isthmus, which is the narrow, right? Isthmus, what's that? It is the, right, what is this isthmus? Isthmus is the narrow part of the fallopian tube, right, having narrow lumen. Okay, so narrow part it is, and its lumen is even narrow. So just keep it in your mind. Is that clear? So it is the narrower part, right, and having the narrower lumen. Is that clear? So this is about the parts here. Okay, so fallopian tube overall, what will it do? It will conduct the ovum towards the uterus, okay, by peristalsis. Is that clear? So what this fallopian tube overall is going to do, or even if it is the zygote, by peristalsis. So I believe you know what peristalsis is. So it is the rhythmic contraction and relaxation, because muscular part is also there in the infundibulum. So that is something you have to remember. So any doubts? Any doubts? So this is what it is. Next part is, students, it is uterus, okay, which is also known as hystera, which is also known as womb. Am I clear? So it is inverted, pear shaped, right? So when we are saying pear shaped, it means it is pyriform. Is that clear? Is that clear, everybody? So we are going to talk about the uterus, right? So in the case of uterus, what is it? It is the inverted, pear shaped, or the pyriform structure. Am I clear? Yes, please. Am I clear? This is what uterus is. Inverted pear shaped, or I would say pyriform structure. Is that clear? Yes. Yes, everybody. Is that clear? If there is any doubt, do let me know. If there is any doubt, do let me know, guys. Do let me know. So it is the hystera or the womb. Done. So it is the inverted pear shaped muscular structure lies between rectum and urinary bladder. That's how I'll write, urinary bladder and rectum, right? That's how it is present. Is that clear? So it is the inverted pear-shaped or the pyriform structure, muscular structure that lies between urinary bladder and the rectum. Am I clear? That lies between urinary bladder and the rectum. So any doubt here? Yes, please. Any doubt here? Sure? Sure? So beta, is it hollow or not? Is it hollow or not? Yes, please. Is it hollow or not? Is it hollow or not? Yes, please. Is it hollow or not? Guys, it is hollow. It is hollow. Right? Of course it is hollow. That's the basic point. Wait. I'm not talking about the layers yet. So let's say this is the uterus. Of course, it is inverted, it is pyriform, it is hollow. So this part is basically the fundus, right? This part of uterus is basically the fundus where the implantation occurs. So this part, you'll call it as the body. This part here, the cylindrical part here, can you see that in between? This is your cervical canal, okay? And here you have internal sphincter orifices, that is opening, and this one is the external, okay? Then this part next to it is vagina. So in the case of this uterus, you people can see that this part is the fundus here, then the body, and then this cylindrical part, okay? Then the cylindrical part which will further leads into the vagina, it is basically the cervix. Is that clear? It is basically the cervix. So definitely it is hollow. Okay, definitely it is hollow. Done? Done? Right? So upper expanded part is what? Right? What is this upper expanded part? Upper expanded part is the body or the fundus, while the lower part is cervix. Is that clear? So fundus is that upper dome-shaped part of the uterus. Okay? Okay. Done? Done? So this is the upper. This upper part is what? This is your fundus. This body, this is the body. Obviously, it is narrow, and here towards this side, it will be more narrow. And the cylindrical part is cervix. So is that clear if I write it in this way? Or do I need to write it in the points? Is it okay? Will it work? Yes, please. Will it work? Yes, please. Done? So body is the main part here, right? And it will continue with the cervix. Done? So cervix having narrow cervical canal as well, right? And this cervix, it joins with the anterior part of vagina. Okay, anterior part of vagina. And do you know that, students, that cervical canal, do write it, plus this vagina, together they make the birth canal. Do you know that? This cervical canal plus vagina, together they make what? They make the birth canal. Is that clear? Is that clear? Done? Done? Any doubt? Sure? Done, beta? So, body of uterus communicates with cervix by internal opening Sphincter muscles are present there, and cervix communicates with vagina by external OA. So this is what you have to remember. Okay, here internal, here external. This part is what? This part is vagina. Am I clear? This part is what? This part is vagina. Any doubt? Any doubt? Sure? Sure? Yes, please. Done. So students, just a minute. So write it down. When you talk about the vagina, it is basically the copulatory organ. Okay. In the females, this one is the copulatory organ. It is fibromuscular. But remember, non-glandular. This is important. It doesn't have the glands. Right? External genitalia is having the gland, but in the vagina, no glands are there. So just write it down. It is the copulatory organ. It is fibromuscular. It is non-glandular. Am I clear? Right? It is non-glandular. Am I clear? Then we'll talk about the histology. Okay. Then we'll talk about what? We will talk about the histology of uterus. So in the case of uterus, remember, we'll be talking about 3 layers. Okay. So outermost is going to be perimetrium. Innermost is going to be endometrium. And the middle one. Okay. And the middle one is going to be myometrium. The middle one is going to be myometrium. So that's what you have to remember. So obviously the function of these layers is obviously definitely of protection. So this one to clearly I can write here protection. Now when you talk about the myometrium, myo word is associated with muscles. Okay, so myometrium is having which muscles, students? Smooth muscles, right? Smooth muscles, and we know that they are involuntary, right? We cannot control their function with our own will. It is involuntary, students, right? These smooth muscles, they grow too much. They become the largest one during the pregnancy, and right, the rhythmic contraction, right? Peristalsis basically. So rhythmic contraction of these muscles help in childbirth. Do you know that? The rhythmic contraction of these muscles help in childbirth. And when you talk about the endometrium, it is innermost glandular. So if it is glandular means it will have glands. It will have gland means it is going to provide some secretion. So can I write it like this? That it is innermost glandular, that is secretory, and the implantation, do write it, implantation of blastocyst occurs here. Yeah, implantation occurs here. So yes, even this is a previous year question. Even this is a previous year question. Kindly write it down. Quick. Kindly write it down, guys, quick. I hope it's done. Yes, please. Sure? Is it clear? Confusion. What is the confusion? So So guys, yes, even this endometrium undergoes cyclic changes during menstrual cycle. Done? So this is it. So this is about the histology of the uterus. This is about the layers. So you should know myometrium is having the smooth muscles. And do you know when a female is pregnant, to induce the labor pain, the oxytocin injections are given. And oxytocin, right, we will discuss its function, right? So remember, this is the oxytocin, okay, that acts on smooth muscles. I told you already, oxytocin will be released by it is formed by hypothalamus, stored in neurohypophysis, and then it will be released. So it acts on smooth muscles, cause their rhythmic contractions. Am I clear? Cause their rhythmic contractions and relaxations. So basically it will cause the peristalsis there. So this oxytocin. Done. So now the next part that we have to discuss is the external genitalia. Let's talk about it. So I hope this diagram is clear. I hope these ligaments are clear. So mesovarium word we have discussed, ovarian ligament we have discussed, endometrium, myometrium, perimetrium we have discussed, students, right? And uterine cavity, you know, body of uterus, fundus of uterus, and then see internal OS, external OS. This is the cervical canal, cervix, and this one is the vagina. Any doubt? Any doubt? Ovary is also done, right? Yes, please. So this is what you need to know. Up to this part, all clear? Yes, guys, be quick. Yes, please, be quick. Done, students? Chale, so let's talk about external genitalia. Is that clear? Let's talk about external genitalia. Okay, so guys, this is the vagina. It is around 10 centimeter long, and do you know that its opening, not completely, but yeah, its opening is covered by hymen. Okay, its opening is covered by hymen. This is what you have to remember. Okay, its opening is covered by hymen. So this is what you can see. We will use this diagram later. Now we will talk about the external genitalia. So you just have a look, then we will discuss it. So when you look at this part, okay, so this part, can you see that this is the, right, this is what? This is the mons pubis, right? That cushiony layer that is going to protect. The fat is present there. You can see that the hair growth is also there, right? If you can see the structure here, you will see this tiny finger-like structure which is known as clitoris. That clitoris, okay, you hear these words, I'll tell you. It is also considered as the reduced penis, right? Because it is also made up of erectile tissue, plus it is reduced penis, and plus it is also made up of what? It is also made up of erectile tissue. So it is also having that loose skin, we call it as prepuce, right, students? And then, can you see this part? So this is the, this part is the vaginal opening. This part is what? Vaginal opening. Just above this, this is the urethral opening. So in the case of females, the urethral opening and the, right, the urethral opening and the vaginal openings, they are different, right? Is that clear? Is that clear? Is that clear? Sure? Sure? So vaginal opening is there, Then here you have the external orifice of the urethra, right? And then can you people see this? This, right? These are the folds of, right? These are the skin folds, students. So this part is basically labia minora, right? This part is basically labia minora, right? Labia minora. I'll write about it and see this fold in the surrounding, this fold, right? This fold, this is going to be labia majora. Okay, this is going to be your labia majora. So this is what you have to remember or know. This is the anus part, anal opening, obviously. So firstly, tell me the location clear, then we will discuss about it. Yes, please. Is it clear? Up to this part all clear? Then just a minute, see this. Right here you will have the better idea. So this is the mons pubis, mons vermin is also the name, I'll check it. So then this is that prepuce, that loose fold of skin, glands of clitoris. So this is the clitoris part. So here you have the vaginal opening covered by the hymen, and this is the urethral opening, okay, from where the urine will come out. So the period's blood, it comes out from this vaginal opening. Is that clear? So now here in this diagram, this part is very clear. You can see the labia minora here, and these folds of skin are the labia majora. Is that clear? And here, because vagina, it is non-glandular. Just now we have discussed it, right? Orifice means opening. So I told you it is covered by hymen, right? We'll come to this. So here in the surrounding, can you see the glands here? Can you see the glands here? Because now we are just going to write about it. So you people can see here you have greater vestibular glands, also known as Bartholin's gland. You have lesser vestibular glands also, right? These are the openings here. These are the openings. They'll provide the lubrication during the sexual intercourse. Am I clear? Firstly, tell me, am I clear? So if this part is clear, then we will write about the external genitalia. That is also known as vulva. Right? So if diagram part is clear, then it is going to be easy for you. Okay, so here mons pubis, labia majora, minora, clitoris, and all will be considered. So mons pubis, which is also known as mons veneris, labia minora, labia majora, clitoris, and even we also discuss the perineum there. We'll show you that part also, right? So yes, obviously hymen part also you will consider, right? That opening also we'll talk about. So this part is clear, guys. Any doubts? So if it is clear from the diagram part ki okay, I know the position, then do let me know in the chat section quick. Then do let me know in the chat section, guys, quick. Quick. People, quick. Yes. So it is the fatty cushiony tissue. Just a minute. You can see it or mark it from the NCERT as well. So here देखो fallopian tube, fimbriae, everything is given na. So let's highlight this part from the NCERT. Okay. So layers done. Yes. So this is the external genitalia. Okay. Hymen, clitoris, everything is there. So mons pubis, which we are also using the word veneris. So it is a cushion of fatty tissues covered by skin and pubic hair. Am I clear here? So it is a cushion of fatty tissues, right, covered by the skin and the pubic hair, which is meant for protection. Then labia majora, you know that it is towards the outer side. So it is the fleshy fold of tissue which extend down from mons pubis and surround the vaginal opening. Is it done? Labia majora, right? Yes, please. Yes, please. Labia majora. So this labia majora is homologous to scrotum. It is what? It is homologous to scrotum. You can simply write down. It is homologous to scrotum, right? Inner side is hairless, but outer side having sebaceous, sweat glands and hair follicles. I hope you are marking it. That's why I asked you to keep your NCERT with you all. So labia majora, it is homologous to scrotum. Is it done? It is homologous to scrotum. So inner side is hairless, students, right? Outer side will have sebaceous, sweat glands and even the hair follicles. Am I clear? And even the hair follicles. Is that clear? Is that clear? Sure? Any doubt? So labia minora, it is paired folds of tissue under labia majora. So opening of vagina is covered by hymen. So can you tell me why is it partially covered by hymen? Anybody? Why is it partially covered by hymen? Anybody? Why? Why partially? Why not completely? Because students, if hymen will complete the vaginal opening completely, then how the period's blood, the menstrual blood comes out? Number one. Number two is when there is the sexual intercourse, it is believed that when female have the first sexual intercourse, the females when they have their first sexual intercourse, due to the penetration of the penis, the hymen, it's just tear off, right? But that results in a basically the bleeding, and that indicates the virginity. But that is not true. Why? Because hymen can even rupture if a female is very active in the sports activity, like if she's doing the horse riding, even the cycling, and sometimes it just rupture. And many times it can also happen that even after the sexual intercourse, it stays like that. So this is something we have to keep in our mind. So having the hymen or the rupturing of that hymen during the first sexual intercourse, and that indicates virginity, is just a myth. Is that clear? Is that clear? Right, so clitoris, it's a tiny finger-like structure. I told you it is considered as equivalent to what? It is reduced penis. What's that? It is considered as equivalent to reduced penis. It is also having the erectile tissue, but it doesn't have the 3 bundles. It is having the 2 masses. And what's that? That is your corpora cavernosa. This is what we have here. Is that clear? Is that clear? So it's a finger-like structure. You should know its location. You should know its position. So where is it placed, guys? It is placed at the upper junction of 2 labia minora. So yes, I can ask or frame the question from this part. Instead of upper, lower can be given. Instead of labia minora, labia majora can be given. So when you write such question, when you solve such question, you should be clear about it. Right? You should know what exactly it is saying. So what's the point here? That it's a tiny finger-like structure which lies at the upper junction of the 2 labia minora, not at the lower, but at the upper junction of the labia minora, just above the urethral opening. Am I clear? Above the urethral opening. So it is often torn during the first coitus. I told you about the hymen already. So this is something that you have to keep in your mind when it comes to the external genitalia here. If there is any doubt, then you do let me know. Okay? Do let me know, guys. Done? And just like males, skin, yeah, glands of clitoris is covered by prepuce. Okay? And that's how we are discussing it now, urethral opening or the vaginal opening. Yes, please. So that is how it is. Urethral opening or the vaginal opening. So urethral is on the anterior side and vaginal is on the posterior side. Is that clear? Is that clear? Sure? So just above this urethral opening, upward to this, right, at the junction of the labia minora, you will find the clitoris there. Is that clear? Is that clear? Sure? Sure. Now, this is the diagram where about the Bartholin ducts is mentioned. So, as I said, a Bartholin glands, right? So, here they produce a fluid, or I would say alkaline fluid, during sexual excitement. So, that also you can write down. Quick, write it down. Quick. Just quick, guys. So this is the greater vestibular gland. Done. So see, it is the Bartholin gland, greater vestibular gland. So as I said, it produces an alkaline fluid, right, during sexual excitement. So you can write down. So this one is homologous to, because it is producing a fluid during the sexual excitement. So it is homologous to your corpus, also known as bulbourethral gland. I believe this is clear. So its location is on the either side of vagina. So vagina is non-glandular, but it is present on the either side of the vagina. Right, students? Right? Sure. So there is lesser, greater na, lesser vestibular gland is also present, which is also known as Skene's gland, students, which is also known as paraurethral gland. I hope you guys are aware of it. This is another gland. Yes, are you aware of it? Are you aware of it? Do write it. Thank you so much, guys. Do write it. Thank you, Maria. Thank you so much. Thank you, beta. So we have the greater vestibular gland or the Bartholin gland, and lesser vestibular gland or the Skene's gland or the paraurethral gland is also there, right? Paraurethral gland is also there, right? Yes, please. Do you know its function? So it is located near urethral openings, right? It secretes a fluid that it secretes a fluid that even it secretes a fluid. Basically, see, when you are talking about this, it is considered as the female prostate, right? Because see, it also protects from the UTIs, urinary tract infections, and even when there is the sexual intercourse, right? And even when there is the passing of the urine, so it helps, right? So it provides the lubrication, it releases a fluid, so that helps in both, beta, even in the urinary health, even in the sexual health. That's how it works. Okay, that's how it works. So you can write it secretes a fluid that helps in urinary and sexual health. So that's something you people can keep in your mind. So this is about the glands. Once done, do let me know. Then we will start the ovary part. Okay, let's talk about the ovary, the structure of the ovary. Till then, you please write down quick. Yes, please do it. If it is done, do let me know in the chat section. Okay, guys, done? Chaliye. Next is ovary. Okay, when you talk about the ovary, the outermost covering here is going to be see. So this one is going to be germinal epithelium. Here also the story is same, right? PGCs, they'll find their way to the gonads. Now, beneath germinal epithelium, tunica albuginea is present. Okay, which layer is present just beneath it? Tunica albuginea. So I am writing it here. So this tunica albuginea, what's that, students? This is the white fibrous connective tissue. What's that, people? It is the white fibrous connective tissue. Am I clear? Now, this part is what? It is the stroma, right? It is the stroma of the ovary. And here in this stroma, you'll see the outer part is the cortex, students, just like kidney, just like adrenal gland. This is going to be the medulla. Okay, cortex and medulla is there. If you ask me that, ma'am, is there a clear-cut differentiation? No, there is no, but yeah, medulla will have the blood supply, and this cortex is the site of oogenesis. Okay, cortex is the site of oogenesis. So this part is basically the hilum, right? So blood vessel enter or leave through this part. This is the hilum. Blood vessel will enter or leave through this part. So medulla, it will have obviously connective tissue, the blood supply. So firstly, tell me this part is clear. Done? Done? Yes, please. Is it done? So you are talking about the ovary. We are saying the outermost covering is the germinal epithelium, which is having the cuboidal epithelium. Then we are saying that layer just beneath it, what is this? This is tunica albuginea, and below that we have this complete part is what? This complete part is stroma. What's that, students? This complete part is what? This is the stroma. So this stroma is having two parts. It is having the outer cortex and it is having the inner medulla, right? So if you ask me, ma'am, is there any clear cut differentiation in these two? No. Medulla part will have abundant blood supply. Is that clear? Is that clear? Is that clear? Yes or no? Is that clear? Yes or no? Done. So this is the cortex, which is the site of oogenesis, right? And here in the cortex, you people are going to see the follicles. So what are follicles? Group of cells. So we'll see many follicles here. So follicles are not added further after birth. They all will be added before birth only, during the embryonic stages only. That's what we have to discuss. But up to this part, if it is clear, do let me know. So I'll be back in a minute. Till then, you draw it and tell. Yes, please. Done. Sure. So look at the diagram. See, this is the ovary under the microscope. Can you see that? These are that follicles we are talking about. Okay. These are the follicles that we are talking about. We'll discuss about it. And look at this. Right, they say we'll start with them because we are talking about the cortex region. So we are saying, see, this is the cortex region. As I said, there is no clear-cut demarcation that this is cortex, this is medulla. But in the cortex part, what will occur? Oogenesis will occur. So oogenesis is what? It is the formation of the ovum, right? But when you talk about this topic, you cannot read it without understanding folliculogenesis. Folliculogenesis. Genesis again means formation. Folliculogenesis means the formation of the follicles, and within these follicles ultimately the female gamete will form. So now in general, I'm just telling you this, like here these are some terms which you have to keep in your mind. We have the primordial follicle, primary follicle, secondary follicle, tertiary follicle, and then comes the pre-ovulatory follicle, or basically the mature or the Graafian follicle. Then the secondary oocyte will release from it, and then finally the corpus luteum will be there, and then it will degenerate. So these are the things which are present there in the cortex, right? This is something that we see in the cortex. We start from the primordial, then primary, then secondary, then tertiary, then pre-ovulatory, or in simple words, mature follicle, right? So that is what we have to discuss. Let's discuss the same diagram from the NCERT, and after that we will talk about the mammary glands. So here also you can see, can you see that primordial, then primary, secondary, this is a tertiary showing the antrum. And if you will see, like you know na, this year also NCERT just got upgraded. So if you check out your NCERT, in that NCERT ovum is written, but actually it is not ovum. It is the secondary oocyte having metaphase to arrest. Okay. And then see corpus luteum, yellowish, temporary endocrine gland that releases progesterone, and then it will get degenerated. Okay, so this is something that you guys can rectify in your own NCERT. Okay. Okay, so now because you have to understand this topic, so we will also talk about the oogenesis, and slowly with that oogenesis part you will get to know about this, and then I'll tell you about the follicles. Okay, then I'll tell you about the follicles, right? So firstly I'll tell you the stages here, then I'll relate it with I'll tell you the follicle formation, then I'll relate both the topics. Okay, so oogenesis, so it means formation of what? Formation of ovum, of course. And when will it start? See, it starts in embryonic stages. So spermatogenesis, it starts when the boys, they will attain the puberty, but it will start during embryonic stages and completes at the time of fertilization, not before that. At the time of fertilization it is going to complete. So now rather than writing, you just focus on what I'm trying to teach you. Like, so when a baby girl is in her mother's womb, like if you talk about me, my mother, right? When my mother was in her mother's womb, I was in my mother's ovary. It is like that. Okay, it is like that. So what is the point here? The point is when, like I said, the oogenesis, it is the formation of ovum, and it starts during when? It starts during the embryonic stages. This is what we have discussed, guys, that when will it start? It starts during the embryonic stages. So this is what we have to continue as well. Okay, this is what we have to continue as well, right? So what exactly is going to happen, right? Yes, so what exactly is going to happen when we talk about it? Okay, so okay, one second. So what exactly is going to happen? Like when a baby girl is in her mother's womb, in her ovaries, right? The story is same. Primordial germ cells, they'll find their way there, right? And they are going to form oogonia. Like they'll form oogonia. That oogonia, they proliferate. When we say they proliferate, so it means it is going to increase the number, right? Divides and increase number. Divides and increase number. That's what I'm saying. Divides and increase number, so that there will be so many oogonia. Firstly, tell me one by one, right? One by one, tell me. This step is clear? So this is not the meiotic phase yet. This is a kind of pre-meiotic phase or the proliferative phase that we are going to discuss, right? This is the pre-meiotic phase, or pre-meiotic means before meiosis phase, that we are going to discuss now. So tell me, any doubt here? Any doubt here? Yes? Yes, please. Yes, please. Done? No, guys, I'm not feeling tired. I'm not well. I have fever and the cold, right? So that's why just looking for the chair again and again to sit. It's just like I'm not well, just fever is there, but it is all manageable. So they will proliferate. So by the time, in humans, right? In humans, when female is in, that the gestation period is for 9 months, right? So when it is the second month of gestation period, around second month of gestation period, so you will see that ovaries will have approximately 3 lakhs, around 3 lakhs oogonia, and by 5th month it is around 7 million oogonia, right? And then proliferation stops, and it enters in meiosis. So what am I trying to tell you? The information is not given in NCERT. So all I want you to understand is that you just listen to me now, right? You just listen to me now and tell me that up to this part all clear? No, I don't want to end the class, guys. You just listen to me and you just work on it. Okay, so up to this part all clear? Yes, please. Yes, please. So, जैसे in the female, like initially when the baby is developing in the mother's womb, so initially that when the development is there, there is no differentiation male and female. Initial part I'm talking about, like the initial weeks of the pregnancy, right? And then that PGCs will find their way, right? And then the germ cells will form, and that's how the proliferation will start. So that is why I have mentioned the word key in the humans. I'm talking about the embryonic stages now. Okay, I'm not talking about after birth thing. I'm just talking about the embryonic stages at this point of time. So I want you people to listen to me carefully and to absorb this information. Ki by the second month of the gestation period, second month means already like 1 month will 4 weeks, 8 weeks. So by that time, you know, little bit of the idea is there, right? So by that time, I would say that when you're talking about the female, right? So by the, I would say when 7th week onward now, right? So the clarity will be there, male, female, SRY gene, but that is a different science. Let's not talk about it. So here, like by the second month of the gestation period, you'll see the ovaries will have around 6,000 of oogonia. By 5th month, 7 million oogonia. Then proliferation will stop, and then it will be like, chalo, let's enter in the meiosis now, and let's enter in the meiosis, and let's become what? Let's become what? Let's become the oocyte. This part is clear. This part is clear. Done. This part is clear. Enters in meiosis, and of course, firstly there will be the meiosis one, and then become oocyte. The stages are same, but yeah, this process is different, right? As I said, I'm just talking about the embryonic stages at this point of time. Okay. Now, these oogonia, still embryonic stages. It will enter in meiosis 1. Of course, meiosis 1 is having the prophase 1. So it will enter in the meiosis 1, in the prophase 1, and then obviously oogonia, it will grow, right? It will become oocyte. Of course, your primary oocyte it is, has entered in the meiosis 1, then in the prophase 1, and then there will be an arrest. Okay, then there will be an arrest in the diplotin stage. I hope you remember these things, right? Female part is little, I won't say complicated, but yeah, not exactly like the males. So I hope you remember this part, that when you people talk about the meiosis, we have meiosis 1 or meiosis 2. Meiosis 1 will have same trick. It is PMAT students, prophase 1, metaphase 1, anaphase 1, and telophase 1. Then when it is prophase 1 is having 5 substages. I hope you remember L, Z, P, D, D. Everybody. Prophase 1 is having 5 substages, L, Z, P, D, D. Or you can say, little Zara, please do not dance. Right? Little Zara, please do not dance. So here, leptotene, zygotene, pachytene, diplotin, diakinesis. Leptotene, zygotene, pachytene, diplotin, diakinesis. Leptotene, zygotene, pachytene, diplotin, diakinesis. Is that clear? Is that clear? So see, the oogonia, right, by the 5th month of the gestation period, as I said, the proliferation will kind of stop, and even after birth there is no proliferation. All the oogonia will form during the embryonic stages only. So by the 5th month it will be around in millions, 7 million, or sometimes more than that. Then what will happen? They will start growing. They'll form the oocyte. It will enter in the meiosis 1. Then there will be an arrest, right? An arrest in the diplotin stage. It is also known as dictate arrest. Right? Dictate arrest. And that's not like that all the oogonia or oocyte developed here, they will grow into that ovum. Of course not. But this is going to happen here. Is that clear? Is that clear? Done? So, this stage maintains until female matures. That is from birth to puberty. Like, it will be in embryonic stages, and then from birth to puberty, this stage will stay, right? This stage will stay. Any doubt? Any doubt? Sure? Sure? Any doubt here? So this is going to happen, right? This is going to happen in the embryonic stages. Now, even in the embryonic stages, I'll come to that stages later. But remember, in the embryonic stages, right, here in the cortex part like we were discussing, let's suppose this is your primary oocyte. When this primary oocyte, okay, I'll write it. When these primary oocytes are covered by, right, they are covered by follicle cells, right, like ovarian cells. You can also call them as ovarian cell, follicle cells. They are present in the cortex. They have lost the potential to make the gametes. So I'm writing it here like this: have lost the potential to form gametes. Okay, so when the primary oocytes are covered by the follicle cells, then they form follicles. Follicle cells or follicular cells or ovarian cells. Like let's suppose this is your primary oocyte, just giving you the example. Let's suppose the follicle basically means group of cells. Okay, so this is the primary oocyte. This is covered by these follicle cells. So, and this part is the primary oocyte. So together you are calling it as, like let's suppose if it is the beginner, it will be your primordial follicle. When it will grow a bit, right, then you will call it as primary follicles, right? So this is the difference in the follicle and the oocyte. Okay, this is the difference in the follicle and the oocyte. So when it is covered by the follicle cells, right, or the follicular epithelium, right, so it is the ovarian follicle, right? So rather than saying the primordial follicle, just to avoid the confusion right now, I would say that you can call it as the ovarian follicle. Okay, okay, so primary oocyte plus, oocyte plus follicular epithelium. Follicular epithelium, it forms what? It forms the follicles, ovarian follicles. Done. Oocyte, when they are covered with the follicular epithelium, then you are calling, right? Yes, please. Then you are calling it what? Then you are calling it as the ovarian follicle. Any doubt? Any doubt? Please do let me know. Please do let me know. Any doubt? Thank you, beta Rabia. Thank you. Any doubts? So this part is clear. So whenever your oocyte will be covered by the follicular epithelium, you'll call it as the ovarian follicle. So ovarian follicle, follicular cells, this is how it is. Done. So this is what we discussed till the embryonic stages. Now next line. From birth to puberty. So from birth to puberty. Matlab, firstly I'll tell you, at the time of birth, ovaries have millions of oocytes, right? Millions of oocytes. No further oocytes will be added after birth. Am I clear? Am I clear? I hope you are writing the notes as well. So then from birth to puberty, what is going to happen? Students, follicular atresia will be there. That is follicular degeneration. So imagine, millions of follicles were there at the time of birth, right? But later on, what's going to happen? from birth to puberty. So at the time of puberty will happen. So follicular atresia, follicular degeneration is there. So at the time of puberty, each ovary contains, right? Each ovary contains 60,000 to 80,000 follicles. Each ovary, write down, right? Each ovary contains 60,000 to 80,000. Yes, please, 80,000 follicles. Sure? So you first write it, then we'll discuss it. Guys, sure? Yes, please. Done, students? Sure? Okay. Now, so this is what you have to remember. So remember, before birth, a small amount of, like even before birth, right? Just say, let's suppose this is your primary oocyte. It will be covered by, I told you already, follicular cells. It is not big in size. So you can call it as primordial follicle. This is the beginner, no? So what can we call it, guys? What can we call it? Primordial follicle. Everybody just write it down in the chat section. What do we call it? We call it as primordial follicle. Is that clear? Right? So this is initially it is a single layer of follicular cells, or you can even call them as granulosa cells. Am I clear? Am I clear? Yes, please. So it is oocyte plus surrounding follicular cells, right? And how these follicular cells are connected? These follicular cells, they are joined by gap junctions. You know, continuous cytoplasmic connections. So that is how they are connected. So any doubt? Any doubt? Sure? Done, beta? So this is going to happen. So at the time of birth, and even it is believed, just see, here we have mentioned now that millions of follicles are there, right? So at birth, millions of follicles in each ovary, and then, as I said, they will be degenerated later. That is what I have discussed. So now I am saying that it is believed, right? It is believed that these granulosa cells provide nourishment to the oocyte and release a ovum maturation inhibiting factor, right? So it is believed that in the childhood, okay, right, it is believed, you understand this part beta, you will get to know each and every line today, okay? So it is believed that during the childhood, what is happening, that throughout the childhood, right, throughout the childhood, these granular cells, they provide the nourishment to the oocyte and release ovum maturation inhibiting factor. Why? So that factor, right, that factor will keep it in the primordial stage, right? That factor will keep it in what, right? It will keep it in that primordial, in that dictator rest only, right? It is believed, okay? It is believed that it will keep it in the dictator rest. Now you know that at the time of puberty, okay? Now what is going to happen? So at the time of puberty, you know that what's going to happen? Gonadotropins, they release. Yes, please. What will release, people? At the time of puberty, what will release? Gonadotropins will release. Yes or no? Gonadotropin will release, yes or no? Right? Yes, please. And remember, so gonadotropins release, that is your FSH and LH. So what will they do? They will act on these follicles, right? What will they do? They will act on these follicles. So during the initial time, right, the secretion of these hormones is less. And then it increases, okay? Then it increases. Are you getting my point? Then it increases. Are you getting my point? Done? Yes? Sure? Yes or no? So initially what is going to happen? Initially, ovary along with follicles start growing. Initially, when even the hormone is less, because see, imagine it in this way. What is going to happen? Initially, the hormonal secretion is less, right? But still, the hormones are when they get released from this pituitary, then what is going to happen? Initially, the ovary will increase. The size of the ovary growth will be there. The ovum, the oocyte will also increase. But initially, what is going to happen? Initially, what is going to happen? That growth is not too much, right? That, like, you can say that some cells, right? Some cells of this, I would say, follicular cells, yeah, granulosa cells, they'll start dividing. So obviously, initially primordial follicle it was, then it will become primary. So as I said, initially the growth is less, but later on, what's going to happen? What will happen after that? After that, guys, the hormonal secretion will increase. The hormonal secretion, it will increase. Now when it will increase, these granulosa cells, even their division will start. Now when the hormones are also increasing, so during that month, what's going to happen? Around 6 to 10 follicles, they'll start growing. Their granulosa cells will start growing. So it is not like that you will get one primary follicle. There will be many. They'll start growing. They'll start growing. They'll start growing. So tell me, up to this part all clear? Up to this part all clear? So first stage of follicular growth is moderate enlargement of. So that's why I have mentioned here that initial growth, right? Initially ovary along with the follicle start growing. But here in the bracket I'm writing it moderately. It's not too much, right? It's not too much. It's a little bit of the growth. Okay, beta? Okay. So it is the moderate enlargement of, right? Right. It is what? It is the moderate enlargement of, okay, moderate enlargement of what? This oocyte. There is the moderate enlargement of oocyte. Am I clear? Initially this is going to happen. Am I clear? So up to this part, if everything is clear, then do let me know, everybody. Yes, please. It is just 2 to 3 folds, not much. Done? Done? Yes, please. Done? Is that clear? Yes or no? Right, then after that, then follows growth of granulosa cells in the follicles. So of course, what will they become? They will become primary follicle, right? Primary follicle. So you can write FSH. FSH specially helps in proliferation of these granulosa layer. That's why the name is follicle stimulating hormone, right? So tell me, this part is clear? Write down. Write down. So after birth, that primordial follicle, it is under the influence of the hormones. So it will start growing a bit. Initially, the growth is slow. Then the growth will accelerate, especially because of the FSH. So the more and more layers of FSH will start growing. So this part is clear, guys? Quickly tell me. This part is clear? Up to this part is clear. And please maintain the notes here. That will be important. Yes? Done? Of follicles. So FSH helps in growth of 6 to 10 follicles per month. So it is not just the one. Done? It is not just the one. Is that clear? Is that clear? Sure? ठीक है? So next line is rapid, that is fast, proliferation. So guys, are you writing it? Yes or no? Please tell me. If you are not writing it, then I will move very fast. You tell me. Are you writing it? Yes, please. Are you writing it? Guys, say yes or no. Wake up. So initial proliferation of granulosa cells, right? Initial or rapid proliferation of granulosa cell give rise to many layers, right? Give rise to many layers. Okay, give rise to what? Give rise to many layers of these cells. Now the next part is, now the surrounding, just imagine, then later on I'll make the diagram. Let's suppose this is your granulosa cell. Okay? In the starting, this is your granulosa cell. Then under the influence of hormones, let's suppose this is growing. It was primordial, then it started growing. These layers, they will also increase. Let's suppose this is your primary. Just imagine. Now, when it is further growing, because it is under the influence of FSH, so you will see, because where is it taking place? It is taking place in the ovary. So ovary mein further stromal cells are also there na. These are the granulosa cells or the ovarian cells or the follicular cells. So in the surrounding also there are some other stromal cells. So what is going to happen? These stromal cells in the surrounding, students, they'll start expanding, and these stromal cells which are covering these granulosa cells, they'll start making a layer known as theca interna. I am not talking about the externa yet. I am saying around these granulosa cell, under the influence of FSH, because granulosa cells are also dividing, right? The stromal cells in the surrounding, they'll expand, they'll form theca interna, and it will develop the ability. It will have the receptors, or not the receptors, I would say. It is having the receptors of the LH as well, and it will release the estrogen. Is that clear? Is that clear? Yes, please. Yes, please. Yes, please. So write it down. Okay. So write it down. So as the follicles expand, the stromal cells surrounding it get condensed, not expand, they get condensed and form a layer called theca interna. What is the name of the layer? Theca interna. So if you have to visualize, this is how we can just imagine these are the granulosa layers. So surrounding will have condensed layer, and then what are we calling it? We are calling it as the theca interna. Tell me. Theca interna. Is that clear? Is that clear? Sure? Sure? Done, beta? So this theca interna releases estrogen. Releases estrogen. This is important. You can write down. Now, theca interna is having theca externa in its surrounding, like with further development, of course, in its surrounding, and this is highly vascularized, but this is not going to release the estrogen, students. So it is just highly vascularized. It is what? It is highly vascularized. Am I clear? Am I clear? Done? Yes, please. Is that clear? and this is highly vascularized and develops as a and forms a capsule. So this is your, right, I'll come to the zona pellucida thing as well. This is your secondary follicle, right, students? This is your secondary follicle when it is having this theca externa interna. So initially the growth is less, right? So primary follicle, so slowly primary follicle will develops into the secondary follicle. So this part is clear. This part is clear. Wait. I'll talk about the zona pellucida as well. Okay. Now, theca interna cells, right? Because they have LH receptor, students. So luteinizing hormone act. Further, the estrogen will release. So theca interna cells keep on releasing estrogen, right? It will keep on releasing what? It will keep on releasing estrogen, yes, for few days. For few days, that results in the formation of a fluid-filled cavity, right? That will result in the formation of a fluid-filled cavity called antrum. So then antrum will form. So antrum is the cavity having, it is also known as, it is having liquor folliculi. So exactly what exactly it is? It is the estrogen in it. So because granulosa cells are, they'll keep on releasing the estrogen. So that will be accumulated there. A cavity will form. So you can imagine it like this. So let's suppose this is the oocyte, right? It is having the granulosa cells, then theca externa interna. So let's suppose these are the theca interna cells. So they have developed they have formed a cavity, the antrum here. Am I clear? Antrum here. So antrum is having the estrogen. Is that clear? Okay. So antrum appeared in the mass of granulosa cells. Okay, I'll show you the diagram now. In the mass of granulosa cells. So look at this part. Can you see this? See. So tertiary follicle will show the antrum. Okay. Yes, please. So tertiary follicle will show the antrum. In the granulosa cell, this mass will form. Done? Yes, everybody. Is it done? Right? Students, done? So early growth, this early growth, like now, yes. So antrum is there. Now you will call it as your tertiary follicle. So it is the tertiary follicle in which antrum is there. Right? In the NCERT also, we have just read it. Is that clear? Sure? Sure? Yes, please. Yes, please. Sure. So early growth up to antral stage is due to FSH only. Do you know that? Due to FSH mainly. Later, this growth is accelerated. Now initially it is the FSH mainly. So FSH is acting on the follicle cells. Follicle cells are proliferating, plus they are releasing the estrogen. So yes, we can say that this estrogen is released due to the action of the FSH. So FSH is acting on the follicular cells, proliferating, then releasing estrogen. So later this growth is accelerated. Later this growth is accelerated, and then FSH receptors, yes, initially FSH receptors develop on that large granulosa cells. Yes, students, on that large granulosa cells. So this is the positive feedback, right? Like FSH is releasing, acting on estrogen. Further, that is also increasing the FSH receptor. Is that clear? Right? Right? So later this growth is accelerated, and then the FSH receptors developed on that large granulosa cells. This is the positive feedback, and these cells become, because they have more receptors, and these cells become, at last we will conclude also, more sensitive to FSH. So tell me, up to this point, just recall it. Close your eyes, recall it again. Right? Close your eyes and recall it again. If there is any doubt, do let me know. Quick. Yes? Are you able to? Sure, guys? Now next is what? Next is what? Then FSH and estrogen. Right? Now see, this is important. This is very helpful for the menstrual cycle as well. So they combine, right? Both of them. They combine, and due to which what will happen? Now the LH receptors will start forming. I told you na LH receptors are there, but initially it is the FSH. So then what is going to happen? Your FSH and estrogen, what will they do? They'll combine. They'll promote the LH receptors, luteinizing hormone receptors on that theca interna cells. And then what's going to happen? LH stimulation will also increase. And now it is not just the FSH, LH will also start playing its role. Is that clear? Is that clear? Yes or no? Yes or no? So estrogen is increasing. When estrogen increases, yes, it promotes the LH secretions. Okay. Okay, so together they help in the proliferation of that thecal cells and the secretion as well. Right, students? So once that antral cell begins to grow, the oocyte itself also enlarges in the diameter when that antral cells, when this antrum starts growing. Is that clear? Okay. Any doubts? Any doubts? Sure. Guys, I have seen one thing, na? Matlab these days students are really not interested in quality content, right? Devansh Arora, ma'am, practical. Practical of what, beta? What practical are you talking about? Huh? I think you are a female, hai na? And if you are not, I'll give you some injections, okay? So that you also start producing the eggs. Then you will see the practical, don't worry. So guys, like see, those who are actually here to learn something, the interact and answer the question, because this time I'm teaching this oogenesis in a little different way, right? I'm covering the hormonal part there also, right? Individually, I'm not writing this is primary, this is secondary, this is tertiary. I want you people, right? I want you people to imagine this thing. So what is happening after the birth? We know that ovaries, the follicles, many follicles, they are degenerated from birth till puberty. Now, when it is the time of puberty, then we know that gonadotropins are going to release. That gonadotropins, FSH and LH. FSH will act on the follicular cells. So slowly, your granulosa cells or ovarian cells or the follicular cells, they will increase their number. They'll proliferate. The layers will increase. So initially, we had that small primordial follicle. Then it will grow a little. Initially, the ovary will grow a little. Even the oocyte will grow a little. That is the primary follicle, right? Then the layers will start proliferating. Slowly they will. Then we will have the secondary follicle. What will we have? Secondary follicle. Now when that layers are growing, the surrounding stromal tissue, that the cells will also condense. They will form theca interna first, then outside the theca externa. It is the theca interna that will have the receptors that will release the estrogen ultimately. Okay, then that theca interna will release the estrogen, and that estrogen, students, will form a cavity, antrum, in that granulosa cell mass, and then slowly that cavity will grow. Because why? Because positive feedback loop is going on. Estrogen will release, granulosa cells will have more FSH receptors, more FSH is there, estrogen will increase. Now these two hormones collectively, FSH acting on ovaries, they are releasing estrogen, right? More estrogen means more LH receptors will also start forming, and that LH, that luteinizing hormone, right? That luteinizing hormone, is that clear? That luteinizing hormone, what that luteinizing hormone will do, now that luteinizing hormone will also start releasing. So estrogen and LH together, now they will act. Is that clear? Is that clear? Yes. What's the doubt? What's the doubt? Quick, this topic will take time. Rest everything will be as per NCERT, right? Quick. So up to this part all clear? Up to this part all clear? Yes, whosoever is any doubt, please ask me. Then we will solve the questions. Sure? Are you sure? So they combine to promote LH receptor, luteinizing hormone receptors on granulosa cells. Are you getting my point? On what? On granulosa cells, thus allowing LH stimulation, right? Thus allowing what? Thus allowing LH stimulation along with FSH. Am I clear? Along with FSH. So this part is clear, everybody? Yes, this part is clear, everybody? Done? Sure? So this increase in estrogen plus this increase in LH, what will it do? So it increases proliferation and secretion of thecal cells. Of thecal cells. So this part is clear. So both of them, they will start acting like this. Is that clear? Okay. So now obviously once antral cells grow, oocyte also grow, right? It is present at one side and is covered by granulosa cells. Is that clear? And is covered by granulosa cells. Understood? And is covered by granulosa cells. Done, students? Done? Yes, please. So once antral cell, no, antral cell, no, it is antral follicle. Once antral follicle grows, right, the in size, oocyte also grow. It is present at one side and is covered by granulosa cells. So you know, just now this is happening. Okay, yes please, this is happening. Now all of sudden what is going to happen? This will happen, right? That follicles, basically what is happening, antral follicles, they are growing. Oocyte, it will also grow. It enlarges in diameter. What will it do? It enlarges in diameter, and it is present at the one side, covered by the granulosa cells. Now you know just what is going to happen after the week of this growth, right, and just before the ovulation. This is a proliferation is going on, okay, growth is going on, okay. Right before the ovulation, or I would say after the growth of this, right, after a week growth, right, a week of growth, right, but before the ovulation, rest all the follicles, because I told you in the starting 6 to 10 follicles they'll grow, right? So after a week of growth, or just before ovulation, one follicle will start growing excessively, and rest other degenerates, and rest others degenerate. One will start growing excessively and rest other will degenerate. It happens, right? Just when? Just before the ovulation this is going to happen. Are you getting my point, students? Just before the ovulation, after a week of growth, but before the ovulation, okay, right, one of the follicle start growing excessively and rest other, what will happen? They will degenerate. So this largest follicle, it continues to grow and other stops, and it will form Graafian follicle. Okay. Grow and form Graafian, or I would say mature follicle, right? Graafian follicle. Write down the mature follicle, the largest one. Is that clear? Is that clear? Okay. Done, students? Done. So now what is happening? Because follicle is growing excessively, na? This follicle, your oocyte is placed. I'll talk about the oocyte growth as well. Right now I'm more concerned about the folliculogenesis, right? So you people can see, this is going to be the antrum here, okay? So now the next line that you all can write is ki oocyte is eccentrically, means it is not there in the center. It is eccentrically placed and is covered by the granulosa layers, granulosa cells. That is it. Yes, please. This part is clear. Sorry, this part is clear. Sorry, this part is clear, guys. Yes, please. Yes, please. Now next line. In primary follicle, and even bache, I have read a standard book. In that standard book, na, there was a standard book, Gilbert. In that, though, it was mentioned in the case of primordial follicle only, in the embryonic stage only. So, in primary follicle, right, in primary follicle, right, a glycoprotein layer layer called zona pellucida. What is the name of the layer? Zona pellucida. In the bracket I'm writing it. It is the primary egg membrane. What's that? It is the primary egg membrane. So in primary follicle, a glycoprotein layer called zona pellucida appear in between oocyte and granulosa layer. Right? So if you read the book, in that book it is mentioned as primordial follicle. Okay? And now in this mature Graafian follicle, the granulosa layer, so you can imagine it in this way. This is your oocyte. Here you have that glycoprotein layer, so it is acellular. Of course, here you have all the granulosa cells. It is like that, right? That is what I'm trying to show you. So now in this mature Graafian follicle, the follicular cells, right? The follicular cells near the zona pellucida, right? Write it down. Quick. Guys, quick. Write it down. Yes, please. Right, near the zona pellucida become elongated and form secondary egg membrane called corona radiata. Called corona radiata. Write it down. Yes, please. Yes, please write it. Quick. Sure, guys? Done? So this is the corona radiata. No, zona pellucida is acellular. That's why I have mentioned now that it is a glycoprotein layer. So if it is made up of glycoprotein, means it is not made up of cells. See, this is how you can make it. Just a minute, huh? I'll show you. I'll show you guys. Just allow me a minute, please. So if I have to show you this one, like let's suppose So here you can see, guys, that this part, okay, this theca externa, this theca interna, plus these granulosa cells. I'm writing it like this. This is your theca externa. Ultimately, it is also from that ovarian cells. This is your theca interna. Done. Then these are your follicular cells, or I would say granulosa cells. These three are what? These are the follicular cells, or I would say the ovarian cells. Are you getting my point? These are the follicular cells, or I would say, right, these are the follicular cells or the ovarian cells. Is that clear? Is that clear? Right? And here, this is your oocyte. Okay, this oocyte is covered by zona pellucida. That's the primary egg membrane. And then in its surrounding, you will have corona radiata, secondary egg membrane. Okay, and then the, see, innermost layer of granulosa cells form cumulus. This is again the granulosa cells only, oophorus, that surrounds the egg in oviduct, or the secondary oocyte in oviduct. So this is the terminology that you have to remember. So this theca externa interna cells made up of ovarian follicle, granulosa cell made up of follicular cells or the ovarian cells, granulosa cells also. Antrum is developed, oocyte is eccentric, the zona pellucida is developed by the oocyte, corona radiata is formed by the egg, right? And the innermost layer of the granulosa cells here, it will form cumulus oophorus that will surround the egg in the oviduct. So this is what I'm telling you, right? This is what I'm telling you. Is that clear? So write it down. Okay. Take care. So about, you can write down here that about 2 days before ovulation, the amount of LH secretion increases, right? And before, and 18 hours before ovulation, it is at peak. Okay, it is at peak. Done? Remember this. So it is the LH surge that is required for final or full growth of follicle and, right, for final or the full growth of follicle and, right, for final and the full growth of follicle and for ovulation. So if there is no LH, there will be no ovulation. So write it down. Quick, do it. Guys, note down, it is important. Quick. I hope it's done. Yes, not 18 hours, it is basically 16 hours. Okay? So next line that you can write is कि prostaglandins are also very important for ovulation. No prostaglandins that also cause the contraction. I hope you remember. No ovulation. Right? No ovulation. Yes, please. Is it done? Yes, please. So students, LH is very, very, very important. You know that in the animals, the most abundant protein or the is collagen, right? Collagen is present in our extracellular matrix. Now, collagenase is also released by this LH. Prostaglandins are also released by this LH. So, when the LH amount increases, okay, this will convert these follicular cells into the progesterone forming cells. Slowly, slowly, the lutein protein will get accumulated. You know it very well. So, when there is the LH surge, because it is releasing the collagenase, some plasminogen activators are there, your prostaglandins are there, then what will happen? Like, can't you see so many things are forming, too much pressure that will result in this rupturing of the follicle, and due to which the secondary oocyte will come out. Now this secondary oocyte will come out, but now here in which stage it is in the metaphase 2 arrest. Basically now I'll relate it with the diagram because I majorly focused on the folliculogenesis where students make the mistake. So I'll show you a few diagrams here so you'll be able to get it. So see this, like initially primordial follicles are there, then right, basically don't look at the diagram, the point that I'm going to say. So slowly see the just a minute, look at this part. You have the primordial follicle, you have the oocyte, ovarian cortex cells are there, granulosa cells are there. Then I told you, I have mentioned it, even if you will read the Gilbert, in that way it is mentioned that even during the embryonic growth in the primordial follicle stage, the zona pellucida forms. So here granulosa cells are there, oocyte is there, and you know that zona pellucida. I told you zona pellucida is acellular. It is just made up of what? It is made up of glycoproteins. Okay, students. Now slowly see the growth is taking place. The growth is taking place. These granulosa cells, their cell number is increasing because FSH secretion is also there. That is helping in the proliferation here. Now, when it comes to the tertiary follicle, right? Because now here you know that theca cells, they'll also form, right, students? The theca cells, they form, they'll release the estrogen. So estrogen will be released, it will be collected here in the mass of granulosa cell. It is forming the antrum. So antrum is having liquor folliculi, right? That is basically it is having the estrogen. Now as I said, many follicles they were growing, then all of a sudden one follicle will start growing too much, rest other will degenerate. That will grow, that will form mature follicle or the Graphian follicle, my dear students, right? That will form the mature follicle or the Graphian follicle. So in that Graphian follicle, you can see the oocyte is eccentric. It is having zona pellucida. So near to it, the granulosa layer, which is near to the zona pellucida, that will form the corona radiata cells, right? And the innermost layer of these granulosa cell will form the cumulus oophorus cells, which will cover this in the oviduct as well. This is the antrum. So you have theca interna, you have these granulosa cell, you have theca externa, plus here the basement membrane is also there, that is separating it like this. So just have a look of this particular part. Quick. Just have a look. Quick, guys, quick. Quick. Please be quick. I hope it is done. Is that clear? Sure? So basically what is happening, now if you will ask me that ma'am, when will this dictate arrest will overcome? When your LH is at peak, right? When your LH level, like pituitary glands, they'll start releasing LH. That LH, right, you can write down, LH breaks the dictate arrest. So if it is going to break the dictate arrest, means the meiosis 1 will be completed, meiosis 2 will start. But in this Graafian follicle, from this Graafian follicle, it is not the full-fledged grown, yeah, fully grown secondary oocyte, fully grown ovum. It doesn't release, but it is the secondary oocyte which is in the metaphase 2 arrest. So this is the sequence that you guys can remember. See this, PGCs, oogonia. Okay, overall I'm telling you, it will form primary oocyte. That is again 2N. That will undergo meiosis 1. Okay? Of course, that will form secondary oocyte. Here, the division is unequal. Okay, it is haploid, and there is one more polar body, polocytes, that are also haploid. So this is of large size, this is of small size. So this one will undergo meiosis 2. It will form large one, ooted, the smaller one, polar body. I'll come to this part. Just let me write first. So that's the sequence that we have to understand now. So what exactly is happening? See this, guys. Your follicles are growing, early follicle, secondary follicle. It is the tertiary in which you will see the antrum. Then, right, and the second meiotic division will start here in the Graafian follicle. But when the LH is too much, then your dictate arrest will overcome, right? And then from Graafian follicle, when there is the ovulation, when there is the release of secondary oocyte, guys, so it is in the metaphase 2 arrest. Okay, it is in the metaphase 2 arrest. So this is something you have to remember. Wait, I have added a flow chart as well. So here you guys can see that what exactly is happening. So in the embryonic stages, your PGCs will form oogonia. Oogonia, they'll proliferate. They will enter in the division phase. Then there will be an arrest in the diplotin. So this is the embryonic stage only. The meiosis arrest will be there. So from birth to puberty, follicular atresia, that is degeneration of follicles, will be there. No follicles will be added after birth. So in the adult life, when the girl will attain the puberty, right, the first meiotic division will be completed, right? Why is it so? It is under the influence of the hormones. So when your LH level is high, the dictate arrest will overcome. The second meiotic division will start. But again, then there is an arrest. Arrest where? In the metaphase 2. In the metaphase 2. So the secondary oocyte, because it is not, the secondary oocyte has started the meiosis 2, but it is not able to complete it. So secondary oocyte is released, not the fully grown ooted, right? So this is something that you have to remember. So let me show you the diagram. Because see, menstrual cycle you have completed with it. Mammary gland will hardly take 5 minutes, and then we'll come back to, we'll be talking about the capacitation, and then finally, ye dekho, sorry, pre-antral phase and the antral phase part is there. So here you guys can see. Here, look at this diagram. Okay, so see this. Primordial, then primary, then secondary ovum is there in it, right? This is the mature Graafian follicle. So this liberated, it is not the ovum actually, it is the secondary oocyte at the metaphase 2 arrest. Am I clear? Done, beta? Am I clear? Sure, look at the diagram. Primary, then tertiary, then Graafian follicle. So it is in the metaphase 2, sorry, it is in the metaphase 2 arrest. Okay, you write it down in your notebook as well. Quick. Done, students? Yes, please. Sure. So quickly write it. So we have we have primordial follicle. Even this primordial follicle will grow a bit, right? So you will get these primary follicle, which will later grow at the puberty, but in the embryonic stages we see that. So then comes the primary follicle, then comes the secondary, then comes the tertiary, then you have the Graafian. So from that Graafian follicle, right, from that Graafian follicle, what will happen? The ovulation occurs. What will release, students? This will be the secondary oocyte, but here it is in the metaphase to arrest. Done. Why this ovulation will occur? It is due to the LH surge. It doesn't matter how much FSH is, it doesn't matter how much your other hormones are, but LH surge is required for the ovulation. Then after ovulation, right, after ovulation, this Graafian follicle, it will turn into the corpus luteum, right, that will releases what? Progesterone. So if there is pregnancy, this corpus luteum, the temporary endocrine gland, the yellowish body, it stays. Otherwise, it degenerates, it forms corpus albicans, the white bodies, and that is how it is. That is how it is. So here initially, right, it is in dictate arrest. When slowly these follicles grow, I would say in secondary or tertiary, right, because LH is also increasing. So dictate arrest, overcome, and then it enters in the meiosis, completes the meiosis 1, enters in meiosis 2. Is that clear? So pituitary begins secreting LH, due to which dictate meiotic block is broken. Is that clear? Is that clear? Yes, please. Yes. Any doubts? Any doubts? Guys, it doesn't matter how much you spam, right? It doesn't matter how much you spam. I'll complete today's chapter and I'm not going to take any class further, right? I'm not going to take any class further in Game of NEET 3.0, okay? Because I have seen that you guys are not sincere and you are really not interested in studying. Okay, so don't worry, I am going to finish this chapter today and then bye-bye from my side. I will not even take a single class. So this is it, people. Acha, this is, okay. So this is it, people. This is how it works. Okay, so overall, how come is the division? So now you can just focus on it. So now you know PGCs, they will form oogonia. So oogonia is diploid or haploid? Oogonia is diploid. That will grow in its size. That is going to form primary oocyte. That is again diploid. That is again what? That is again diploid. Okay? Because oogonia will grow. After that, what's going to happen? This primary oocyte, it will undergo meiosis 1. You know that this meiosis 1 will not be completed. It will be in the dictate arrest. Okay? But after attaining the puberty, it will be completed and the secondary oocyte will form. So in the case of females, this division is unequal. Okay? It will result in the formation of a large secondary oocyte, haploid, and a small, right, and a very small haploid polar body. Okay, so you will not see here the equal division. You will not see just like the sperms, right? In the case of spermatogenesis, what were we saying, right? I'll draw the comparison here, that spermatogonia 2n, then it was forming primary spermatocyte, again 2n, then that was forming secondary spermatocyte n, and they are 2 in number. Then this is the meiosis 1, then there is the meiosis 2. So here you people will see spermatid, 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 and spermatid. Then they'll form sperm. But here you will have secondary oocyte, of course haploid, polar body, of course haploid, but there is unequal division. You will have large secondary oocyte that will develop, that has to form the zygote. And then there is the meiosis 2. So meiosis 2 will start, but it will complete at the time of fertilization. So here you will have ooted, then again polar body. So sometimes students, they ask me, ma'am, whether 2 polar bodies will form 3. See, if this polar body will get degenerated, so this is 1, this is 2, but let's suppose if it doesn't get degenerated, then of course it will also undergo meiosis 2. It will again form 2 polar bodies. So then in totality you will have 1, 2 and 3. So that's why it is written 2 to 3 polar bodies. Okay, okay. So here we can write one thing. See, 1 primary spermatocyte, it will give you what? It will give you 4 sperm. But 1 primary oocyte will give you what? 1 ooted or egg. Am I clear? Am I clear? Now you tell me 200 secondary spermatocyte will give you how many sperms? Then 200 secondary oocyte will give you how many egg? This is what you have to fill in. Thank you, Nargis. Answer this. Quick. Answer this, answer this. We'll finish the menstrual cycle with it and then we'll discuss it further. Quick. Answer it. I'll sit for a while, you answer it. Quick, guys. Guys, quick, then we'll write the next part. Is it done? Chalo, I'll see the answers. So obviously 200 will give you 400 beta. See, 1 secondary spermatocyte is giving you 2 spermatid or 2 sperm. So 400, hai na? So it will be double. So that is it. But 200 secondary oocyte will give you 200 ooted or the ovum only. That is what you have to remember. That is what you have to remember. How come 800 beta? I have started from this part. Okay, so this is the comparison, right? This is the comparison. So let's see this from the NCRT part once and then let's discuss the menstrual cycle from it. There are 2 to 3 points more which I need to discuss about this, the fate of the corpus luteum, and it is the LH actually that will convert that thecal cell into the progesterone secreting cell, they'll have the lutein, then later they will release the estrogen. So we will discuss it. So now, so the process of formation of mature female gamete is called oogenesis, right? So it is initiated during embryonic development, number 1. Okay, so when couple of million of oogonia are formed within each fetal ovary. So no more oogonia, do underline this line, that no more oogonia are formed and added after birth. Done. These cells start division, enter into the prophase 1 of meiotic division, get temporarily arrested. So in the book itself, you people can write here that here we are talking about the dictate arrest. Which arrest are we talking here? It is the dictate arrest. Is that clear? Is that clear? So now you'll call it as the primary oocyte. So primary oocyte, when it is surrounded by the layer of granulosa cell, then we are calling it as the primary follicle, but it is primordial then primary. So yeah, it is true that even in the primordial follicle, your oocyte will start forming a layer, zona pellucida, right? So a large number of these follicles degenerate during the phase from birth to puberty. That is follicular atresia. At puberty, 60,000 to 80,000 primary follicles are present. So see, we have discussed the NCERT, we have clarified it, isn't it? Isn't it? Then the primary follicle is surrounded by more granulosa cell and a new theca and are called secondary follicle. So then they will transform into tertiary follicle. They have antrum. Theca layer is organized into inner theca interna, outer externa. So primary oocyte within the tertiary follicle, see, it is written here, grows in size and completes its first meiotic division because LH secretion has started here, right? So this is unequal division. So there is a large secondary oocyte and a tiny first polar body. Now tell me, NCERT is easy for you or not? Now tell me, is the NCERT easy for you or not? It's easy now? It's easy, right? Now it is. Yes, now it is. Now the next is the secondary oocyte retains bulk of the nutrient-rich cytoplasm, right? Obviously, because later it has to develop into the zygote. So first polar body, what will happen to it? It can, students, degenerate or it can stay. If it stays, it will go for meiosis too. So at present, we are not very certain about this. So tertiary follicle changes into Graafian. Secondary oocyte will form a new membrane called zona pellucida surrounding it, right? So the Graafian follicle, so they have mentioned that secondary oocyte forms, but if you will read the standard books, the foreign author books, so in that case it is even mentioned, right? So I was reading that Gilbert, in that it was mentioned that even in the primordial follicle stage you'll start seeing this. Okay, so in primary follicle also. So then you go as per the NCERT, no worries in that. So that's it. See, differences in the spermatogenesis and oogenesis are asked now. That is again a PYQ. So that's it. So this is the diagram that they have given. 1 second. 1 second. So this part. So kindly observe it. So fetal life may this is happening from birth to puberty, the degeneration. Then secondary oocyte will form first polar body, then second polar body and ovum. And if it stays, it will again divide. Okay. Right. So you check it out, and there is a just 5-minute break. Only 5-minute break. Then we'll start the menstrual cycle. You take the 5-minute break. I have to mention 3 to 4 points. Then menstrual cycle and the mammary gland, it will hardly take 30 minutes, and then that capacitation of sperm part, insemination capacitation part we will discuss, and then we'll stick to the NCERT only. Okay, okay, just do it. Done. So you can write the next line so that we can finish the topic. Okay. So LH, one second. So LH is having a special effect on thecal cells. This is what you have to remember. And so LH is having a special effect on thecal cells and granulosa cells. Do you know that? And finally, convert them into progesterone releasing cells. Okay, so that's what you have to remember. So if there is no LH, there will be no ovulation, students. Am I clear? If there is no LH, there will be no ovulation. Okay, no ovulation. And remember, after ovulation, once the ovulation is done, obviously the level of even the estrogen, then after that the level of estrogen will go down. Is that clear? And the level of progesterone will go up, because now the granulosa cells, they will start releasing the progesterone. Is that clear? Yes, please. Is that clear? Sure? Are you sure? Okay, so you guys can write one more line that after 12 days after 12 days of ovulation, corpus luteum starts involuting and its secretions will decrease. Am I clear? Yes, please. It starts involuting, and then after that, what's going to happen, students? After that, what's going to happen? Its secretions will be decreased. Its secretions will be less than. Okay? Okay? Sorry for this. So it's too much cold here. Done. So it starts involuting and its secretions decrease. That's it. Any doubt? Yes, please. Any doubt? So after that, your Cl will turns into the corpus albicans. Okay? Okay, so if fertilization will occur, the corpus luteum will stay, it maintains the pregnancy, the progesterone secretion will continue, but if it doesn't, then corpus luteum will degenerate. So it will, like if there is no fertilization within 9 to 12 days, it is going to degenerate. Understood? Right? Within 9 to 12 days, it is going to degenerate. Understood? Sure? Are you sure? So that's something that you have to keep in your mind. Okay? Okay. So let's move to the next topic. So here we go. So students, from this part, what sort of question can come? They can even ask you which hormones are pituitary hormones, like your FSH and LH, and they both are not steroidal. Estrogen and progesterone, they are the steroidal hormones. Remember, these are the protein hormones, but they are the steroidal hormones. So if it is a steroidal hormones, it is a class 11 concept. Their receptors, where are you going to see their receptors? Their receptors are intranuclear or intracellular receptors are there. Okay, so you can see initially FSH will release. When the FSH amount will increase, the follicle stimulation will also increase. So initially this follicle growth is due to the FSH only. Later you know that even the LH secretions also induce. When FSH, it acts on the follicles, you know that even the estrogen will also release. Are you getting my point? the estrogen will also release. Now look at this estrogen amount. So when this is LH is high, estrogen is also high, and high LH is responsible for the ovulation. So in comparison to that, FSH is less, and even it is quite less. So once the ovulation is done, we know that it will form the corpus luteum. So at this time, I told you already that see, the estrogen level will go down, but the level of progesterone will go up, and the FSH and LH will be at the same level. Are you getting my point? So this will regress. Then finally the corpus luteum will regress. So meanwhile, what are the changes that are taking place in the case of uterus? So initially when follicles are developing, initial stages where all the hormones are low, so menstrual cycle is going on, menses, bleeding phase is going on. Then slowly when estrogen level will increase, right, your FSH, LH is there, so you can see the uterine endometrial lining, it is getting thicker. Once this corpus luteum will release, you know that it will release progesterone. Its main function is to maintain the pregnancy, because whenever females are having the periods, the body, right, the body is preparing itself for what? The before periods, because basically every month body is preparing itself for what? Body is preparing itself for the pregnancy. If pregnancy occurs, then fine. If it doesn't, the menstrual cycle occurs. So progesterone, it will act on this endometrial lining, the thickness you can see, its glandular nature increases, and that is how it works. So this part is clear, this part is clear, yes, this part is clear. Then I'll give you guys the break. So see this. So let's discuss about that. So reproductive cycle in the female primates, that will include your monkeys, your apes, the human beings, it is the menstrual cycle, right? So estrus cycle, this is your homework. You'll tell me about the estrus cycle that you will see in the case of dogs, buffaloes, right? You have to tell me about this in the comment section. Then first menstrual cycle, it is menarche, very easy, but it is a PYQ. On an average in the case of humans, right, this cycle is, students, basically for 28 to 29 days. Okay, 28 to 29 days. Just for an example, right, just day 1, 2, 3, 4, 5. You can consider it as the bleeding phase, the initial phase, the bleeding phase or the menstrual phase. Right, what phase it is? Bleeding phase or the menstrual phase. Now when you talk about the day 6, 7, 8, 9, 10, 11, 12, 13 or the 14. So this phase, it is basically the proliferative phase, or I would say follicular phase, because here your follicular growth is going on. Then this 14th day, वैसे it is plus minus 2 days, this is going to be your ovulatory phase, the day of ovulation, right? So on an average we are saying na, the cycle is for the 18 days. Then 15, 16, 17, 18, 19, 20, 1, 2, 3, 4, 5, 6, 7 or 8. So this complete phase, this is the luteal phase or the secretory phase. Are you getting my point? This is the luteal phase or the secretory phase. So when this phase is there, all the hormones, right, be it your FSH or your estrogen, because you know that estrogen secretion will be stimulated by this, even your LH, even your progesterone, they all are low. So by the end of this phase, students, slowly the hormonal secretion will start. Now in the FSH, slowly follicular phase, FSH secretion start, then estrogen, and then slowly when this will be the time, right, then you will see the LH secretion will also start, right? So 14th day, day of ovulation, how is it possible? It is possible due to the LH surge, So after that, the amount of estrogen will be less. It will go down. But what will be high? Your progesterone will be high. So that's how you have to understand it. So this bleeding phase is for 3 to 5 days. It will last for this. You can say the proliferative phase is for 1, 2, 3, 4, 5, 6, 7, 8 days. Then this ovulation phase. Though we write it the 14th day, but it is plus minus 2 days. Then 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. These 14 days will go for the luteal phase or the secretory phase. So after the formation of this corpus luteum, so after 12 days, if it doesn't get fertilized, like if there is no fertilization, then this corpus luteum will start degenerating, and then it will result in, you know, the shedding of the lining, and then the slowly shedding of lining, the bleeding phase will approach and starts. Okay, so that is something you have to keep in your mind. Okay guys, and see, this time period, if a couple, they want the baby, so this, right, there's 14 days, or I would say this time period is basically, right, for the, if they want to conceive, then they can have the unprotected sexual intercourse in this phase. Okay, right? So when you talk about the rhythm method that we will discuss in the reproductive health, so in the rhythm method we see this thing, right, that ladies, they calculate their periods time, right, they calculate this, and accordingly, you know, they consider the safe period or not so safe period when they can have unprotected sexual intercourse, or when can they avoid. Is that clear? Is that clear? Okay, so this is about the menstrual cycle part. So see, so once the ovum is released during the middle of each menstrual cycle, so now what you have to do, see, you have to take care of one more thing. Just say here, the ovulatory phase, the menstrual cycle is for how many days? 28 days. So you have day 14th is of what? The ovulation na. So if the cycle is for 30 days, then you have to calculate. So day 14 will go for the ovulation. Then rest will be there for the luteal phase. Like you have to calculate it. You should know, right, that when will, which day will come. Chalo, I'll talk about it, but you first discuss this. So the major events of the menstrual cycle are shown in the figure. Chalo, we have discussed this. So menstrual flow occurs. It lasts for 3 to 5 days. Any doubt? Any doubt? Yes. Any doubt? Tell me. Yes, please. Done, students? So menstrual cycle results due to the breakdown of the endometrial lining of the uterus and the blood vessels, which forms liquids that comes out through vagina. And you know that is why hymen is not covering it fully, right? And don't worry, we'll also write the role of estrogen and the progesterone. Don't worry about that. Now, a menstruation only occurs if the released ovum is not fertilized. If it is fertilized, then the pregnancy will occur. Lack of menstruation may be indicative of pregnancy, but not always, because sometimes due to the hormonal imbalance, due to the stress, female can miss the menstrual cycle. Okay, students? Right. So the menstrual phase is followed by that. After the bleeding phase, there is the follicular phase, which I told you already is also known as proliferative phase, right, which is also known as proliferative phase. So during this phase, the primary follicles in the ovary grow to become a fully mature Graafian follicle, and simultaneously the endometrium of the uterus, it regenerates through proliferation. So in this phase, the endometrium will also regenerate. So these changes in the ovary and the uterus, they are induced by, right, in the levels of pituitary and ovarian hormones, right? So the secretion of LH and FSH, their secretion increases slowly, students. Follicular development will be there. Then estrogen will also release. That we have very calmly, we have discussed and mentioned while writing the oogenesis. Done? Done? So both FSH and LH attain a peak in the middle of the cycle about 14th day, but there is the LH is maximum. So this is the LH surge. This LH surge is there for the ovulation. And remember, even LH induces the prostaglandin secretion. So no prostaglandin, even then also there will be no ovulation. LH is required, prostaglandins are required. Is that clear? The ovulation is followed by luteal phase, the secretory phase, right, students? And the corpus luteum secretes large amount of progesterone. This is the anti-abortion hormone. It maintains the pregnancy. It will act on the endometrial lining. It will increases its secretory or the glandular nature. Is that clear? So the corpus luteum secretes large amount of progesterone, which is responsible for maintenance of endometrium. So endometrium important for implantation, right? So during pregnancy, all events of this cycle stops. And you know that females in the late 50s, in the late 40s or in the 50s, they get the menopause, so it will stop. So in the males also, if the androgen secretion decreases, then in their case also the gamete formation can decrease. Okay, so this is it. The diagram is important, and that is what you have to remember. Okay, that is what you have to remember. Is that clear? Is that clear? So guys, I have discussed this part in detail, so now I am expecting you people to read it from the NCERT. Is that clear? Sure? Are you sure? Now let's suppose if a female is having the menstrual cycle for 40 days, then what will be the, now we'll solve some question, day of ovulation. Then beta we will talk about the mammary gland. Okay, what will be the day of ovulation? So see, your luteal phase is kind of having fixed duration. Okay, so the follicular phase can vary. So 14 minus 14 days, so it will be 26, day 26. That's how you can count it. So always remember, you have to minus the 14 from it if you want to check the day of ovulation, because follicular phase duration can vary, right? But the corpus luteum phase, its duration is almost same. Is that clear? Done? Okay, so do write it down. So this information is important. Am I clear? This information is important. Am I clear? Chaliye, so let's Mama. Okay, guys, answer this question and tell me, are you able to solve it or not? Are you able to solve it or not? Yes, please identify the correct one. High levels of estrogen triggers the ovulatory surge. Oogonial cells start to proliferate, give rise to functional ova in regular cycles from puberty onwards. Sperm released from seminiferous tubules are highly motile. Yes. Yes. Oh, sorry. Yes, please. Exactly. Because sperms released from seminiferous tubules are not highly motile. They need to get stored in the epididymis. Then only they'll get the functional maturation. Right? Okay. So, seminal plasma, the fluid part of semen, is contributed by. Who's going to give the seminal plasma? Who's going to give the seminal plasma? Quick. Who's going to give the seminal plasma? Yes, please. Obviously 1, 2 and 4. Even the epididymis can also contribute. Then spermiation is the process of the release of sperms from. That is again a PYQ. So spermiation is a process of the release of sperms from, of course, from seminiferous tubule. Okay, then answer this. Yes, please. I like this question, guys. Solve it. So mature Graafian follicle is generally present in the ovary. Yes, so it's easy. You can just go for the most relevant option. You people can just go for the most relevant option. Sure, guys? It will be 11 to 17 days. This is going to be the most relevant one. And for the ovulation, I told you, you can go for ±2. Chaliye, next part. Which one of the following is not a male accessory gland? That's quite easy. Ampulla. Okay, chaliye, question number 9. Question number 9. Guys, question number 9. Quick. Let's solve this NCERT exemplar. Done? The spermatogonia undergo division to produce sperm by the process of spermatogenesis. Choose the correct one with reference to above. Okay. C, right? It is C. Good. What about the 10th? What about the 10th one? Head. Head will have the genetic material. देखो ना, acrosome is having the enzymes. ठीक है? Tail will provide the motility. Middle piece will have the energy. So head will have the genetic material. So yes, you can select it. है ना? Or which among is having the 23 chromosome? मतलब which one is haploid? Of course, secondary oocyte. Understood? Understood? And now this one, the vas deferens receives ducts from seminal vesicle and opens into the urethra as. So can you see that these are the PYQs? Can you see that these are the PYQs? So if you can see that these are the PYQs, so you know that you are not allowed to miss, right? Because sometimes some different kind of questions they ask. So you are not allowed to miss what? This part. Chalo, fill this. Fill this. Try to fill this. Quick. Quick, guys, quick. Yes, please, anybody? Give it a try, guys. Alright, you have no need to select. You have to tell the function, right? The female reproductive organ associated functions are given below in column A and B. Fill the blank. So pregnancy, just say ovaries, they'll do the ovulation. Oviduct, right? In the oviduct, even the fertilization also occurs. Okay. So fertilization So vagina will result in the birth. So what should be there in the A and the B part? So solve it and tell it to me. Okay. So now let's discuss about the mammary glands. Okay. Now let's discuss about the mammary glands. Okay. So all these diagrams done in detail, we have covered it. So if you will just complete these notes now, you are not going to get anything beyond that. Okay. We have covered everything in detail. Trust me on that. Even the NCERT reading is also done and dusted. NCERT reading, handwritten notes, all the things are done. So female reproductive organ done, the diagrams done. So this is the diagram in the NCERT. You should know the parts. So sometimes students get confused that fimbriae are the close to the ovary, but no, fimbriae are not even considered as a true part of the ovarian duct. It is just that infundibulum is forming these finger-like extensions. Okay, so this part should be clear to you. Okay. Oh, sorry. Then comes this endometrium, undergo cyclic changes, done. External genitalia also done. So this is the mammary gland part. So nipple, the darkly pigmented area is areola. This yellowish structure that in the surrounding you see, this is the adipose tissue, fat tissues. Look at the ribs here, and this is the pectoralis major muscle. So do remember this. Now here, right, you people can see this. This single duct is the lactiferous duct, then ampulla, mammary duct, alveolus or lobes. So this is what we have to discuss, students, right, beta? So again also here also the NCERT will work, okay? Here also the NCERT part will work. And wherever you guys are having any doubt, you can just put that in the chat section. I will definitely resolve it later, okay? Okay, done, students? Chaliye, so let's discuss about that. So basically, what will you see? Look at the diagram. First of all, you have the nipples from where when the baby will give the suckling stimulus, the milk will come out. This darkly pigmented area is what? This darkly pigmented area surrounding is areola. Can you see the lobules? So here we will talk about the alveoli, right? Their clusters are going to make it. So this is something that we have to read now, okay? Right, this is something that we have to read now. You know that in the case of females, right, the mammary glands are well developed, right, and even after the childbirth, the milk production, that is lactation, will start under the influence of the hormones. This is what we know. Under the influence of the hormones, this milk production will start there. This is what we know. Now when you talk about the structure here, you will see that there will be many alveoli there, or we can also call them as acini. There are many alveoli. You can call them as a group or the cluster of alveoli, right? This is the cluster of alveoli or the group. So something like this, right? Something like this. So this is a kind of cluster. So these are the many alveoli. So students, remember one thing, right? Your anterior lobe of pituitary after childbirth. I'm discussing it here. We will discuss it later as well. So your anterior lobe of pituitary, right, after childbirth. After when? After childbirth, right, releases prolactin, right? So that prolactin acts on these alveoli and they secrete milk. Okay, they are going to secrete what? They are going to secrete the milk. So that is what you have to keep in your mind. So many alveoli or acini are there, right, and together, जैसे many alveoli or acini are there, जैसे there are many groups, many lobes are there, many lobes are there, many lobes are there, right? There are many lobes. Is that clear? Right, there are many lobes. Is that clear? Is that clear? So you can simply say that first of all, there are many alveoli or the acini, right, students? Right, students? So, right, they form what? They form the mammary lobes. And there are around 15 to 20 mammary lobes. This is 1, 2. So here you will have many alveoli again will form it. Many alveoli will again form it. So as I said, so 15 to 20 mammary lobes are there. Is that clear? Is that clear? Right? Sure. So prolactin, pro means production, lactin. So acts on alveoli, they secrete milk. right? So in its cavity that milk will be stored. If let's suppose this is the alveoli, so in this cavities, right, in this cavity the milk will be stored. So this point is clear, everybody? Yes, please. This point is clear, everybody? Right, now there are many alveoli, there are many mammary lobes, then obviously there will be many mammary tubules as well, right? Yes or no? Yes or no? Yes or no? Right? Right? So basically many mammary lobes, so that is why, right, that is why there are many mammary tubules, because all these alveoli, they will open into the tubules. So now these tubules, they will join, right, many mammary tubules, they will join, they will form many mammary ducts, right? These many mammary tubules, they will join, they will form many mammary ducts. So basically it's like alveoli, their cluster will form the lobes. 15 to 20 mammary lobes are there, right? Then all that alveoli will finally open into what? They will open into the tubules. So that tubules will combine and they are going to form what? They are going to form the ducts. So all that ducts will finally open into one mammary ampulla. Ampulla, you know, na, kind of wide structure, and then that will open into one lactiferous duct. So whenever baby will give the right suckling stimulus, then what's going to happen? When baby will give the suckling stimulus, then then oxytocin will be released. That is the milk let down hormone, and the milk will come out from this lactiferous duct. So make this flowchart first, then we'll discuss it next. Quick. Sure? So that is what you have to remember here, right? So this is the diagram. So diagram should also be clear. So let's revise it once from the NCERT, okay? So functional mammary gland, as I said, right, they are paired structure that contain glandular tissue. So if it is glandular, means it is secretory. Is that clear? And yes, the amount of fat is there. So girls, when they attain the puberty, the estrogen releases na, so estrogen acts on the mammary gland, the tissue, this fat formation also increases. That is why the size of breast also increases. So it is because of this estrogen, right? So, and it is also advised, students, that one should not take the packed food, right? So because that plastics, they contain the bisphenol A that mimics the role of estrogen, right? That is why the fat production induces. Even such kind of injections are also given, right, to some girls in some regions, so that they, you know, grow fast, their body grows fast. Mentally they are obviously childish, but their body, they grow quickly. Such type of chemicals are given. So bisphenol A, it is present in that plastic, right? If it is not of good quality, then that plastic, from that plastic, that bisphenol A can leach into that food, and if you keep on taking that food, that is why we also say na, the person get fatty when we eat food like this. So it is not just because that is the fast food. Bisphenol A is also the reason there. Okay guys, so now the glandular tissue of each breast is divided into 15 to 20 mammary lobes having cluster, means group of cells called alveoli or SNI. So cells of these alveoli secrete milk, and you know that which hormone is mainly responsible? Prolactin. Progesterone also during pregnancy act on the breast, help in, you know, preparing them for this milk production. Then alveoli open into the mammary tubules. Tubules, they will form mammary duct. Several mammary ducts, they will form wider ampulla, that is single, that is connected to the lactiferous duct. So this is all about this topic. If you guys are still having any doubt, then please ask me that. Okay, because the next topic that we are going to start is we'll discuss about the perineal secretion and the insemination. So do you need break, or can we just work without break? Chalo, one more thing. Do you know how the estrogen acts? There are many roles of this estrogen, right? There are many roles of this estrogen. Jaise it causes endometrium. You write it, then I'll think of the break. So it causes uterine endometrium to begin its proliferation. That is the point, right? Then increase, you know that, just read it, FSH receptors on granulosa cells. That is one of its functions, right? That is also one of its functions. So endometrial lining thickenings causes this FSH stress receptors on granulosa cells, right? Done, beta? Okay. And along with that, while causing the pituitary to lower its, because it gives that feedback inhibition, no? Causing the pituitary to lower FSH secretion. To lower what? To lower FSH secretions. Is that clear, everybody? Yes, please. Is that clear? Then the next point is it stimulates granular cells to secrete inhibit as well. Stimulates granulosa cells secrete inhibin. You remember inhibin part? We have also discussed in the case of Sertoli cells. Inhibit that further decreases FSH secretion. Am I clear? That further decreases FSH secretion. Is that clear? Sure. So guys, do read it and tell me if there is any doubt. Done? Any doubts? And progesterone, as you all know, I have discussed it multiple times. So it is basically anti-abortion hormone, maintains pregnancy, right? Maintains pregnancy, decrease myometrium contraction. So because myometrium contractions will decrease, if these contraction increase, obviously it will not let the implantation occur properly, and it increases glandular and adhesive nature of endometrium. Okay, so that's it. So as of now, this is all about this. So rest we will discuss after a short break, right? So you tell me, yes please, you tell me guys that 20 minutes break will work. Then see, we will discuss about the capacitation, perineal erection, capacitation, right? Then we'll stick to the NCRT part. Okay, then you know that then it is not much, okay, because we will stick to the NCRT part only. Okay, right? So capacitation is important, not in NCRT, but it is important. So let's finish it, right? Why to leave even a single topic? Let's finish it. Okay, let's finish it. Done? So do you want break, or if you guys are fresh, we can just stretch it in one go. We'll finish it by 2 then. Or do you need a break? If it is a break, if break is required, then do let me know in the chat section. Quick. Break is required. So break time till 12:20 a.m. So happy morning, guys. See you in 20 minutes. See ya. Phew. Hi everyone, welcome back. How are you all? Sleepy or not sleepy? Yes, please. Hello. Guys, am I audible? Am I audible? I believe it is a yes. Okay. So now let's start the next topic. Okay. Let's start the next topic. So it is the fertilization. But before starting that part, so next topic that we have to start is fertilization. So before that, let's talk about the pineal erection part, right? So already I told you that in the penis, basically what is the point? In the penis, we know that the erectile tissue is there. What is there in the penis, guys? Yes, please. What is there in the penis? Penis is having the erectile tissue. So in the erectile tissue, during the sexual arousal, there will be the blood flow. And I told you it will be under the control of ANS as well, right? Right, it will be under the control of what? It will be under the control of ANS. So when you talk about the pineal erection, right? Firstly, I'll talk about it, right? When you talk about the pineal erection, right? So basically, erection is caused by parasympathetic nerve fiber, right? It is caused by parasympathetic nerve fibers, right? It is caused by what? Parasympathetic nerve fibers. You know that normally this parasympathetic, yes please, normally this parasympathetic nerve fiber, normally what is the case here? See, it releases the, like it will also releases the nitric oxide here, right? So basically the dilation will occur, the blood flow will be there, and that is how the erection will occur. So it is caused by the parasympathetic impulse, right? So it is in contrast to most other parasympathetic nerves, they are believed to release, right? They are believed to release nitric oxide in addition to acetylcholine. Do you know that? In addition to acetylcholine. Done, beta? So, and ejaculation. Okay. So, this is regarding the erection. But when it comes to the ejaculation, it is the function of sympathetic nerve fiber. Okay. I'll write it. So it is due to the sympathetic nerve fiber. So I am not going to explain this mechanism in detail, but I hope this part is clear to everybody. Any doubts? Any doubt? Yes or no? Any doubt? Right? So when the sexual stimulus is there, right, when it is intense, okay, so obviously because of that, right, the sympathetic nerve fibers, they'll just eject, they will just release this, right, the semen part, and that is what, right, so if it will be released in the female genital tract, you know that what is going to happen, it is the insemination. So transfer of male gametes into the female genital tract with the help of the copulatory organ penis is basically the insemination. In the starting itself, we discussed it, isn't it? Isn't it? And now, it is not like that if sperms are there in the female genital tract, then always the fertilization will occur. No, fertilization will only occur when the ovum is also present there. So students, when there is the ovulation, right? Just now we have discussed it. When there is the ovulation, I told you it is due to the LH surge, right? So this secondary oocyte which is released here, right, this secondary oocyte in metaphase 2 arrest which is released here, so it is viable for 24 hours indeed, it is living for 24 hours indeed, and initial 15 hours, right, in the initial 15 hours, matlab they are the best hours for the fertilization, right, initial 15 hours. Is that clear? Initial 15 hours are the best for the fertilization. Is that clear? And see, now it is not like that if semen is released into the, right, if semen is released into the female genital tract, then immediately sperms will go and they are going to fuse that egg. Not at all. Not at all. There is one more important phenomena, that is the capacitation. Not given in NCERT, but trust me guys, it is very important. So it is basically the charging of sperms. What is the meaning of capacitation? Yes, everybody. What is the meaning of capacitation? So capacitation is the charging of sperm. What is the meaning of capacitation? Capacitation is the charging of sperm. So it occurs in the female genital tract. It occurs in the female genital tract. So what is the meaning of this capacitation? See, in the capacitation, basically, the sperm will be prepared so that it can fertilize the, right? It can fertilize the ovum. If the sperm will not be charged, if it will not be capacitated, then it won't be able to penetrate the cumulus oophorus. I told you already, when the egg will be released, students, it will be covered with this cumulus oophorus. Further, you know that corona radiata, zona pellucida, they all are there. Even the cumulus oophorus, the layer of granulosa cells is also there. The layer is also there. So now what is the point that I'm telling you here, that if the sperm is not charged, if it is not capacitated, it cannot even penetrate that cumulus oophorus. Why is it so? Because when the sperm is discharged in the female genital tract, it is in the semen. It is covered with many unnecessary things, like the cholesterol coatings are there, right? Many other proteins are also covering it. So they just hide the receptors which should bind with the ovum. So we have to expose that receptor so that it can just show the binding with the ovum. So that charging has to be done, and then the motility will increase, and finally the sperm will reach where the ovum is, that is in the ampulla region. Okay, so here you people can see the plasma membrane of the epididymal, right? The plasma membrane of the epididymal spermatozoa, it contains a complement of surface molecules, the proteins and the carbohydrates, illustrated here. Matlab here they are covered, the receptors, they'll be covered, they'll having many proteins, the carbohydrate. So these surface molecules in this epididymis sperm, they are coated with seminal plasma protein. Matlab epididymis is providing some secretion, seminal plasma is having that. So they are covering the molecules, the receptors present on this sperm surface. So when sperms are exposed to female genital tract, these seminal plasma coatings along with the surface molecule, they'll be removed so that they can just expose these portions, and that is how the charging of sperm will be there. And obviously there is the influx of, right, there is the influx of ions as well. So when we are saying capacitation, so it is the set of physiological changes. What is it? It is the set of physiological changes that occurs in sperm and make it fertile to fuse ovum. So that is what the capacitation or the charging of sperm is. Is that clear? So they are the set of physiological changes due to which sperm will make it make it fertile, or I would say make it competent to fertilize ovum or to fuse ovum. Competent will be the better word here. So let's write it. So this is what the capacitation is. So where will it occur? So obviously it occurs in the female genital tract. Calcium ion is very, very important for this, right? What is very important for the students? Calcium ion is very important for this. So you can just write down. So capacitation will require your bicarbonate, serum albumin, or the calcium ion. Capacitation require serum albumin, calcium ions, very important for the fertilization, and HCO3 negative, that is your bicarbonate ions. Is that clear? Is that clear? So what is very important, right? What is very important? Your calcium ions. Is that clear? So during charging, calcium will enter and your potassium will leave. Is that clear? So I am writing it here that in capacitation, calcium enters, potassium leave, right? Calcium enter or potassium leave. So that is going to happen during the charging, right? So the pH changes, all the cholesterols that are covering it, they'll be removed, right, Bacha? Yes, Vishesh, what do you want to ask? You can just simply type your question in the chat section, okay? Okay? Done, beta? Sure? Are you sure? Any doubt? Any doubt? Yes, please. So I don't think that there is any other topic that I have missed. Okay, so not capacitated sperm can held in humulus. Okay, now you know which layer it is. Okay, so in a capacitated sperm or in a charged sperm, intracellular pH increases. Even the cholesterol is removed from sperm surface so that receptors can get visible. Are you getting my point? Okay, so that receptors can get visible. Is that clear? Is that clear? So there is the activation of many other, right? There is the activation of many other pathways, right? So that is not required for your class. So that is why I will not discuss it, okay? Okay. Sure. Done? Chali. So this is what you have to remember. So the next part is what? Next part is the fertilization, right? So when it comes to the fertilization, there are certain steps. So in our case, do we have the internal fertilization or the external fertilization? Tell me. Tell me. What do we have? External fertilization or internal? Yes? Internal or external? Guys, please tell me, what do we have? External or internal fertilization? Quick. External or internal fertilization? Internal. Thank God you know that. So very first thing is the chemoattraction. So chemoattraction of the sperm to the egg, right? So chemoattraction of the sperm to the egg, right? By the substances released by egg. So yes, it is true, right, that there is the, okay, so there is the chemoattraction in between these two. That is why, right, this fertilization is also species specific, that the species of the same, the organisms of the same species, right, their gametes can fuse. Okay, their gametes can fuse. Is that clear? Is that clear? So this is what we have to remember. Now the next step is, so the fertilization will occur. How will it occur? It is not like that the sperm is charged, it will directly penetrate or no. No, we have to talk about the acrosome reaction. We have to talk about the cortical reaction. We will discuss about the how can we prevent the polyspermy. So second point may it will be ki binding of sperm to the zona pellucida, to the extracellular. So then comes the binding of sperm to the extracellular envelope. Jaise you can consider the example of zona pellucida of egg. So this is going to happen. And in the third step, I'll talk about the 5 steps here, right? And in the third step, there will be the exocytosis of the acrosomal vesicle to release its enzyme. Is that clear? So what is this? The acrosomal reaction in simple words. So there will be the exocytosis of the acrosomal vesicle. So this will happen, and then you know that the sperm is going to, because there is the exocytosis of the acrosomal vesicle, means basically what is going to happen, students? So basically you can say that the enzymes will be released, right? So after that the sperm will pass through the extracellular envelope, and finally the fusion will be there. So sperm pass through extracellular envelopes of egg, then the binding of sperm and egg membrane occurs. Is that clear? So this is going to happen here. So binding of sperm and egg membrane occurs here. Am I clear, students? So that is how the fertilization is going to be. So these are the 5 steps. In some organisms, the second step becomes the third one. So this is how it works. Is that clear? Is that clear? Right? So vaginal secretions are very important. That will make the sperm highly active. Okay, so the capacitation occur. Okay, so fertilization may, this is what we have to read. So this part is clear. Firstly, tell me. This part is clear. Sure? Everybody? Are you sure? Are you sure? So what is going to happen? And so these are the steps that has to be followed. And other than that, in fertilization, right? Firstly, we will discuss about the syngamy. Very first thing that we have to discuss is syngamy. So in the syngamy, there is going to be what? There is the fusion. See, like there is a difference in these terms. So syngamy mein the very first thing is there will be the union of male and female gametes. This is going to happen first, students. There is a union of the male and the female gametes. Then there is the plasmogamy, right? Basically, there is the intermixing of their cytoplasm, right? There is what? There is the intermixing of their cytoplasm. And finally, students, there is the amphimixis, in which what's going to happen? Their pronuclei of gametes fuses, right? So it is the pronuclei of gamete that fuses. Am I clear? Am I clear? Right? So pronuclei of gametes fuses. Sorry, this is going to be your karyogamy because it is the nuclear fusion. So this is going to be your karyogamy, and finally comes the, right, the nucleus will fuse, and finally comes the amphimixis. So I'm just telling you the difference in these terms. So when it comes to the amphimixis, so in the case of amphimixis, you will see there will be the intermixing of what? The genetic material of chromosomes. So that is how we are going to see this. Okay, that is how we are going to see this. So basically, when you talk about the fusion here, the fertilization here, the acrosome reaction occur, the enzymes, they'll pass the content. So we'll talk about that. What do we have in the acrosome? So basically, see, acrosome, I told you, who has formed it? Golgi apparatus. So there are many enzymes in the acrosome, my dear students. We call them as lysins. Just say you will talk about the zona lysins. You will talk about the acrosins. You will talk about the hyaluronidase enzymes. Firstly, you understand it, then we will write it. Just say hyaluronidase enzyme, right? Hyaluronidase enzyme. So you know that we have a cementing material. Just say this is one cell, this is another cell, this is another cell, this is another cell. Who's holding it together? You know that hyaluronic acid, right? Even in the skin products these days, they have given so many ads regarding this hyaluronic acid, right? That many skin products, they use it. Okay, so that's why the wrinkles should not be there. I hope you know, you have seen that, right? Hope you guys have seen that. So when this is the hyaluronidase, so it will just break this so that sperm can find its way through these cells. Then obviously zona lysine is there, right? Then there is the acrosin, right, which will act on that acrosin or zona lysine, or even the corona radiating the corona radiating enzymes are also there, right? So that all the layers can be penetrated. So this is the primary binding when you talk about now. This is sperm receptors and the ovum. So it is with the zona pellucida because it is made up of glycoproteins. I'll show you the animations as well. Okay, I'll show you the animations as well. This is not regarding this, just showing you this, right? Just say sperm nucleus is coming, acrosome is there, right? The acrosomal reaction will be there. But see, this part, I think this is of, yeah, I believe this part is basically for the, maybe this is for the jellyfish. Chalo. So you can just simply see, you can just get an idea of it. Say acrosome is coming, so acrosomal content will be released. So enzymes will make the sperm to pass through these layers. Finally, this is how it is going to pass. You guys can see that. Then I'll show you the animation here. But before showing you the animation, I would like to discuss this part from the NCERT. Because guys, if we go with as per the NCERT, na simple NCERT translation, then to it will finish like this. And from this part, they don't even ask in the detail. But whatever is required for your exam, keeping 2025 in mind, we'll discuss it accordingly. Okay, beta? So here you can see during copulation, that is coitus, semen will be released by penis into the vagina, insemination done. These sperms swim rapidly, passes through the cervix. I told you before that the capacitation is required, right? And even the prostaglandins present in the female genital tract, they also help with the contraction of the female genital tract so that, you know, sperms can pass. Then they reach the ampullary region. Ovum released by the ovary is also transported there. The fertilization takes place. So yes, it's a PYQ that where the fertilization occur. Told you multiple times. Now, it will only occur when ovum and sperms are transported simultaneously. That is why the copulation leads to the fertilization and the pregnancy, number one. Then fusion of sperm with an ovum is fertilization. Okay, so during fertilization, sperms comes in contact with zona pellucida. So this is the primary egg membrane, my dear students. This is the first statement that you have to remember. Secondly, this is acellular. This is made up of glycoproteins, right? It is having ZP1, 2, 3 proteins like that. So it induces changes in the membrane that block the entry of additional sperm. So they have kept it very simple in the NCERT. They are simply like, once the sperm reaches the ovum, right, it binds with it, the membrane permeability changes. That, like the way we discuss in the neural control and coordination, right, the influx of ions and all will change it. So same is the case here. So this is a slow block to polyspermy, right, that other sperms will not be able to penetrate because the membrane polarity has changed. So it ensures that only one sperm fertilize an ovum. So this is basically the monospermy, right? It is not, and why it is important? Because single sperm, single ovum, obviously 2n will be there. If multiple sperms will start fusing, so how the diploid number will be maintained? It will not be maintained. Is that clear? Right? So zona pellucida is there, plasma membrane is there. So see, after the fertilization, there is the completion of meiotic division of secondary oocyte, another PYQ. So I'll firstly discuss it simply from the NCERT so that guys, you can just understand these basics, okay? So second meiotic division is also unequal. One more polar body will form and the haploid ovum is now called as ooted. Already shared the flow. Then students, the haploid nucleus of the sperm and the ovum will fuse. You will get the zygote with 46 chromosomes. And how the sex of the baby will be decided. If the sperm is X, see, female will always form the gametes having X only. If sperm is X, XX, female. If sperm is Y, XY, male. So it is the male who's going to decide the gender, right? It is the male's gamete that is going to decide the gender, okay? Okay, so mitotic division starts as the zygote moves through the isthmus of the oviduct called the cleavage. So guys, the cleavage in the mammals is the slowest, right? It is the slowest cleavage of the animal kingdom. Here it starts around after the 30 hours of the fertilization, where zygote will form 2 blastomeres, then the 4, and so on. That is how it works, right? So zygote, it will divide, it will form 2 cells, 4, 8, 16 blast stage, right? So morula, then blastilla will be there. So embryo with 8 to 16. You guys can see this, A, B, C, D. A, This is egg. B is showing the fertilization. C is showing the two-celled zygote. So initially when that zygote is growing, initially the cytoplasm will not increase. Why? Because students, it is the cleavage that is going on. What is going on, people? It is the cleavage. So cleavage may there will be no G1 and G2 phase. Okay, so you know that in G1 and G2 phase, cytoplasmic increase will be there. There is only S phase. So DNA duplication is there. Like when the zygote, when it is dividing into two cells, both are diploid. The DNA content will be divided, but the cytoplasmic increase is not there. Right? The level of cytoplasm will not increase. So that is why in the initial divisions, you won't see an increase in the size, right? Around some initial divisions, they don't show this increase in the size of the zygote, and later on it will be, because cleavage is having no G1, G2 phase, only S phase. That's why it will go on rapidly. Oh, sorry. Then see blastula. This is the blastocyst. Finally, that blastocyst will get implanted. So this is important, right? This is blastocyst. This layer is the, I'll discuss it in detail, don't worry. Trophoblast layer. This is your inner cell mass. The cells which are in contact with this inner cell mass, we call them as cells of Rauber. R-A-U-B-E-R. This is your blastocele. Okay, what is it? It is your blastocele. So that is how it works. Am I clear? So it will get implanted. So here that implantation part is very, very, very easy. But yeah, we will discuss it a bit, right? So I'll show you. I have kept it for all of you. So I'll show you an animation here. Okay. So see, so many sperms are there, right? And then so you people can see, see, they are moving towards the uterus. Millions will trying to make it through the mucus. Slowly they are going. I hope you all can see. Yes. Then you people can see that they are finding their way, right, to the ampulla, right? Prostaglandins are also helping. After the charging only, right, this is how they are going to move. Okay. Yes, even the WBCs there are going to kill it. So now see, they are moving, they are finding their way. See this. So finally, you guys can see this. So that's how it is trying. So now I'll stop it a bit here. You people can see this part. See, sperm is trying to enter, right? The acrosome reaction will start. So inside the zona pellucida is a very narrow type way, just outside the egg cell membrane. So you see this, how is it moving? You see this first, then I'll explain. Okay? See, other sperms cannot penetrate because the membrane polarity has changed. Okay? So the fertilization membrane is impenetrable here. Can you see that? Further sperms are not able to bind there. Okay? So So that is how it is. So this is the animation. Just I was just checking out. Even in the case of embryonic development also, we'll discuss it, right? Done? Chali. So let's discuss that. Right, so basically what's going to happen, the sperm, I'll write it in a very simple way, right? So now you know that the sperm is charged, isn't it? Isn't it? So sperm, what will it do? It will penetrate through corona radiata. Okay, because it is charged, so it will pass through that cumulus oophorus, hyaluronidase enzyme will help, right? So it will help. So corona radiata by a hyaluronidase enzyme. Now I'm writing it, you can also write it, right? So hyaluronidase enzyme, because it will dissolve that cementing hyaluronic acid. I hope you are writing it. Yes, please. So sperm penetrates through the corona radiata. How? By the hyaluronidase enzyme, right? Then acrosome reaction will occur. So first of all, this enzyme, it dissolves what? It dissolves hyaluronic acid. It dissolves hyaluronic acid. This is the first thing that it is going to do. It is going to dissolve hyaluronic acid. Is that clear? Is that clear? Right? Yes. So it cements follicle cells. Okay. So it is going to dissolve it. This first part is clear. The enzymes are coming out. This is what it is going to do. Yes, please. Yes, please. So it is an acrosome, obviously. Then comes your zona lysine. Then zona lysine also known as what? It is also known as acrosin. Now what this zona lysine or acrosin will do, right? So basically, beta. Yes, please. Yes, please. Up to this part all done. So after this, the sperm, okay. See, the sperm attaches to zona pellucida, right? So I told you this is the primary binding, right? So it will attach to the zona pellucida, right? It is having some receptors. Primary ZP1, ZP2, ZP3 receptors are there. Firstly, it is the ZP3. So basically, it is the initial binding is the primary binding, right, beta? So a sperm attaches to the zona pellucida, right? Zona pellucida. So sperm bind to sperm receptor here, right? So when sperm binds to, just a minute, guys. So first, it will dissolve hyaluronic acid. The sperm will attaches to the zona pellucida. When sperm binds to the receptors of zona pellucida, it initiates acrosome reaction, right? It initiates acrosome reaction. Are you getting my point? It initiates what? It initiates the acrosome reaction. Is that clear? Is that clear? So acrosin enzyme is released. So after this, what will happen? Acrosin, which is also known as anybody, zona lysine or sperm lysine, is released. So now according to you, what this enzyme will do? Yes, people. According to you, what this enzyme will do? Anybody? What this enzyme will do? Yes? Please tell me. Yes, please. Anybody? What will it do? Like acrosin or the zona lysine is released and help sperm to do what? To facilitate its way through zona pellucida. Is that clear? So initially there is a binding only. So after that binding, the acrosomal reaction will occur. right? After that binding, the acrosomal reaction will occur, and after that acrosomal reaction, what is going to happen? This enzyme will help the sperm to penetrate or to facilitate its way through zona pellucida. Then finally, the sperm and the egg membrane will fuse, and the sperm will be phagocytosized by the secondary oocyte, right? Then sperm and secondary oocyte egg membrane fuses. Syngamy will be there. Then sperm is phagocytes by the secondary oocyte. Right now, by this time, now you know that this is basically the fertilization, right? Right now, basically, you will see that there is the completion of meiosis 2 and the second polar body will again form. So write down up to this part, right? Write down up to this part, then we will write it further. Quick. Right, so here I have written a sperm and the secondary oocyte egg membrane fuse. So this is important. The sperm egg head and the oocyte will fuse with each other, and then they will form a single cell. Okay. Done? So sperm is phagocyte by the secondary oocyte, then it completes its second meiotic division, right? Then after that, what is going to happen here, students? It is going to complete its second meiotic division. Is that clear? And the second polar body is expelled out. Am I clear? Because obviously, again, second polar body will form because the division is unequal, right? So here, mature ovum contains, yes please, so mature ovum, it contains what? It contains obviously 23 chromosomes. That is the haploid number, yes or no, everybody? That is the haploid number, yes or no? Yes? Right? So on entering, because on entering the ovum, or I would say the secondary oocyte, this penetrating sperm has swelled up. Your phagocyte sperm has swelled up. Okay. This is also going to happen here, right? It has swelled up, right? And its proximal centriole, because I told you already, right? It is having two centrioles. So its proximal centriole. What will happen to its proximal centriole, my dear students? So its proximal centriole enter the ovum, right? Its proximal centriole enter the ovum, right? And then the meiotic division complete. Okay, so here I have mentioned it complete, but how? Because on entering the ovum, this penetrating sperm has swelled up, its proximal centriole enters the ovum and help in completing division. So I told you already, and if not, do write it. This egg, it doesn't contain its own centriole. Okay, right? It doesn't contain its own centriole. So centriole is present. Okay, so centriole, where is that centriole coming from? It is coming from the egg. Is that clear? Is that clear? So mammals may, in the case of mammals, beta, in mammals, the complete sperm enters ovum. Is that clear? The complete sperm enters ovum. So these are few points which I believe you people should know. Okay. Okay. So sperm nucleus absorb water from the egg cytoplasm, will enlarge, and then it will become the male pronucleus. Okay. Noted? So it will become male pronucleus. Noted? Yes, please. Noted? So that is how it is. So you know that only single sperm should fuse here. So for that, acrosome reaction and the cortical reaction will occur. Okay? Done, guys, or not? Sure. Okay. So basically, acrosome reaction plus cortical reaction prevents polyspermy. Okay. They will not allow the multiple sperms to fuse. They'll promote the. So that is what you have to mention. Okay, chalo. So cortical reaction may what is going to happen? See, this is the egg membrane, na? This normal, and here this is the perivitelline space. So here you see there are some cortical granules, students, right? They are also made by the Golgi apparatus. They are also present. So by the time, you know what, when the first sperm reaches there, what is going to happen? The calcium ions, right? They diffuse into egg. Do you know that? They diffuse into this oocyte. Yes, how many of you are aware of it? So calcium is very important for this process. So within a few minutes, sperm penetrates, right? Penetrates the zona pellucida of ovum. The calcium ion diffuses inwards. Okay? Okay? Done? So cortical granules are present below this egg membrane. So by the time sperm penetrate zona pellucida, okay, so the calcium ions diffuse into the oocyte. Okay, oocyte. Done? Okay. Yes, please. So calcium ion diffuses into the oocyte, due to which these cortical granules, right, they are present below this plasma membrane of the egg. So these cortical granules present below plasma membrane diffuse with this egg membrane and pass their content into perivitelline space. Okay. So it will pass its content into perivitelline space. So if you are not aware of it, let me tell you, this is the normal egg membrane, cortical granules. Here comes your ZP, and there in between these two, you have the perivitelline space. So when it will pass its content there, you know, a kind of impenetrable membrane will form. So the further sperms will not be able to bind with the receptors of the zona pellucida. Is that clear? Is that clear? So I'm just writing it here. So further sperm can't bind to the receptors of zona pellucida. That's how it works. So primary binding will not occur here. So write down. Quick. Write down. Done? Done, beta? So that is how the fertilization occur. So then there is the formation of the zygote. So you know that zygote, right? So already discussed it with you all. So what is the site of fertilization? So site of fertilization is, right? Site of fertilization is ampulla. That is again a PYQ. So do remember this. Okay. So now it takes 5 days for this zygote. It takes 5 days to reach in the uterus and 7 days for implantation after fertilization. Imagine. So yeah, this is the information you need to know. So let me discuss that with you all, then we'll write it. See what's going to happen. As I said, it is the slowest cleavage of the animal kingdom. The zygote will divide after 30 hours. So this zygote will take around 5 days to reach the uterus or the uterine cavity, and it will take around 7 days for the implantation. So basically after 5 days, like let's suppose today fertilization occurred. So after 5 days, it will reach the uterine cavity. It will survive there for 1 or 1.5 day, and then it will just feed on the secretions of the endometrium there, of that uterine milk there, because by the time the progesterone is also high, the glandular nature of endometrium is there. So endometrium will keep on releasing the things. I'm not talking about its layers now. So that will provide the nourishment to that zygote, and it is still there in the zona pellucida. It's not like that it will came out of the zona pellucida. Zona pellucida is still intact. Why? Because that zona pellucida will ensure that ectopic pregnancy should not occur. Zona pellucida will ensure that this egg should not bind or implanted to any place other than uterus. Are you getting my point? So that is how it is going to work, right? So the different stages of the development will be there further. So here you guys can see, right, if the ovum is fertilized, then you know that next is gestation or pregnancy that will occur, and this is what we are talking. Okay, this is what we are talking. So when you are talking about the gestation period in humans, and you need to know the gestation period of rabbit, gestation period of elephants. So in the case of humans, so it will take 266 days, that is 38 weeks, or 280 days, that is 40 weeks. So this is about the gestation period in the case of humans, right? So development from fertilization to the birth it is, right? That is fertilized ovum to the, so it is development from fertilization to birth. So this is what the gestation period is. Is that clear? Is that clear? Yes or no? Yes or no? So if the last menstrual period is considered as the start of the gestation, then you consider it as the 280 days. Okay? Okay? Done? Sure? Are you sure? So just look at this diagram. So here you guys can see, right? You can mark it in the NCERT as well. So here you people can see. Now you guys can see. We have discussed the extra point. They are needed. So you can see the fertilization occurred here. Where? Ampulla. Okay. Then this is the 2-cell stage blastomere. So animal. Sorry. So yes, mammalian cleavage, I'm not just talking about the human, I'm talking about the mammals, is the slowest cleavage of animal kind. Is that clear? It is the slowest cleavage of animal kingdom. Okay, this is the slowest cleavage. Done. So, and here when you talk about this fallopian tube and how will it reach, as I said, it will take 5 days because slow movement is there. Fallopian tube is having ciliated columnar epithelium. That is again a PYQ. You can just write down. Okay, so that will help in its movement. So after 30 hours, this cleavage will occur, right? 2-stage blastomere, then 4-stage. So initially the size is not growing, if you people notice. Initially the size is not growing, if you people notice. Yes or no? Yes or no? Quickly tell me. Yes or no? Right? And guys, we didn't write the slow block to polyspermy, that is change in the polarity, right? You people can write it. Okay. Sure. So you can just simply write this first. So slow block to polyspermy, you can remember. The fast block of polyspermy is the change in the electrical potential. So you can just write that down. Slow block to polyspermy is your cortical reaction or the acrosomal reaction, having enzymes and all. So you just check it out. If there is any doubt, then please do let me know. Is it done, students? Sure? Okay, so let's discuss the next part. Just allow me 1 minute. Okay, so now these divisions, the cleavage is going on, right? So zygote, that will divide. This is the 2-cell stage. So which division is going on first? It is the cleavage. So as I said, the slowest one. So here there will be no G1 and G2 phase, right? So no cytoplasmic growth initially. Okay, no cytoplasmic growth initially. Okay, so S phase is there, definitely that will just double the DNA. Now I will add one more point here, beta. You know what? Just say normally what happens that these zygotic genes, they didn't start the transcription, they didn't start the protein formation. So the oocyte, right, the genes of the oocyte, they takes the control, but not in the mammalian cleavage. In the mammalian cleavage, that control will be there with the zygotic genes only, right? So initially when there is the, right, initially what is going to happen? Initially cytoplasmic, right? Initially what's going to happen, people? Initially cytoplasmic volume doesn't increase, right? Cytoplasmic volume doesn't increase, right? The cytoplasmic volume doesn't increase, right? Cleavage of nuclei occurs at the rapid rate. So what is going to happen? Cleavage of nuclei occurs at rapid rate. So this is going to happen in the starting part, people. Okay, this is going to happen in the starting part. So nuclear to cytoplasmic ratio increases, but cytoplasmic to nuclear ratio decreases. So nuclear to cytoplasmic because nuclear number is increasing now. Ratio increases, but cytoplasmic to nuclear ratio decreases. Am I clear? Am I clear? So this is going to happen for the initial phases, right? So that is why it is going to happen. So you guys can write it down as well. Okay. So after the 13 cycles of division, right? After the 13 cycles of division, then you will see, though in the mammals, so initially the zygotic genes take the control, then you will see the cytoplasmic growth is also there, right? So around after, you can say that after, it can vary, after 13 cycles of division, somewhere you will see the G1, G2 phases introduced. So when they are introduced, then obviously guys, what is going to happen? Cytoplasmic growth, and hence the size of that zygote will also start increasing. So this is the initial stages. Okay, this is about the initial stages. Any doubt? Any doubt here? So first cleavage is going to be meridional. Okay, 2 blastomeres will be there, and other will divide equatorially. So that's what you can just remember. So like, let's suppose first division, this is like this, meridional. And then after this, second will be like this, equatorial. So yes, you can say that mammalian cleavage is rotational, right? Mammalian cleavage, it is going to be, students, rotational. And remember one thing, in humans, the fate of these blastomeres, right? The fate of these blastomeres is not fixed. So it is the indeterminate cleavage. Like, it's not like that. It is not like that, students, that initially, right, it is decided that this part of blastomere will form the leg, or this part will form this cell or that cell. No, it is not like that. Any blastomere is, initially these blastomeres are totipotent. They can grow into the any part. So that is why, right, just say we talk about the twins. So we talk about the maternal twins and the fraternal twins now. Yes or no? When we talk about the twins, we talk about the maternal or the fraternal twins, if you remember. Yes, everybody. If you remember, you people can see this, right? You people can see this. So identical and the fraternal twins, matlab one egg twins, identical they are, maternal twins they are, two egg twins, fraternal twins they are. So one egg twins may, just let's suppose if let's suppose after the formation of zygote, when the blastomere forms, if that blastomere gets separated, it can develop into a completely new organism, new individual. Why? Because in that case, what is the point? What is the point? The blastomere is totipotent. So such twins are mono, like single zygote, and their sex will be same, right? So if one is male, another one will also be male. One is female, another one will also be female, because that blastomere is developing. But sometimes what happens, two eggs are there and both of that get fertilized. Then in their case, sex can be same or it can be different. Is that clear? Is that clear? I hope it is clear. Right? I hope it is clear. So this is about the cleavage and the division part here. Yes, students. So it is the blastocyst that will get implanted. Okay. So it is the blastocyst that will get implanted ultimately. It is the blastocyst. So in the case of blastocyst, yes. So I will not dive deep into that as we are running out of time as well. So let's target the NCERT portion here mainly. Let's suppose if this is the part, okay? So I told you already, these are the trophoblast cells. Okay, trophoblast cell layer. It contains, students, proteolytic enzymes, right? It is still in the zona pellucida yet. It is not hatched yet. It is still in the zona pellucida. Now, here you will see this is the inner cell mass. What's that? This is the inner cell mass. It will form future embryo. That's what you have to remember, right? This layer will form extraembryonic structure. The cells which are in contact, with this inner cell mass, we call it as cells of rubber. And this is what? This is the blastocoele. So your trophoblast cells, they release the proteolytic enzymes. And that will digest the zona pellucida, then the surrounding endometrial lining, and then it will just implant it into it. Let me show you this part. I have added the wonderful diagrams here. You see this, guys? See, this is the blastocyst inner cell mass that will form the future embryo. Okay, slowly, this is that endometrial lining, right? We also call it as deciduous because it has just swelled up. It is having the stored food. Okay, it is glandular also. So see, it will release, right? So why it is in zona pellucida? So I'm just writing it here. Zona pellucida prevent ectopic pregnancies. Okay, it will prevent what? Ectopic pregnancy, right? It means avoid implantation at any site other than uterus. Right? This is sometimes it gets implanted in fallopian tube, then result in its rupturing and all. So zona pellucida prevents it. So finally, this slowly it will release. See this, the proteolytic enzymes. Slowly it will start. See, it will start forming these structures, right? Extraembryonic membrane. So it will penetrate, and finally when it gets penetrated, now can you see that? It will be covered from the upside. Just say, let's suppose if I have to show you, let's suppose though that it is in the fundus part here, let's suppose if here, just imagine if here you have the implantation, so it will slowly eat it up, then it will just penetrate in it, and then further it will be covered by a kind of plug like this, right? It will be deeply penetrated. Okay, it will be deeply penetrated. Wait, I'll show you this, right? So you can see that. So uterine mucosa will be re-established. This is your endometrial lining. Already told you implantation will occur here. Your inner cell mass then further divide, will form bilaminar disc. It will form epiblast and hypoblast, these two layers. Okay, and slowly around its amniotic cavity will form, right? And here you guys can see the way it is going to develop. I'll discuss it. Okay, don't worry about that. We'll talk about it. So you can see this is the baby. Okay, this is a placenta part, right? So from the embryo side, it is the chorionic villi that takes part in the placenta formation. Maternal tissue is there and chorionic villi. So there is a very intimate connection is there. It is not like that it is going to penetrate the maternal blood supply and paternal. No. So they are like this. So through diffusion the exchange occurs. Okay, through diffusion the exchange occurs. Done, students? So you can see the placenta, the amniotic sac in which this baby is there. So these are the stages of the development. Okay, so you can see that, initially the zygote, the 2-cell stage, right after 30 or 36 hours, then 4-cell stage, after 60 hours, 8-cell stage. Then this is the morula, right? Mulberry-like structure. You people can see that. Mulberry-like structure. Then see this. This is how the blastocyst will form. And slowly the proteolytic enzymes will result in its hatching, and this will form the chorionic villi further, and this is how it is going to penetrate. You people can see that. Okay, you people can see that. I have added the wonderful diagrams. Trust me, the diagrams are really wonderful. Okay, so see, that's how it is going to grow and form slowly. So embryonic membranes are there. We'll discuss about it. So that's how the stages are. You have a look. Okay, You just have a look. So see, this inner cell mass will form the layers, epiblast and hypoblast. Okay, and slowly they will develop, right? They'll further form the germ layers also. So when it comes to the germ layers, ectoderm and mesoderm, endoderm, we are triploblastic, 3 germ layers. They form our organs. Just say ectoderm forms the nervous system, your mesoderm forms the muscular system, like this. And when you people talk about the extra embryonic membranes, so it will be amnion, right? Amniotic fluid, right? Amnion layer, amniotic cavity, it will form having the amniotic fluid. So you have the chorion, you have the chorion that will help in the placenta formation. Chorionic villi are there. Later on, they will just get condensed at one part, just like a disc. Then you talk about the yolk sac, right? You know that yolk sac in the fetal development stages. Initially, it also forms the blood, RBCs, erythropoiesis. It helps in that erythropoiesis. And then allantois is also there, right? It's somewhere its role is there in the excretion, but here it is not that well explained. Okay, so that is what we have. Right, that is what we have. So placenta formation is there, you know that, due to which we just feed on our mother. We are just totally dependent on our mother. So you can see the stages there. So guys, see from the zona pellucida, the hatching is there with the help of that proteolytic enzymes. Then see, these enzymes will help to penetrate this endometrial lining. Finally, this will get pushed into this endometrial. Then there is a kind of plug. It is going to cover it. Slowly, this inner cell mass is forming layers like bilaminar disc, epiblast, hypoblast. They are further growing. They are forming ectoderm, right? Endoderm, mesoderm will further form. Slowly, these trophoblastic layers, they are forming this chorionic villi that will form the intimate connection with the endometrial lining. And see, that's how finally the umbilical cord is coming up, that will be attached there. So that is how the further growth occurs, right? Right, so let's continue this portion from the NCERT part. Okay, so let's write it. So after implantation, finger-like projection appear on the trophoblast chorionic villi. This is a direct PYQ, my dear students. It is a direct PYQ. So chorionic villi will form, right? What will form? Chorionic villi will form. So these villi are surrounded by what? Uterine tissue, right? The endometrial lining, of course, and the maternal blood. So these chorionic villi and the, right, I'll add the extra point, but first I'll cover the NCERT, and the uterine tissue, they are interdigitated like this with each other and form a structure which we call as placenta. So which part? The chorionic villi from the placenta fetus side, and the uterine tissue, you're talking about that endometrial part, from the mother's side. So this is the interdigitated connection. It's not like that blood supplies are mixed. By a diffusion, the exchange will occur, right? By a diffusion, the exchange will occur. Okay, beta? So placenta will facilitate the oxygen and the nutrient supply. It helps in removing the waste as well, right, students? So it is connected to the embryo through umbilical cord. Another MCQ. Okay, another MCQ. So placenta is also the endocrine tissue. So that's a very important question, right? That's a very important question. So which hormones are released by placenta? So first thing is HCG, right? And it confirms pregnancy. Do you know that? It confirms pregnancy. I'll discuss about it. Then human placental lactogen, also known as human somatotropin hormone. Then estrogen, progesterone, right? And it is also released as relaxin, which is even released by the ovary. So blastomeres, they are forming morula. Morula continues to divide, transforms into blastocyst. I think it has just shuffled, right? So blastomere arranged, outer layer is trophoblast, inner cell mass is there. So trophoblast will attach to endometrium. Inner cell mass will differentiate as embryo. That is again a PYQ, right? So a blastocyst will get implanted in the endometrium. This is implantation. Okay, this is what implantation is. So this part is quite easy here. Okay, so see, HCG, HPL and relaxin are all only produced during pregnancy. This is a repeated PYQ, guys. You write down. right? Because relaxin will relax the pubic symphysis so that it can facilitate the childbirth. HCG confirms the pregnancy. We'll discuss it. Human placental lactogen, right? When it was studied, it is also believed that it helps in milk production, but it is having other roles as well. I'll discuss it with you all. So during pregnancy, the other hormones, estrogen, progesterone, cortisol, prolactin, thyroxine, they are also increased. But these three will only release at the time of pregnancy. So this is a PYQ, right? So metabolic changes will occur. So after placenta implantation, inner cell mass, students, they differentiate. Outer layer is ectoderm, inner is endo, and mesoderm will soon appear between these two. So it will do the organogenesis, right? So inner cell mass is having certain stem cells which have the potency to give rise to other tissues, right? So from this part, from this paragraph, multiple times you get the MCQs. Never ever leave this paragraph. So you better underline this, right? I'll finish the NCERT portion. So those who want to leave the class, they can simply leave, right? But I'll add few points here, and those who can stay, they should stay, right? They should stay. Okay, chaliye. So this paragraph, this is very important. I'll underline all the important points here, right? So this is dog, elephants or cats, their gestation period, your homework. So after first month of pregnancy, the embryo's heart is formed. And you know, doctors, this is what they listen and this is what they check. So let me mention it. See, 0 to 3 months, this is first trimester. Like this is the 12 weeks. 4, 5, 6. Second trimester. So do not confuse trimester with the month. Then 7th, 8th or 9th month. This is going to be the third trimester, right? This is going to be the third trimester. Okay. So legally abortion is allowed in the first trimester. And if in certain cases, if it is required, yeah, it is allowed in the second trimester. Then the signature of 2 medical practitioners are required. Okay. So let me discuss this paragraph because it is one of the, literally one of the most important paragraphs here. So heart sound, when will that be formed? By the end of, so after 1 month of pregnancy, embryo heart is formed. So basically after 1 month of pregnancy, you can say the 10th or 12th week maybe. So by the end of 2nd month of pregnancy, right, fetus will develop limbs and digits. So this is what you have to remember. By the 1st month, the heart sound will be heard with the help of stethoscope. By the 2nd month, students, the fetus will develop limbs and digits. By the end of 12 weeks, like first trimester, most of the major systems are formed. Just limbs and external genitalia organs are well developed. We have discussed it today as well. The first moment of the fetus and the appearance of hair, that is again a PYQ during 5th month. So 5th month means 2nd trimester. So by the end of 24 weeks, the body is covered with fine hair, very fine hair, and eyelids separate, eyelashes are formed, and by the end of 9th month of pregnancy, it is ready for the delivery. So this is one of the most important paragraph. As such, you will get the questions from this part. Am I clear? Am I clear? Yes, please. Am I clear? Sure? And you know, na, elephant is the slowest breeder of animal kingdom. Elephant. So here, the gestation period is 18 months to 22 months, right? So elephant is what? Elephant is the, yes please, so elephant is the slowest, right? Slowest breeder. Okay. Okay, so cat ka to very less, it is just 65 days. And what about dogs, guys? What about dogs? So you write it. What about dogs? So cat is 65 days, 18 to 22 months. And do read about the elephants. So what about dogs? Gestation period. So dogs ka gestation period, it is just like, it will be 58 to 68 days. That's what you have to remember. So this is one of the most important paragraphs here. So rest for the parturition or the childbirth, right? The vigorous contractions of the uterus by the end of the pregnancy will cause the delivery, and the fetal ejection reflex, obviously fetus is going to start that. So parturition is a neuroendocrine mechanism, right? So fully grown, fully developed fetus is going to start it, students. Okay, so this trigger release the oxytocin. Told you when I was telling you the mammary glands. So oxytocin will release from maternal pituitary. It acts on uterine muscles, will cause that pulsations, right? The vigorous contractions. So stimulatory reflexes between the uterine contraction and auxin secretion. The contractions will become stronger, and that will result in the expulsion of baby through the birth canal. And you know that for the lactation, milk production is done by your prolactin. So colostrum is formed initially, which is having IgA, immunoglobulin A, that will provide the passive immunity, right, to the baby. Why passive? Because these are the ready-made antibodies that baby will get. So it is rich in vitamins. Okay, so this is about the colostrum. So those who want to leave the class, they can, because now I will be discussing some important points, some extra important points here, right? So from NCERT part, it is done and dusted. We have discussed each and everything, including the exemplar questions today, right? Including exemplar question today. It's just like I'm not well, otherwise so I would have finished it earlier. Yes, please. So anybody having any question, then I'll pick up the extra point. Sure, guys? Sure? Are you sure? Done? Done. So see, you can write the extra points here with me. So I told you now, this is the blastocyst, right? I think this part I have already covered. So trophoblast, right? Because we are also, we don't have much time, so that is why I'm just writing the chorionic, but this is more than sufficient. Chorion. So this will form chorion. Okay, this is what you have to mention. So this is going to form what? This is going to form the placenta. Then inner cell mass, it will form layers. It will form epiblast and it will form hypoblast. Okay, so here you can see trophoblast will not form any embryonic structure. Epiblast, it will form the embryo, even amnion. Hypoblast will form your yolk sac. This is what you guys can write. Okay, done. So it will form the complete embryo or the amnion. So already told you that it is the endometrial secretion that will provide the, right, that blastocyst the nourishment. Okay, okay. So zona pellucida will prevent ectopic pregnancy. That is done. The role of progesterone done, right? Okay. Okay, so next point is what? See, now you know that when a female is pregnant, okay, so the progesterone level is there in the body. Yes, please. This endometrial, the progesterone acts on it, right? Progesterone acts on it, will make it more glandular. So, you know, due to the, so cells here will swell. Then you use the word decidua for it. Do you know that for endometrial cells? Because they have the food in it. They have the food in it. So this is what you guys can remember, because they have what? They have the food in it. Is that clear? Is that clear? Right? So you know what? So initially that blastocyst is going to take the nourishment from it. Matlab even after during the first week after implantation, during first week after implantation, during first week after what? After implantation, embryo obtains nourishment from embryos obtain nourishment from decidua only. Right? Do write it. Embryo obtain nourishment from decidua only. Done? ठीक है बेटा? So 16 days after fertilization, right, placenta starts providing nourishment. So basically you can say that it's like 1 week after implantation. Okay, it is like 1 week after implantation. Done? Done? Do you know that, just let's suppose when there is the implantation, okay, that is how you have the blastocyst now. So these chorionic villi, initially they are all over. Then they will just form a kind of disc-like structure. Like they just get, they are not, they just get collected at one point, right? So then this placenta is also called as discoidal placenta. Do you know that? The placenta is also called as discoidal placenta. Which placenta is there? Discoidal placenta. Okay, and even, right, it is also the interstitial, we also call it as interstitial placenta. Why interstitial placenta? Because it deeply penetrates with the maternal part. It is also known as hemochorial placenta. This is the terminology that we can use for the placenta. Am I clear? So it is also known as hemochorial placenta. Is that clear? So hemochorial placenta on the basis of histology, discoidal on the basis of chorionic surface. So discoidal point is important, right? Because chorionic villi, they will just form a disc-like structure. Understood? Understood? Okay. So on the basis of distribution of villi, it is discoidal. So deciduate it is because there is a, you also call it as deciduate placenta. Why? Because there is close association of, right, close association of chorionic villi and the maternal tissue. Okay, so these are some bullet points that you all can write. Is that clear? Is that clear? Sure? Done? Any doubt? Do you know that, students? Now there are some facts, some interesting facts that we are going to discuss. Do you know that by the end of pregnancy, right, by the end of pregnancy, we see that in the fetal blood, right, when you talk about the partial pressure of oxygen, it is around 30 mmHg. And in maternal blood, your mother's blood, and in mother's blood, do you know that it is around 50 mmHg. So, but still, this is a very important fact. Do you know that adult HB is different from, fetal HB, like fetus hemoglobin is different from adult hemoglobin, right? Fetal hemoglobin is different from adult hemoglobin and can bind to oxygen even, and bind to oxygen even at low partial pressure. So basically, fetal hemoglobin is having more affinity, right, for oxygen. And now some facts, right, some general facts I'll discuss with you all. So just let me, okay. So the point is, basically the point is, जैसे, see, we say that if there is no fertilization, right, you listen to this, we are saying after the fertilization, somewhere around 7 days, the implantation occurs. And we know that the body is still there in the luteal phase. Just let's suppose female is in her, female will release the egg on what day? On day 14, right? On 14th day of her menstrual cycle. If we are considering on an average, it is for 28 days. So on 14th day, she is going to release the egg. Let's suppose the fertilization will occur. So 7 days, right? Let's suppose on 15th the fertilization occurs. So 16, 17, 18, 19, 20, 21, 22. Like it is around day 22 of the menstrual cycle when this implantation will occur. Now, obviously, the female is still there in the luteal phase. Still the progesterone is there. Once it gets implanted, you know that HCG productions also start. Human chorionic gonadotropin. Like that through blood, it reaches to the ovary, right? It reaches to the ovary. That there is no need to destroy the corpus luteum. You can maintain the corpus luteum. Then that corpus luteum will be the, right? Then that corpus luteum will be what? Then that corpus luteum will be the corpus luteum of pregnancy. So that is how it works. So it is the HCG which will be checked in the blood, which will be checked in the urine, right? So it forms during the pregnancy. So your HCG, HPL, relaxin, they forms during the pregnancy only. So it is the HCG that confirms the pregnancy. Human chorionic gonadotropin. Is that clear? Human chorionic gonadotropin. Is that clear? Is that clear? Is that clear? So fetal, right? So next is fetal hemoglobin is, and so that is why, you know, initially, that is why even initially, like by the time the placenta develops, because it will take over the function. So initially, during the initial time of the pregnancy, if the ovaries will be removed, like somewhere in the first trimester, so the pregnancy will not be continued because corpus luteum progesterone is required. Once the placenta take over the function, then there is no problem at all, right? Then there is no problem at all because then placenta will take care of everything. Is that clear? Then what is going to happen? The placenta will take care of this. Is that clear, students? Sure. So interesting now? Interesting now? Done? Sure. So HCG, it helps like that, okay? So trophoblast cells will release your HCG. Any other doubts, students? Done, students? So just keep it in your mind. Now your human placental lactogen, it acts on in lower animals. It also help in the milk production. We also call it as human chorionic somatotropin, right? So basically, do you know that it is also believed that due to this hormone, right, because fetus is growing, fetus is developing, it demands more and more oxygen, it demands more and more glucose. So because of this hormone, you know, the glucose supply will be more to the fetus in comparison to the mother, because the metabolism of fetus is more. So once the fetus is fully grown in the uterus, then fetus is clear that I don't want to spend the time here, right? I want to go out because this place is not able to fulfill my needs. So fetal ejection reflex starts, right? And then finally the mother's pituitary will release the oxytocin. So that's how it works. Relaxin, it will relax the pubic symphysis. You know, the pelvic girdle, two coxal bones, they are attached. Pubic symphysis is there. So that it can relax it, it can soften that. So relaxin will be released by your corpus luteum as well as by placenta. Okay? So by the 5th week, you will see all this. I think even the I don't remember the week exactly. So that is how these hormones work. Even when you talk about the placenta, through placenta, students, through placenta, IgG, it is IgA through colostrum, and colostrum, you write a short note on it. It is basically protein rich, it is having low fat, right? And it is having basically IgA. So placenta will pass IgG. So that will also provide the passive immunity to the baby, right? And you know, it fight with, you know, not teratogens, you know it very good otherwise. So teratogens can cause, these are the agents, chemical agents or whatever agents that can cause the birth defects, right? So placenta also prevents that. Okay, placenta also prevents that. So these are some points related to it. So no doubt in the parturition, in the childbirth, right, just say oxytocin is given, right, to induce the labor pain. Like honestly speaking, students, if I'll get a chance now, so I'll discuss that, right? I'll discuss that in detail with you all, right? That what, during first pregnancy, for how long the labor pain lasts, right? It can last for few hours, few hours to like 2 to 3 hours to many hours as well. It can, right? The breech baby and all. There are many points, right, that I would love to discuss because this chapter is literally very interesting. And in my batch, right, I have taken a decent amount of time for discussing this chapter. Like I have discussed all that hormones, what they work, low hemoglobin, high CO2, and all of that things, right? Even in the fetal development, I told them about the parturition, the childbirth, right? All the position, right? That everything, even I have shown them the videos that what kind of push, how that baby will come out, all the things we have discussed. Okay, but yeah, no doubt it is not the part of your syllabus. It is not given in the NCERT, but overall they are very, very interesting, right? So if I'll get a chance, so I'll keep a special class on an academy platform. So that will not be regarding that it will come in the NEET exam. That will be regarding just for your knowledge, because it's not always like ki yaar nahi ma'am, we just want marks, that's it. So in that way we have to read. But right now your focus should be marks only, because if once you qualify this, you go to the MBBS college, you can be the gynecologist, then you will get to know about it in more detail. So that is why I'm just sticking to the topic. Otherwise in the batch itself, in the nexus batch of mine, right, even I've told them which hormone is working like what, like the human placental lactogen's function, human chorionic somatotropin's function. This hormone is going to decide the time of the birth. That is how it is going to work. But like multiple extra things we have covered there. So now, because we have 6 months, we need to be very focused. We need to complete our syllabus on time, and we have to decode the NCERT. So that is something I have done for you. Prolactin, oxytocin role, which is known as milk letdown. So prolactin will help in the milk production, but when it comes to the milk ejection, so oxytocin will play the role. So I have discussed these points, right? Do not miss anything because I have covered it in detail, right? I've literally covered it in detail. So all I want you people to do is just keep on studying. And the next chapter that I'm planning, and I will take next chapter only if I will get at least 50 comments in the comment section. Yes, ma'am, the sessions are beneficial. Continue that. We will be sincere. If I'm not going to get it, I will not continue. I'll be very honest about it. I am not going to continue the series. I'll focus on my batch only then. Right, so if you really want me to continue the session, then put it in the comment section that yes ma'am, it's all going well, right? We want the sessions like this. So Let's see. So this is all about the today's session, right? I have shown you the NCRT exemplars as well. We'll share the PDF in our official Telegram group, that is Unacademy NEET English. So do check out the NCRT exemplars, okay? Do not miss any question from the NCRT exemplar as well. So I'll schedule a special class on Unacademy platform. We'll share the link in my own Telegram group as well, right? So you guys can check it there and we can discuss it. So you can see the suckling stimulus, it triggers, right? Can you see that? Oxytocin that will come out from the posterior lobe. So that will help in the milk ejection, milk let down hormone test. Okay, this is the flowchart that I have added. And about the parturition also, let me show you. So here you guys can see, right? The onset of labor will be there, and sometimes, you know, false labor pains they feel, right? Okay. So see the flexion there, the rotation of the head, then extension, external rotation of head, and the uterus immediately after birth. So placenta will separate from the uterine wall. So this is the umbilical cord. This is a connection. This is this navel, the belly button. Umbilical cord is attached to this point. So it is a connection between us and our mother. Okay. So I hope you guys have liked the session. We have completed all the details here. So as I said, so I'll be waiting for your comments. So if I will get minimum 50 comments there, yeah, yes, ma'am, the sessions, the minimum 50, maximum should be more than 500, okay? Okay. So then I'll continue it. Otherwise, so I have realized कि नहीं यार, students तो बस, they are not here to study. So if you are here to study, do let me know in the chat section. So take care, guys. See you all soon. So tada.